major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors: Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil.
The theme of the 51st Annual Convention of the PSN is revitalizing the landscape of research and education through microbiology in the Philippines. This morning's pre-convention workshop is hosted by the Philippine Network of Microbial Culture Collections. This is Marian De Leon, and I'm the current treasurer of the PNMCC and liaison officer of PSM, and I'll be your host for today. May we request everyone to kindly direct your attention on your screen for the invocation to be led by Reverend Father Echanes Kadiogan of the Trinity University of Asia, and join us in singing the national anthem to be led by the Trinity University of Asia Chorale.
Let us pray. Almighty and eternal God, we thank you for our life and all your blessings. We thank you for this day you have given our microbiologists under the Philippine Network of Microbial Culture Collections to gather for their Preconvention Day with this learning and sharing opportunity on the trends in microbial identification. We thank you for all attendees and participants, our speakers and hosts, and all involved in the preparation and conduct of this event. As we proceed, we humbly ask the guidance of your Holy Spirit that everyone may make the most of this seminar and be all the more inspired for better service to you in such field. For you are delighted with the work of all who serve your purpose through the example of your Son, Jesus Christ our Lord. Amen.
Bayang Bayang Magino, sa sakit ko'y buhay, lupang nangingiti, sa pag-ibig, sa manlulupig, di ka pagsisihin, sa dagat at bundok, sa simoy at sa langit mo bukha, may dilag kang tula at awit sa paglayang minamahal. Ang isang ng watawat mo'y tagumpay na nagdidinig. Ang bituin at araw na kailang pamay di magdidinig. Lumay ang araw na walang ating pagsinta. Buhay langit sa piling mo. Ang ligaya na pag may mga api, ang mamatay ng dahil sa'yo.
Thank you very much, Father Kadiogan, and to the Trinity University of Asia Chorale. But before we start, may we request everyone to follow the following webinar guidelines. Kindly turn off or put in mute mode your microphone when you are not speaking. Unless otherwise, turn on your microphone when you are about to speak. When someone is in the middle of his or her discussion, kindly use the chat box when you have something to share or to ask. At this point, to formally open the pre-convention workshop, may we request on screen Dr. Ursula Bicol, the President of the Philippine Network of Microbial Culture Collection and the current treasurer of the Philippine Society for Microbiology, for the opening remarks.
Thank you very much, Dr. Marian. We are very glad to have the audience for this day to witness the different methods and recent trends on microbial identification. It is my pleasure to invite all of you and welcome you to this pre-convention webinar of the Philippine Society for Microbiology. Thank you very much.
Thank you very much, Dr. Ursel, for that very short but very sweet opening remarks. At this point, may I now call on the corresponding Secretary and former President of the PNMCC, Ms. Diane Dison, to formally introduce our first speaker.
A pleasant morning to all. Please allow me to introduce our first speaker for this morning. She is the head of the Natural Science Research Institute, NSRI, College of Science, University of the Philippines, Diliman. She is also the head of the Microbiological Research and Services Laboratory, NSRI. She earned her doctorate degree in biology at the University of Houston in Texas, USA. She earned her Bachelor of Science in Biology and as well as her Master of Science in Microbiology, both at the University of the Philippines, Diliman. She is also a diplomate of the Philippine Academy of Microbiology, PAM, Philippine Society of Microbiology Incorporated, confirmed in 2001. Her research interests and specialization in microbiology specifically includes microbial systematics, culture collection, environmental, food and water microbiology, and microbial metabolites. She has received numerous prestigious awards and also has published numerous numbers of researchers, both locally and internationally. She is the founding member and the current Vice President of the Philippine Network of Microbial Culture Collections Incorporated. To talk about the topic, phenotypic identification using Biolog Microbial Identification System. Friends, ladies and gentlemen, let us welcome Dr. Maria Auxilia Siringan.
Good morning to all. First, I would like to thank the organizers, the PNMCC and PSM, for inviting me to speak in this webinar, Trends in Identification. I will discuss the phenotypic identification using Biolog Microbial Identification System. Dutch scientists L.E. Dan, Doreen de Jong proved that microbes could be distinguished from each other based on their abilities to utilize different carbon sources. This was the major findings of his dissertation in 1926. All original studies on bacterial taxonomy were based on phenotypic traits. The phenotypic traits are the observable characteristics which are determined by genetic makeup of the organism and influenced by the environment. As presented in Burke's Manual of Determinative Bacteriology, the current approach in bacterial systematic is the polyphasic approach, in which the phenotypic traits, the chemotaxonomic traits, and genotypic traits are considered to establish the taxonomic ranking of microorganisms. Thus, it is essential that phenotypic traits of microorganisms be determined, as these data are of taxonomic importance. One microbial identification system that can generate phenotype-based fingerprints, which can be used in determining the taxa of various microorganisms, is the Biolog Microbial Identification System. There are 3 levels of the system, namely the manually read system, the Biolog Gentry MicroStation, which is a semi-automated system, and the Omnilog, which is a fully automated system. The Biolog system employs a carbon source utilization technology, a patented redox tetrazolium dye. It has the ability to identify 2,900 species of bacteria, yeast, and filamentous fungi. The identification system has 4 identification assays, namely the Gentry microplate, for gram-negative and gram-positive aerobic bacteria, the anaerobe microplate, the yeast microplate, and the filamentous fungi microplate. It has 4 databases, namely the aerobic database, the anaerobic database, the yeast database, and the filamentous fungi database. There are 95 tests in the Biolog ID system. Since we have 2 possible results, there are 10 to the 28 combinations of results. Thus, there are more tests and more resolving power. This enhances the capability of the system to accurately identify many more species. The main focus of this presentation is the Gentry microplate. The microplate is intended for the identification of both gram-negative and gram-positive aerobic bacteria. It contains 94 substrates, and thus you have 94 phenotypic tests. The A1 well contains distilled water, which is the negative control, and it doesn't have any carbon source. The A10 well, which is the positive control for chemical sensitivity assays for columns 10 to 12. The technology utilizes a tetrazolium redox dye, which indicates carbon utilization, resistance to inhibitory chemicals, and increased respiration by the bacterium. How does this technology work? When a substrate or chemical, such as glucose, is utilized by the organism in the transport or catabolism of nutrients, synthesis of molecules or cellular structures, or energy production, the electrons are released with the release of energy. The electrons are passed through the electron transport chain in the cell membrane. In the presence of the colorless tetrazolium dye, which is in oxidized form, the dye accepts the electrons and becomes reduced, turning purple. With the utilization of carbon sources, there is an increase in the respiration by the bacterium. With the increase in respiration, there is reduction of the dye and formation of purple color. If there is no carbon utilization, the wells remain colorless and we have a negative reaction. When the microorganism utilizes several carbon sources in the Gentry microplate, wells representing the utilized substrates turn purple. The combination of purple and colorless wells produce a phenotypic or metabolic fingerprint. The metabolic fingerprint is compared to the fingerprints of bacterial species in the Gentry database. If the fingerprint matches a specific fingerprint in the database, an identification of the bacterial isolate is generated. Here is the Gentry microplate layout. A1 well has distilled water, the negative control, and A10 is the positive control for columns 10 to 12. The Gentry microplate contains 71 carbon sources and 23 wells for chemical sensitivity assays. Here is an example of the metabolic fingerprint shown in the Gentry microplate layout and the identification of the isolate. This will be further discussed in the presentation. In some miniaturized identification system, pre-tests should be conducted, such as the oxidation fermentation test, the catalase test to differentiate gram-positive bacteria, the oxidase test to differentiate the gram-negative bacteria. With this system, there are no pre-tests and no follow-up tests. There's no gram staining, no fermentation tests, no catalase test, and no oxidase test that will be conducted prior to identification. Gentry is a universal ID system for both gram-negative and gram-positive bacteria. It utilizes a universal redox chemistry that works for both bacterial groups at the same cell density. For the Gentry ID protocol, the following are the materials: a pre-warm Gentry microplate, the inoculating fluid, which is used in the preparation of the inoculum. Gentry ID has 3 kinds of IF, namely IFA, IFB, and IFC. IFA is used for nearly all the species in the database. One has to determine the suitable IF based on the bacterial group. To culture the bacterial isolate, BUG agar with 5% sheep blood or tryptic soy agar may be used. Other materials include sterile swabs, which have 3 mm diameter, sterile 9-inch pasture pipettes, reservoirs, sterile pipette tips. All procedures should be done in a biological safety cabinet using aseptic techniques. The following major equipment are essential. The turbidimeter, which is used in the preparation of inoculum at desired cell density based on transmittance, the multi-channel or 8-channel pipetter, the incubator, which is set at 33 degrees Celsius, and a Gen 3 microstation. I'll show you the process flow for Gen 3 microplate ID assay. Remember to use aseptic techniques and perform this under a biological safety cabinet. First, restrict the isolate on BUG or TSA plate. Make sure that the culture is pure. Incubate at 32 degrees Celsius for 4 to 24 hours. If you're working with bacillus or other spore formers, incubate up to 16 hours. On the day of testing, standardize the turbidimeter using an uninoculated IFA or other IF. It should be set at 100% transmittance. Using a sterile 3 mm swab, pick an isolated colony from the plate. Then open the properly labeled IFA and release the growth of the organism from the swab by rubbing the swab tip against the bottom of the tube. Stir the cell suspension using the swab, mix well, and aim to have uniform cell suspension. Then, using a turbidimeter with the transmittance, for protocol A, aim to have a 95% transmittance. Once the desired transmittance is achieved, transfer the cell suspension in the reservoir or petri dish. Dispense the suspension in the microplate using an 8-channel pipetter. After inoculation, incubate the microplate at 33 degrees Celsius for 4 to 24 hours. Then, read the plate at 8, 16, and 22 hours using the microstation reader. To read the microplate, log into the microstation software, initialize the reader, and select the appropriate database. Encode the required data or information about the sample. Before loading the microplate, check if the reaction pattern is well developed. Place the microplate reader on the reader tray with the A1 well at the top left-hand corner. Gently push the microplate down in the tray and remove the microplate lid. Once the read button is pressed, the reader calibrates itself and the microplate is pulled inside the machine. The reader takes the reading and displays the result on the screen. The microplate is then rejected. So when you get the data sheet in the screen, you must interpret the results based on two parameters, the similarity index and the distance. Here is an example of the final identification results. Under the green bar is the list of species from the database that closely match the isolates, metabolic fingerprint, and identification. The first phase, or ID number 1, is the best match. How does the machine score the microplate? For the wells with carbon sources in columns 1 to 9, these are referenced against the A1 well, the negative control. Wells resembling A1 well are scored as negative. Wells with noticeable purple color, so that's greater than A1 well, are scored as positive. Wells with extremely faint color or with small purple flecks or clumps, they are scored as borderline. For the chemical sensitivity assay in columns 10 to 12, these are referenced against the A10 well, which is the positive control well. While showing significant sensitivity to the inhibitory chemical, less than half the color of the A10 well are scored as negative for growth. While showing normal or near normal purple color, similar to A10 well, are scored as positive for growth. Uncertain results are considered as borderline. How do we determine if we have a strong or acceptable ID? We need to check on the following. The top choices on the list should be of the same genus or closely related. Then, check the similarity index value or SIM. SIM shows how similar your results in comparison with the database. If the SIM is greater than 0.5, then we have a strong identification. The other parameter is what we call distance or the DIST. This shows mismatches between your results compared with the database. The difference between the distance of ID number 1 and ID number 2 should be greater than 2 for your identification to be considered as acceptable. Let us examine this result. All 4 candidates here belong to the same genus, which is Streptococcus. The next one is the SIM. For strong or acceptable ID, the SIM of the first ID should be greater than 0.5. So let's check. SIM is 0.72. For the distance, the difference between the distance of ID number 1 and ID number 2 should be greater than 2. So we have here 2.675 showing strong identification result. ID results can be assessed at specific incubation period. Note that the higher SIM is used for shorter incubation period. There are other ID assays using the redox tetrazolium dicarbon utilization technology. Here is for anaerobic bacteria, the AN microplate, and there are some differences in the protocol. Inoculum preparation is performed under anaerobic condition. Turbidity of the suspension is set at 65% transmittance. The AN plate is inoculated within 5 minutes of exposure to aerobic condition, and gel is formed after inoculation. Then it is settled for 15 minutes under aerobic condition, and the microplate then is incubated under anaerobic condition. The results are compared to the AN database. This is for the yeast, the YT microplate, wherein you have distilled water in A1 and D1 wells. Here you have to culture the yeast in BY agar and incubate at 26 to 28 degrees Celsius. You can use sterile water or YT inoculating fluid for inoculum preparation, and turbidity of the suspension is set at 47% transmittance. The plate is inoculated within 20 minutes to avoid metabolic activity loss, and then the inoculated YT microplate is incubated at 26 to 28 degrees Celsius for 24 to 72 hours. You have to read the results in 2 wavelengths, 590 nanometer for color formation and 750 nanometer for turbidity, that is for growth. This is for the filamentous fungi, the FF microplate. A1 well has water. In this case, you have to culture the filamentous fungus on 2% malt extract agar and grow it for 5 to 10 days. Incubate at 26 degrees Celsius. You use sterile water for inoculum preparation. The suspension should be at 75% transmittance. Inoculate within 20 minutes as in the YT microplate. Incubate the inoculated microplate at 26 to 28 degrees Celsius for 24 to 96 hours. And you also have to read the results at two wavelengths, 490 and 750 nanometers. When you use the FF plate, you can refer to the FF database. The FF database has photo library where photographs of the colonies and microscopic characteristics of the filamentous fungi are presented. These are in the database, and in that way the photographs will be used for confirmation of the identification. For the gentry microplate, here's a summary of the protocols for the different groups of aerobic bacteria. For each protocol, there's a corresponding inoculating fluid and the cell density based on percent transmittance. Gentry microplates are incubated at 32 degrees Celsius at aerobic condition and read at 750 nanometers. This table shows the AN, FF, and YT microplates and the corresponding inoculating fluid, the cell density, and incubation conditions, as well as the wavelength used in reading the plate. I would like to thank Dr. Lian of Focus Biotech for some of the slides and photos used in this presentation. I will be presenting a video presentation of the Gentry protocol, which was prepared by Mr. Rafael Eugenio and Ms. Maria Janessa Luwage. Thank you for their hard work. And thank you to the Microbiological Research and Services Laboratory of the Natural Sciences Research Institute for the facilities. And thank you to the Philippine Network of Microbial Culture Collections, PNMCC. Maraming salamat po. Please stay tuned for the video presentation. Thank you for your kind attention. Good day. Good morning.
To give you a short background for our second speaker for this morning, she is a University Research Associate 1 at the Philippine National Collection of Microorganisms, PNCM, National Institute of Molecular Biology and Biotechnology, Biotech at the University of the Philippines, Los Baños. She has been affiliated with PNCM since 2016. She received her Bachelor of Science degree in biology, major in microbiology from the University of the Philippines, Los Baños in 2016, and became a registered microbiologist in 2018. Her 6 years of experience in the PNCM includes maintenance, preservation, and identification of bacteria and multicellular fungi. As one of the technical analysts in the organization, she has participated in the provision of microbiological analysis, such as detection and count of microbial pathogens and beneficial microorganisms in food, water, and agricultural samples. Since 2018, she has been involved in two research projects funded by the Department of Agriculture, Bureau of Agricultural Research. Her research involvements concentrate on soil microbial activity and diversity in rice and corn farms. She is the current auditor of the Philippine Network of Microbial Culture Collections Incorporated to give a lecture on the topic Microbial Identification Made Easier, the VTEC 2 Identification System for Bacteria and Yeast. Friends, ladies, and gentlemen, let us give a virtual round of applause to Miss Rose May Ann Capanzana. To give you a short
Good day, everyone. Welcome to the PSM Preconvention webinar. Today, I'll be discussing about the VITEC2 identification system for bacteria and yeast. I guess, just like the microorganisms, the participants in this Zoom meeting are also diverse. Do we have students here? For sure, there are also researchers, educators, and professionals from the industry. But generally, I know we are here because of our interest in the field of microbiology. So, the science of microbiology revolves around these two themes or goals. Number one is to understand the microbial world, and number two is to apply the understanding for the benefit of humankind and the place that we live in. Just look around you and you will see that we have been benefiting so much from the deep understanding of the microbial world. So in the medical field, the identification of human pathogens has become faster, and so the diagnosis and the provision of treatments. In the food industry, it has also been improved since we are already consuming safe food. And in agriculture, many microbial-based technologies are also developed in order to improve crop yield. So in this lecture, we will be gearing on towards the goal of this science to understand the microbial world. And a huge portion of understanding the microbial world is to be able to identify the microorganisms, to be able to characterize, name, and classify them based on an established criteria. So, traditionally, as you can see here, the taxonomy of microorganisms relied heavily on their phenotype or any observable physical or metabolic properties. But after the establishment of the genetic code and the development of several technologies, the taxonomy of microorganisms then relied upon DNA sequence data, which are used to position the microorganisms in an evolutionary framework or to determine their phylogenetic relationships. So you may be asking, What should we follow then in terms of identifying microorganisms? What is trending in the microbial taxonomy? So to answer that question, well, it's the polyphasic approach, wherein the organism is identified based on its phenotype, genotype, and phylogeny. So in polyphasic taxonomy, the phenotypic, the genotypic, and phylogenetic characteristics of the organisms are all involved. So listed here are the common microbial characterization methods. So the phenotypic analysis, gene sequence analysis, multi-locus sequence typing or the MLST, genome fingerprinting, and the multi-gene and whole genome analysis. So how do we really characterize microbes? So as mentioned, the phenotypic analysis is concerned on the microorganism's observable characteristics. I will elaborate on that later on. So in gene sequence analysis, sequence data of highly conserved small subunit rRNA or the 16S rRNA are obtained. So here is the 16S rRNA. Some other highly conserved genes are also used to identify closely related species. And for MLST, the housekeeping genes are the target for sequencing. So these housekeeping genes are located in the chromosome rather than in the plasmid. And they are the key to the essential functions in the cell. So housekeeping genes are used to distinguish species because those regions are more genetically varied compared to 16S. So in this figure, for MLST, the researchers selected these 7 housekeeping genes to distinguish pneumococci. So genome fingerprinting, on the other hand, is used to distinguish strains by PCR and separation of DNA products by gel electrophoresis. So simply, differences in the sizes of the DNA fragments would distinguish one strain from the other. So lastly, the multi-gene and the whole genome analysis. So this image here is the whole genome of Haemophilus influenzae. So the first whole genome that was sequenced in 1995. So as mentioned here are the usual or the most common microbial characterization methods. So several technologies have already been developed for fast and reliable characterization of microorganisms for polyphasic taxonomy. For this lecture, we would like to focus, or I would like to focus on the phenotypic characterization of bacteria and yeast using the Vitek2 biochemical-based identification system. So just to clarify, I am not affiliated with the company of this technology. I will just be speaking as an end user. So let's continue on. So what are the phenotypic characteristics of taxonomic value? So of course, the morphology. I hope you're still not yet tired of observing your isolates under the microscope. Yes, the gram reaction, cell size, shape, colony morphology are still valuable in taxonomy. The motility. Are the cells non-motile? Do they exhibit flagellar or gliding motility? Also important are the cell lipid and cell wall chemistry and fatty acid composition, and also the presence or the absence of peptidoglycan. For this lecture, I would like to highlight on these phenotypic characteristics here because the Vitek2 identification system mainly analyzes these phenotypic characteristics. So the metabolism, the physiology, and other special traits, specifically antibiotic sensitivity. So the metabolism of microorganisms comprises the mechanism of energy conservation, utilization of carbon and nitrogen, the capability to use sugars under aerobic or anaerobic conditions. The description of the physiology is also important. What is the organism's optimum temperature for growth? Thus, the different environmental factors such as pH, temperature, oxygen, and availability of moisture affect the growth of the organism. So other traits such as the antibiotic sensitivity, production of antibiotics, and unique compounds are also valuable. And these unique traits can distinguish one species or strain from the other. So this is an example of a traditional method for phenotypic characterization. Of course, we usually start off by media preparation. Then we do dispensing of the media in receptacles like petri dishes or test tubes. Then we inoculate the pure culture in the media containing the carbon sources, the nitrogen sources, some indicators, inhibitors, and many other substrates. Then we complete the incubation period and then visually inspect the growth of the colonies in the media. So this picture shows you the traditional method or the conventional method for biochemical testing of Salmonella isolates. So now, there is API, which is the miniature version of the conventional method. So the design of API was developed in the 1970s. In API, the substrates are contained in each of these cupules or the small capsule-like containers. So fresh bacterial suspension is inoculated and followed by the incubation of the test strips. So upon incubation, the results are visually inspected by the user or by the handler, and then reagents are added if necessary. So API already has this web-based system wherein you can plug in the results. Then the report will be automatically generated. Now, the same company who developed API has come up with this automated system called the Vitek2 identification system. So in our lab, what we have is the Vitek2 compact. So this is what the Vitek2 setup looks like. So the computer is, the equipment is connected to a computer where the software to view the results are installed. The buttons here are used to operate the machine. This we have two doors here where the reagent cards together with the cell suspension are placed. So we will see more of that later. And this is the Vitek 2 reagent card. So this is what it looks like. So the small wells contain the substrates. So you have here also the barcodes. Then this is the capillary tube which draws the cell suspension so as to distribute them to each of the wells containing the substrate. So a simple comparison between API and Vitek is that API is not automated, while Vitek 2 is automated. So for Vitek 2, the user would only have to confirm the purity and gram reaction of the isolate, then to prepare the cell suspension, then the machine would do the rest of the analysis. For API, it takes 1 to 2 days to get the results, and the turnaround time for Vitek 2, on the other hand, is highly reduced, wherein we can already get results for 2 to 18 hours. Based on experience, we get the fastest results for gram-negative rods and gram-positive cocci, which take about 3 to 4 hours. So a fast turnaround time is really important, especially in the clinical setups. I mean, the faster we get to know the pathogen, the faster we can administer proper treatment for it. So also the knowledge base of the Vitec 2 is uh, distinct from the API. So the Vitec 2 has plant strains. So for example, you will see it later. Uh, the gram-negative database has Ralstonia and Rhizobium species. So these are the types of Vitec 2 reagent cards. Each of the reagent cards have different set of substrates or biochemical tests. So Vitec 2 requires different reagent cards to different groups of microorganisms, specifically the gram-negative bacilli or the gram-negative rods that are fermenter and non-fermenter, the gram-positive cocci, uh, the yeasts, the fastidious bacteria, for example, the Campylobacter, the anaerobes and Corynebacterium, the Corynebacterium and related species, and also the lactic acid bacteria. And lastly, the gram-positive bacilli or the bacillus and related genera. So this is how huge the knowledge base is for these different cards. So the time of results right here is also indicated. So this is how much the Vitec 2 can identify per agent card. So these are the names of the ID cards. So we have here BCL, CBC, GN, GP, YST, ANC, and NH. So each of the card is designated such as this. And these are the microorganisms identified for each card. So we have uh, Bacillus for BCL, Corynebacterium for CBC, the gram-negative, the gram-positive, the yeasts, the anaerobes, and the Neisseria and Haemophilus. So these are the organisms that are that can be identified by through each reagent card. Okay, so what are the tests performed by the Vitec 2? So this automated identification system performs enzymatic tests, acidification, alkalinization, assimilation, inhibitions test, and also other physiological tests. So I will discuss this one by one in detail later. So here are also the types of tests in API. So as a review, so these are the tests for API. So unlike the manual interpretation of the results in API, the Vitec 2 uses an advanced colorimetry technology to detect metabolic activities within each well. So every 15 minutes, these reading heads, these two reading heads, monitor the color changes for each well in the reagent card. So they use 430 nanometer wavelength to detect color reactions related to glycosidase and phosphatase activities, 568 nanometers to detect decarboxylase and glycosidases, and also the color reaction to confirm the resistance to polymyxin B. And for acidification, alkalinization, some enzyme tests, carbon utilization, and inhibitions tests, which normally show blue to dark blue results, they use 660 nanometer wavelength. So I can only share up to this point in terms of mechanism by which Vitec 2 reads the color reaction. If some of you are interested in the engineering of Vitec 2, you can contact me, then I can connect you to the technical personnel from Biomeru. So for the materials and sample preparation, right here. So number one, it is very critical that you have your pure culture of bacteria or yeast and that the correct gram reaction has already been determined. So you can finally select for the appropriate reagent card to use for the run. So for the sample preparation, the DensiCheck Plus and the DensiCheck Plus standard kits are needed. These two are also developed by the same company. So the 0.5% saline solution. So I would like to emphasize that cell suspensions that are not within the appropriate zone of the DensiCheck Plus may compromise the results. So aside from that, you need again the saline solution, stereo stick or swab, polystyrene tubes, vortex of course to homogenize your cell suspension, the reagent cards, the Vitek 2 cassettes. So the Vitek 2 cassettes are like customized test tube racks for the equipment, and a barcode reader. So to give you an overview of the sample preparation, here is a video.
The Vitek 2 compact system simplifies and speeds the identification and susceptibility testing of bacterial and yeast organisms. The purpose of this video is to demonstrate and explain how to set up isolates and test cards for processing on the Vitek 2 compact instrument. It is critical that specimen suspensions and dilutions be prepared correctly to ensure accurate test results. First, gather the items you will need. Patient specimens on culture plates in compliance with the culture requirements table found in the Vitek 2 Systems Product Information Manual. A test tube rack, 12 by 75 mm clear polystyrene disposable test tubes and caps, a bottle of sterile aqueous 0.45 to 0.50% saline with a pH between 4.5 and 7.0. The dispense set adjustable volume saline dispenser received with the Vitek 2 compact instrument startup kit, sterile sticks or swabs, the DensiCheck Plus instrument and standards kit used to measure the optical density of a microorganism suspension, the 2 fixed volume pipetters received with the system and pipette tips, a vortex, if necessary, to prepare a homogeneous organism suspension, the number of Vitek 2 test cards, warm to room temperature, required for setup, lint-free tissue or lens paper, plastic disposable transfer pipettes, powder-free gloves, an approved biohazard waste container, and a Vitek 2 compact cassette. When setting up isolates for testing on the Vitek 2 compact system, an organism suspension is made using the DensiCheck Plus instrument to achieve the turbidity required for the card type being set up. For this organism suspension is used to inoculate the card. For AST card setup, an aliquot from the ID suspension tube is transferred to an AST tube with 3.0 milliliters of saline. This suspension is used to inoculate the AST card. For the AST suspensions, 145 milliliters of the ID suspension is used for a gram-negative card, and 280 milliliters is used for a gram-positive strep or yeast card. Remove the ID test card, now warm to room temperature, from its foil pouch and place in the cassette with the transfer tube pointing away from you in order to minimize accidentally touching the transfer tube. If required by your standard operating procedure, label the top third of a 12 by 75 millimeter clear polystyrene tube with patient identification. Using the dispense set, dispense 3.0 milliliters of saline into the tube. Use a sterile stick or swab to transfer morphologically similar colonies to the saline tube. Avoid picking up any agar with the stick or swab. Mix well to prepare a homogeneous suspension. If necessary, cap the tube and vortex thoroughly. Use the DensiCheck Plus to verify an inoculum density equivalent to the McFarland units required by the ID card. Press the power key to turn on the DensiCheck Plus. Note, in order to prolong battery life, the instrument will automatically shut off after 1 minute of non-use. Verify the tube type is set to plastic. Clean the outside of the tube with lint-free tissue or lens paper. Insert the tube containing the organism suspension into the DensiCheck Plus, ensuring that it is seated at the bottom. The DensiCheck Plus displays a series of dashes. Slowly rotate the tube 1 complete revolution, 360 degrees, before the dash is complete and the reading is displayed. Check that the displayed McFarland unit is within the acceptable range. Test card inoculum density ranges can be found in the Product Information Manual. If the McFarland unit is too high, dilute the suspension with small increments of saline and recheck the value. In order to prevent contamination, do not add additional saline directly from the dispensette. Instead, follow laboratory best practice by dispensing saline for dilutions into a separate test tube and then use a transfer pipette to add the saline to the inoculum tube. If the suspension reading is lower than the required McFarland, add more organism and recheck the density. Once the density is within the correct range, place the inoculum tube in the well in front of the ID card. If no AST is to be set up on this isolate, turn the ID card and place the transfer tube into the suspension.
So finally, after the sample preparation, you will place the suspension, the reagent card inside the machine. So here is what happens inside the Vitek tube. Okay, I hope you had an overview of the mechanism and as well as the sample preparation. So let's now go to the specifics. So as mentioned earlier, here are the types of tests performed by Vitec 2. So the enzymatic tests, the acidification tests, alkalinization test, assimilation, inhibitions test, and other physiological tests. So in this image right here are the examples of enzymatic tests in the gram negative or the GN reagent card. For the enzymatic tests, synthetic enzymatic substrates are used to detect enzymatic activity. So these enzymatic activities are usually indicated by measurable signals such as absorbance or fluorescence. So why is describing the enzymatic content of the cell relevant in taxonomy? Well, it's because the enzyme produced by the organisms are coded by different genes and so are reflective of the genetic makeup of the species. So here are the enzymatic tests for each reagent card. So these unique metabolic activities or patterns are used to determine the species. So most of the tests are actually well established based on several references like the Burgess Manual of Bacteriology, other diagnostic microbiology references, and numerous published research papers. So let's continue. Number 2. For the acidification tests, we will see here if the organism can ferment sugar or sugar derivatives and utilize other protein substrate. We know that carbohydrates and proteins are used for the production of biochemical building blocks and energy of the microorganisms. So in Vitec 2, the presence of acid byproducts upon the utilization of substrate is detected by the color changes in the pH indicator. So right here is the list of the acidification tests performed in each type of the Vitec 2 reagent card. So number 3 would be the alkalinization tests. So it is the utilization of lactate, malonate, well, specifically for this card, the utilization of lactate, malonate, succinate, citrate are indicated by the presence of alkaline end products. So it is the single substrate utilization, so growth in the presence of one substrate. And in the alkalinization medium, so it is the presence of the end product of metabolic activity, so which causes the changes in the pH indicator in the medium. So for the assimilation tests, so here are the tests for alkalinization in each card. So for the assimilation tests, we want to know if the organism has the capability to use a substrate as a sole carbon or nitrogen source. So here is an example of assimilation test included in the YST reagent card. So since yeasts are capable to ferment various sugar derivatives, this trait was used to differentiate yeast species. So the majority of the tests in the YST card are actually assimilation tests. So here are the assimilation tests for in each reagent cards. So as you can see here, we have many assimilation tests involved in the YST card. So for the inhibitions test, the growth in the inhibitory media or the resistance of the microorganisms from inhibitory substances are also used to identify the species. So direct testing of the bacterial isolate against an antimicrobial agent is useful in identification. So for example, many gram-positive bacteria are susceptible to vancomycin, which acts on the bacterial cell wall. In contrast, most of the clinically important gram-negative bacteria are resistant to vancomycin. So therefore, the antimicrobial susceptibility may help to confirm the gram status of the organisms. So you see the combination of these patterns or the results of the biochemical tests are also like fingerprints of the bacterial or the yeast isolates. In the medical field, it is important to also know the antimicrobial susceptibility of the target isolates to be able to provide proper treatment for the patient, or how to handle the treatment of the by the pathogen. So listed here are the inhibitions tests in the reagent cards, and also other physiological tests right here. So here are the interpretation of the results provided by Vitec. So 96 to 99% probability is classified as an excellent identification. 93 to 95 is very good ID. 89 to 92 is good ID. So 85 to 88 is acceptable. So for low discrimination results, Vitec 2 would suggest 2 to 3 taxa for the ID, then provide a separate supplemental tests. These supplemental tests are done offline or they are done manually. So these are the species that can be identified using Vitec 2. Note that many from the GN and GP group are clinically important bacteria. So the Vitec 2 can identify critical and highly pathogenic microorganisms such as Brucella militensis, Burkholderia malae, Burkholderia pseudomalae, E. coli 0157, the Salmonella group, Shigella, Vibrio, and also Yersinia pestis. So these are the species under GN right here. So we have the Enterobacteriaceae and the non-Enterobacteriaceae. So these are the species of gram-positive cocci that can be identified using Vitec 2. So we have here a critical pathogen also, which is Listeria monocytogenes. Here is the list of species that can be identified by the BCL and the CBC card. So note that the BCL and the CBC database are mainly industrially important gram-positive bacilli, Corynebacterium and Lactobacillus species. So in the lab, we usually target non-pathogenic bacillus species with agriculturally important characteristics. So we usually get this slash line taxa here, the Bacillus cereus, Bacillus thuringiensis, and Bacillus mycoides, as well as the Bacillus subtilis, Bacillus amyloliquefaciens, and Bacillus atrophius. They usually come together, and we will talk about that later. So here is also the list of the microorganisms under the CBC card. All right, so these are industrially important. We have here Lactobacillus species, and for YST, we have pathogenic, highly pathogenic organisms that can be identified by the YST. We also have here Cryptococcus theopharmans, which is a highly pathogenic yeast. So, okay, I hope you're still with me. So let's continue. So this is a sample laboratory report with an excellent identification. So the unique bio number is located right here. And for this run, the isolate was identified as Bacillus licheniformis. So with 99% probability. So as you can see, only one taxa has been provided by the VITEC 2. So one species ID has been provided with no contradicting typical biopatterns. So here are the reactions to the different substrates. So the parentheses right here are would denote a weak positive and a weak negative reaction. So another example is a very good identification, but with a slash line taxa. So provided. So Vitek suggested to rule out these three possible identities by doing this offline tests right here. So the numbers in the parentheses indicate percent positive reaction for the species. For example, this isolate X, let's call this isolate X, for example, will be identified as Bacillus cereus only if it will be confirmed to not develop the toxin crystals in rhizoidal colonies. So basically, the zeros here denote that the colonies of Bacillus cereus are not rhizoidal, and the species is negative for the presence of toxin crystals. So in contrast, you can expect to observe toxin crystals if the isolate is if isolate X is Bacillus thuringiensis, because the species toxin crystal percent positive reaction is 100. In short, the presence of toxin crystals is a typical characteristic of Bacillus thuringiensis. So Vitek 2 would provide these offline tests in case of slash line taxa. So these are the contradicting biopatterns, and these are the result of the tests. So right here is the sample supplemental test. So this was provided for our slash line taxa earlier for isolate X. So to confirm, we should confirm if the isolate, the colonies of the isolate are rhizoidal. We have to confirm if its growth on agar is rhizoidal, and also the presence of toxin crystal. So before we can rule out if it is Bacillus cereus, Lycoides, or thuringiensis, we have to confirm the characteristics of these isolates. So for the toxin crystal, the presence of toxin crystal present, that the presence of toxin crystals, microscopic examination using the phase contrast microscopy would be recommended. So this is how you differentiate the three species. So this one is a sample report of an identified isolate. So the isolate may be unidentified because of its very atypical biopattern, or it may not correspond to any taxon in the database. So it would also be better to check the gram reaction and purity of the isolate. So based on an internal study conducted by Biomeru, the accuracy of identification of gram-negative bacilli is 96.2. For gram-positive cocci, 96.1. For yeast-like organisms, the accuracy would be 98.2%, and for anaerobic bacteria, 93.9%. While the industrial manufacturers' tests show that the ID system can give 95.6% correct ID for gram-positive spore-forming bacilli, a 96.7% for the coroniform bacteria. And here is the estimated turnaround time for ID of each group. And for other reviews of Vitek 2, the research group of Funk was able to evaluate the Vitek 2 system specifically for the identification of gram-negative rods. They identified a total of 845 isolates using GN, and the results show that 716 of the isolates with correct identification uh, that and got a total of 716 isolates with correct identification. So that is 84.7% of the total number of isolates. So 32 isolates were identified correctly after doing supplemental tests, but 80 isolates were not identified after doing the supplemental tests. And 7 isolates were misidentified, while 10 isolates were unidentified. So overall, based on their study, the VITEC2 performed well for identification of Enterobacteriaceae and selected non-Enterobacteriaceae. So Crowley and the research group in 2012 evaluated the performance of VITEC2 GP reagent card in identifying gram-positive cocci and non-gram-positive cocci. So the gram-positive cocci group was designated as the inclusivity isolates, while the non-gram-positive cocci group was designated as the exclusivity isolates. So the identity of the isolates that were tested are previously confirmed. So the inclusivity group was comprised of Listeria and Staphylococcus species, while the exclusivity isolates are Bacillus coagulans, Citrobacter, Escherichia coli, Serratia marcescens, Aspergillus niger, and Candida albicans. So basically, they tested these non-GP isolates using the GP card. So that is the exclusivity group. So results showed that 99.2% of the inclusivity isolates were identified correctly. None were misidentified, and also none is unidentified. So on the other hand, 106 out of the 106 out of the 120 exclusivity isolates were correctly excluded, meaning these non-GP isolates were not identified properly using the GP card. So this tells us that the VITEC2 can accurately rule out GP organisms from the non-GP. However, there are still 14 isolates that were incorrectly identified as GP organisms among the exclusivity isolate. Meanwhile, this paper authored by Akahane and the team in 2011 showed the limitation of VITEC2 in the identification of Pasteurella dogmatis. The researchers used two different rapid tests to identify two isolates from a patient. So Pasteurella dogmatis was mistakenly identified as Pasteurella pneumotropica. Simply because the species is not yet included in the database of VITEC2 at the time of evaluation. So upon checking, Biomero has already updated their database and have included Pasteurella dogmatis in the GN list. So for the advantages and disadvantages, of course, the VITEC2 gives fast and accurate results. So there's also reduction of human errors, especially in inspecting the results upon incubation. And there's minimal reagent preparation, as you have observed when we watched the sample preparation. There's minimal machine operation. So once you have prepared the sample suspension or the cell suspension, you can leave it right there, and then the machine does the work. So the machine will give you the identification at the end of the run. And this equipment is relatively low maintenance. So you can actually turn it off so it's not continuously running. You can turn off the machine after use. And it also is very productive since it has the capability to process 60 cards per run. So some of the disadvantages, of course, it's still costly. If your lab is just starting, or it's not it does not need so much routinary identification, this would really be costly. So it's relatively expensive. The Vitec 2 still relies on, of course, it's still conventional. It's just that it's automated. So it's culture dependent. Only those that are culturable can be tested. The gram reactions are predetermined, and also the identification is limited to its database. That's why they are continuously updating their database. And also, supplemental tests are needed if the results were not sufficient to complete the identification. So sometimes, or there are times that the Vitec tool will give you more than one taxa, and so you need to confirm the identity by doing offline or manual or external supplemental tests. Okay, so here is a summary. So we have talked about briefly about the polyphasic taxonomy. We mentioned that phenotypic analysis is still relevant in taxonomy. So phenotypic characterization using conventional and rapid methods are also introduced. Phenotypic characterization using the Vitec2 ID system, which uses advanced colorimetry technology to detect metabolic changes, was also introduced. So the types of tests performed by Vitec2 has been talked about, and the results provided by Vitec2, we also discussed about Vitec2 as an accurate tool for biochemical based identification of microorganisms, as well as the advantages and disadvantages of the Vitec2 system. So that's all. Here are my references. So you can check those on later, and you can also contact me for further questions. And thank you very much for listening. Have a good day.
database.
Good day, everyone. My name is Lu from Singapore, Sumatra is Pacific. I'm very happy to see you here for today's webinar. In this session, I would like to present you the macro identification by multi-top and Ceramis database. These are contents of my presentation. First, I would like to introduce the macro identification and methods, followed by the multi-top, a rapid and reliable new standard in macro identification. Then I will introduce Smarter solutions like Smart ID Plus multi-top with Ceramis database. Then followed by the features and the benefits of Smarter solutions. At last, I will share some of the application examples for macro identification by multi-top. Macro identification is a key task of microbiology. So in general, the macro identification can be defined as a clear microbial characterization by an appropriate test method that is able to retain the name of the analyzed species. So in general, there are three levels methods. They are genotypic, proteotypic, and phenotypic. Genotypic identification analyzes the gene sequence of RNA regions of the bacteria and fungi, while the proteotypic identification analyzes the molecular and the peptide profiles of ribosome proteins. Lastly, the phenotypic identification analyzes the biochemical reactions. Here, I want to highlight the method you use for macro identification. The more away from the original DNA information, the accuracy and the reproducibility will decrease. So it is important to know the identification methodology is a critical factor for accuracy and reproducibility of macro identification. This slide, I just list down some of the methods and the technique that are commonly used for macro identification. Conventionally, we understand the macro identification can be achieved by carrying the biochemical analysis or sensory analysis by viewing the physical and chemical reactions that occur when the microorganisms come into connect with a certain substance. However, this method is quite time-consuming. Sometimes may take more than 2 days with low accuracy. For now, we understand the gene sequencing for the 16S and the 18S rRNA is still the gold standard for macro-identification. It can identify the fastidious and uncultivable microorganisms. However, this method is very expensive and not suitable for routine clinical use. Especially, it requires a powerful interpretation software for data analysis. So instead, the PCR, like the real-time PCR and the multiplex PCR, is probably the most commonly used method for molecular technique. This method can achieve the specific sensitivity and rapid and accuracy analysis for macro identification. It can also detect major pathogens at the same time. However, it still requires a high precise thermocycler for the sample analysis. Other than that, now we understand the multi-top MS analysis has become a rapid and reliable new standard method for macro identification. It enables a very fast less than 1 minute and can get a very high accuracy for the identification in a cost-effective manner. Here, I just want to highlight the multi-top MS identification is at the proteotypic level based on the molecular and the peptide profiles of the proteins. So it requires a high perceived database for the matching. In this slide, I just want to give the further interpretation regarding the technique of multi-top. Multi-top, it stands for the matrix assisted laser disruption and annihilation time of light. So basically, it is an analytical MS technique that uses a laser matrix to create the ions from the molecules. And then the mass to charge ratio of the ion can go to the flight tube and be measured by the flight time in the flighting tube. So eventually we can get the mass spectrum of the samples. This slide just simply lists down the workflow of macro identification by multi-top in just 3 steps. First, we need to have the sample, apply the bacterial sample onto the sample plate. Next, just load the sample into the multi-MS system for the data acquisition. Once we collect the mass spectrum of the sample, we can do the database searching. For the database searching, we need the data analysis software to import the spectra compared to the reference spectra in the database, then based on the similarity of matching to identify the species of the bacteria. Here, just I want to explain why we can use the spectra of each bacteria for the identification. This is because for different the species of the different material bacteria, they have different the mass pattern, mass peak pattern. You can see here. So based on this peak pattern, we can differentiate those different bacteria. Now we come to the smarter solution for macro identification. For smarter solutions, we have the ID plus multi-top mass spectrometer. This is to help you to collect the mass spectra of your samples. We have 3 models: ID plus assurance, ID plus confidence, and ID plus performance. ID plus assurance is just a simple linear multi-top mode. Confidence is a reflection multi-top mode. And ID plus performance is a more advanced multi-top instrument. On top of the MS spectra, actually for the macro identification, we need a powerful database for the data library searching. So we have the Ceramis database, is a spectral archive and the macro identification system. It is an easy to use software for macro identification through mass spectral matching. So basically, this software is just a database. It is a combination of the bacteria, of the algae, macta, and the yeast. So it contains the majority of the main microorganisms related to the clinical, veterinary, food, beverage, and pharmaceutical field, including both superspectral and the reference spectra. For the identification, the software can be very fully automated. So they can directly search against the superstructure in the database. So in case some case the identification confidence is very low, less than 90%, the sample profile can be researched against all available reference spectra in the database. This can be further improve the confidence identification. So in the next slide, I will explain what is superspectral and why we need to use superspectral. Here, for superspectral, each superspectral is derived from the multiple reference spectra of at least 15 individual isolates of the species acquired after varying growth times and from different growth medium to ensure the reliability and the reproducibility. So for example, here we have the around 50 individual isolates of the species bacteria. Then we can collect all the individual mass spectral profile. Then we can get some of the similarity analysis to find the most common profiles among all the spectra. We generate a consensus spectra with an average signal intensity. Usually we use a 70% similarity in a spectra. So based on the consensus spectra, we can create the superspectra by assigning the peak width to each mass signal. So the peak width is assigned according to the specificity to the represented species. So for example, if the peak with genus or family specificity will be downweighted as they do not support the species identification. For peaks with species specificity will upweighted. So in summary, the superspectra summarize the species specific mass fingerprint data. This slide just to explain why we need the superspectra. This is because there are a lot of advantages for the superspectra. For example, they can discriminate the close relative species by reducing the background similarity. They are computed for the different taxonomic levels to reflect the characterization in particular text, and can be used to avoid false result by testing against the reference spectra. And also they can directly identify the components of mixed samples. Here just an example to show how we can identify the bacteria based on the superspectra. So for example, here we have two samples, 539 and the number 582. We can see some of the difference in the two samples on the spectra. Well, when we match into the superspectra, we will see this have the very similarity, the matching point, the matching data point. And eventually, these both samples will identify as the same bacteria, the trichophotin, the rubro, with a very high confidence score, 99%. So this is a way to show you why we need to use the superspectra to get the more accurate identification. This slide just to show you another software we call the string solution. With this software, we can further do the micro-identification down to the subspace and the strange level based on the gene sequence information. And also we can do the class analysis like the molecular phylogenetic classification. This slide just show you how the string solution works. So basically, they compare between the theoretical mass based on the amino acid sequence of the bacterial proteins, and also an accurate more detailed mass spectra of the bacterial strains. So first, we need to calculate the theoretical mass based on the amino sequence and register to the database. Then next, we need to collect the real mass profile with the real samples. Then next, we need to matching both of them. Then we can characterize the serotypes and the genotypes with the cluster analysis like the molecular phylogenetic analysis. Here it just lists down the features of string solution that enable the highly accurate bacterial discrimination, enable the bacterial differentiation based on the difference in the genotype. And also the bacterial strain data can be customized. It supports the molecular, the phylogenetic analysis, and the cluster analysis functions. So we come to the summary of the smarter solution for micro-identification. With the another sample display on the assembly after the data acquisition on the multi-top. We can go to the two ways. One is the ceramic software for the database searching. One is the string solution for the identification at the subspace and the string level. In next two slides, I will give more details how we need to prepare the sample and do the database searching. For the sample preparation is very straightforward. We just collect a colony from the agar plate, the medium, then apply on the multi-top, the sample plate, then mix add on with the matrix solution. For matrix solution, we have two options. One is CCA, one is the SA, depending on the profiles of your samples. Then after the sample drawing, we can see a kind of this crystallization. Okay. Then we can go to the sample analysis and the multi-top. After we get this spectrum, we can do the fully automation database searching, matching the superspectral database and score all the matching peaks. And we can rank the result based on the matching superspectral database, and also can display the result based on the confidence levels. The ceramics database, they can show the color code for the confidence level in the four colors. Green color is highly the confidence for the identification matching up to 99.9%. Yellow color means the highly possible competition is from the 85 to 90%. White color means no ID or is a lower score. Red color here I want to highlight is not mean the error. It's just an alert for some of the conflicting result. For example, here for this particular sample, they matched more than 2 or more species with a very high score confidence in the sample. It means this sample is a mixture samples, so you need to manually selection the result by the user. On top of the matching, the ceramic database software also can do the clustering analysis to get the similarity of the different bacteria. This slide just to summarize the features and benefits of multi-top ceramics workflow. So it is very easy and flexible operation the procedure, just 2 or 3 steps from the sample preparation to identification. With a high throughput screening, you can run up to 1,000 samples per day, because in general, each sample only takes less than 1 minute for analysis. And with high reproducible and reliable the identification by using the super spectra to reduce false discovery. And also they can use a cluster function to the clustering analysis. And also there's an optional function for the MS for the structure analysis. And this is capable for identifying the mixed cultures. So if your sample is a mixed culture, also you can direct to apply the sample into this sample analysis. This is last but not least, this is an open system. It allows the addition of the new space into the database. So you can create your own library, and also you can add your new bacteria mass spectra into the current database. So it is very flexible and easy workflow just from the sample pre-treatment, sample analysis, data acquisition, then database matching. Okay, now we come to the last part of my presentation. Here I just want to share some of the application examples for micro-identification by the multi-top. Here we list down the 3 applications of research papers for the ceremony, the serotyping, solution level. This is a routine analysis in food safety. So from the food samples, we can do the profile analysis, how many athletes we can identify in the food samples. For this application, we identified more than 100 athletes from the food sample. Also, this is an example for the mixture culture identification. So if your sample is a mixture culture, contains more than 2 bacteria, you can also direct to apply to the multi-plate, then do the database search. They can identify the individual and the mixed bacteria culture based on the procedure. Here is just list down some of the more applications on the research paper for micro-identification using the multi-top. Okay, this is the last slide. A quick summary for the ID+, the multi-top ceramics database for the micro-identification. So in general, this ID+ is a very simple, fast, effective workflow for micro-identification. Here, when we talk about ID+, it also comes to 2 parts. One is the multi-top, the mass spectrometer. The other one is the ceramics database search. Okay, now we come to the end of my presentation. So if you have more questions or if you want to learn more regarding the micro-identification on the multi-top, you are welcome to contact.
The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil.
Hello everyone, my name is Nicole. I'm from Shimazu Asia Pacific based in Singapore. Today, my colleague Dr. Lu and myself will be introduced on multi-top technology together with a database from Shimazu on how it can help in microbial identification workflow. Some of you already know Shimazu, but some of you maybe haven't really heard about Shimazu. So we are a Japanese company established close to 150 years ago in Kyoto. So along the 150 years of history, we have hit many different milestones. So for example, the most related to our topic for today will be at the year of 2002, our engineer in Japan, Kyoto, Tanaka-san, who was awarded as a Nobel Prize in chemistry. And this enables or allows us until now, Shimazu is the only analytical instrument company who has got such an award. And also at the year of 2009, Shimazu developed the world's very first ultra-fast mass spectrometer instruments, which we also call it UFMS. So far, Shimazu is the longest serving analytical instruments in the whole wide world. And we were talking about the ultra-fast mass spectrometers. We are not specifically strain ourselves into, for example, the LC-MS triple quadruple system or even ICP-MS. In fact, across the whole board of the MS product Shimazu is providing, is using the ultra-fast MS technology. So that's why if you see MS, especially the UFMS, it is specifically refers to Shimazu MS instruments, from LC-MS to GC-MS to ICP-MS. And because of the UFMS technology, Shimazu has been awarded as the Asia Pacific Company of the Year for mass spectrometry instruments from 2015 continuously to 2018. Today, we are going to introduce our multi-TOF technology. So if you can see here, we have a different type of the multi-TOF instruments, different models as well. So the most related to the microbial identification or microbial ID, it is actually these three models right here. Okay, we call it assurance, confidence, and performance. But we are not just limit ourselves into these three models of the multi-TOF technology. On top of that, we also having a benchtop multi-TOF instruments. If you can see the multi mini, which is really a small and compact multi instruments, as well as the multi 8020. All right, so they both are focusing on the clinical market, clinical studies. Additional to this, multi-imaging has been raising demand from the customer, especially for the advanced researchers, because it offers, especially from Shimadzu, if you can see right here on top, the IM scope QT. So this is actually combines the imaging or the multi-imaging microscope together with a QTOF high resolution accurate mass instruments. So the benefits we can bring to the customers, which allows the customer, the researchers, to conduct both qualitative as well as quantitative analysis. So this is the beauty of the IM scope QT instruments. But we also have the multi-TOF or multi-instruments 7090, and which is also allows the researcher to do a lot of multi-imaging analysis research works. So let's zoom into the microbial identification instruments, which we see here, the three different models. So no matter which model of that, so the key applications for microbial ID instruments, the models, is really for microbial identification, all right? And on top of that, you can also conduct proteomics biomarker identification workflow as well. So what's the difference between all three models? Assurance, it is the entry multi-instruments. It is having a linear detector only, all right? So it is can do a simple, quick, easy analysis. Confidence is next level, which means it has the reflectron technology, which to improve the MS resolutions. In order to improve the MS resolution as well. And if you're looking at the performance, which is the most high end of the three different models, it is a combination of the TOF system, which enables the MS2 fragmentations. So if I convert it to the applications or convert it to customers analysis, it helps customers to do a compound structure elucidations. So if you can see here, so the three different models is actually target for different type of the customers and different group of the research as well. So recently, there are many increasing threats from the microbial contaminants or microbial sickness, illness. So for example, Salmonella, it is the most common bacteria causes foodborne illness. So we have so many different types of the news that's been coming out. But back to 2015, the data is from globally that Salmonella itself accounts for close to 100 million foodborne at that time. That means per year. And it actually causes close to 160,000 deaths a year. And in Singapore, easily we are having about 2,000 cases every year. Okay, this is based on the MOH, Ministry of Health information. And there are more than 2,500 stereotypes has been described based on different human pathogenic stereotypes. So stereotype is actually, or the stereotyping, it is where the core of all the public health monitoring. So I also list down some news back to 2016, 2018. So you can see how bad the Salmonella contaminants can actually causes the sickness, the food industry as well. So when researchers is trying to categorize it, the taxonomy of the biological organisms, a lot of researchers actually is studying the species, strain, and subspecies level, which is right here. Okay. All right, but if you also sometimes we can call it stereotypes as well. So why we are looking at these stereotypes? So for the food poisoning or for the hospital infection perspectives, the species level identification is not going to be sufficient. That's why they have to go down to the species, or they have to go down to the strain level or the stereotypings. Okay, so this is actually to help the researchers or to help the studies, the clinicians to decide or to determine what is the real source of the bacteria contaminations. So even for the bacterial species are identified, but the useful function, the pathogenic properties can be very different, okay? So that is why we are actually looking, or the researchers, they are looking at strain level or the stereotypings. Of course, if you want to refer to the more detailed information from the clinical research point of view, from public health perspectives, you can take a look at the detailed information right here as well. So how the microbial identification workflow like? It is a very simple, very easy workflow. It just contains a few different steps. So for example, the first step, we always have to pick up a single colony, okay? Or you can call it a culture media. And after that, we have to transfer it, apply the bacterial sample onto the plate, those disposable plate. And then we can place the plate directly onto the multi-top system, and which allows the measurement to be automatically started. After that, we going to do is search and against your database. So this is what customer will see as the results. And in the data analysis, the software itself, and you have to import your spectra, which is acquired from the instrument itself. And the workflows for that, it will be comparing the spectrum you acquired through your instruments against the silver spectra, the reference database. After the comparison, the software will automatically process and calculate and share the results on the identified samples. Okay, so this is a very simple, I think 3 to 4 steps of workflow, all right? And here we have actually a sample to explain how the salmonella on the strain solution software can be analyzed. This is actually from Shimazu, the ASMS poster. Okay, so this is about how to improve the multi-top methods in stability and reproducibility of the detection of the biomarkers for the proteinotyping of the salmonella serotypes. So it includes preparation methods and how the single colony sample has been prepared, which is about four steps. Sample preparation is really to start with optimizing of the bacterial concentration, and then you just follow a very simple steps: suspension, sonication, and then you just take out and transfer it onto the multi-top plate. And from the multi-top plate, you will move into the next step because the instrument will automatically acquire the spectra. And then once you have the spectra, you will be searched and against the reference database by using the strain solution software. And this is the whole complete workflow for salmonella on the strain solution identification. Of course, in Shimazu, we are not only limit ourselves to have only one databases, we actually having a few different databases, if you can see from here, okay? And of course, it's based on different serotypes as well. So such databases not only what we are providing to the customer and then you can use it. In fact, the library or the database can be expanded, can be create a new database based on your own research, your own studies as well. By using a similar approach, Shimazu already had a lot of peer-reviewed journals, which is actually explained in details how, for example, how the E. coli 0157, 026, and 0111 are actually being analyzed by using the multi-top technology. What are the differences and how they are look like in terms of the spectral comparisons. And of course, for different bacteria, we have a different peer-reviewed journals as well. So if you're interested, feel free just to Google it, and you can find all of them. So a quick summary here. What is the benefits? What is the features by using the multi-TOF together with our software? So definitely it is a very easy, very flexible operation. So if you're looking at the whole complete workflow, you only need 3 steps for the sample preparation to identification. I hope you still remember. You need to pick up a single colony from your bacteria grow, and then you have to just transfer it onto your plate and then place it directly into the multi-TOF instruments, either the performance, the confidence, or the assurance. And then the instrument will automatically acquire the spectra. After that, it will perform in the software, in the strain solution, the software, and do automatically search and against the reference database. And it is a high throughput screening instrument. It can analyze up to 1,000 samples a day. Of course, it has a reproducible, it has a reliable identification. It is validated using the SuperSpectra database. And of course, it can be conduct discriminations by the different clusterings. So it can do a tracking of the changes and evolutions of the different bacteria as well. And if you're choosing the high-end model, which is the performance, I hope you still remember performance is combining of the TOF and TOF instruments. So the MS2 information is available. So such MSMS information, it can be a good option for structuring analysis, for sequencing analysis as well. And of course, it is capable for identification of the mixed cultures, and it's an open system because you do able to add in the new species to the databases, or if you want, you can create a new database as well. So all these are the features and the benefits from the instrument itself, and this is what we are trying to help our customer to achieve. On top of that, from the software point of view, it also enables the accurate bacteria discrimination by using the software. All right, so it enables bacteria differentiation analysis, and all those string data, the results, it can be actually built up, it can be customized. Or if you want to build up a bacteria string database, you have the option of doing so. And of course, different classification, it is something we also let our customers to have the flexibility to do that. So at the very beginning, we also mentioned the multi-TOF instrument is not just only for bacteria microbial identification application. It can also be applied for proteomics, for biomarker discovery analysis as well. So back to 2018, we actually have one publication on Nature, which you can find from the website. So this is, we're using the high performance plasma analysis, the biomarker analysis for Alzheimer's disease. And this is actually performed on the Shimadzu multi-TOF instruments. So I guess with all this, I'll end my presentation here. It's a very simple, very brief and overview information that you can have some ideas of how the Shimadzu multi-TOF technology and the multi-TOF platform looks like. So now I'll pass the time to Dr. Lu. He will be sharing in details of the microbial identification by using the multi-TOF together with Ceramis database. Thank you very much.
To answer your questions. Okay, so to continue with our last speaker, okay, our last speaker is certainly not the least, is Carlo Cris Aporillo. He is a special science teacher 5 at the Philippine Science High School Eastern Visayas campus. He is a registered medical technologist, a graduate of BS Medical Technology magna cum laude from the Remedios III Romualdez Medical Foundation in Tacloban City. He also pursued his postgraduate studies and was conferred His Master of Science major in microbiology, also magna cum laude, from the University of Santo Tomas. He was trained for molecular techniques as applied in fungal systematics as the Key State Laboratory of Mycology, Institute of Microbiology of the Chinese Academy of Sciences in Beijing, China. This helped him pursue his research specialization in fungal systematics and bioactivities. His paper on diversity and bioactivities of mangrove fungal endophytes from Leyte and Samar, Philippines was recognized as the outstanding science paper for 2021 by the National Academy of Science and Technology. When COVID-19 broke out in 2020, he pioneered the establishment of the Eastern Visayas Regional COVID-19 Testing Center, one of the few laboratories licensed by DOH during the early days of the pandemic. He also served as the consultant of two other COVID testing centers in Eastern Visayas. As an educator, he has been awarded the Outstanding Faculty of Philippine Science High School for multiple years. He was also awarded Inspirational Teacher by the Massachusetts Institute of Technology in 2011. He is active in several professional organizations, including PNMCC, and has served as the president of PNMCC in 2021, and he is currently our public relation officers. He's also the vice president of the Philippine Association of Medical Technologists in Leyte Biliran chapter and a member of the board of directors of the Biology Teachers Association of the Philippines, BIOTA. He's an active member of the American Society for Microbiology, and he served as resource person in several trainings in molecular techniques conducted by ASM in various parts of the Philippines. He has published a number of papers in microbiology and has served as a resource person in various conferences and training, sharing his expertise on research, microbiology, fungal systematics, molecular techniques, and molecular diagnostics. Ladies and gentlemen, let us all welcome Mr. Carlo Chris Aparillo, who will be talking on molecular techniques for microbial identification.
Magandang umaga po sa inyong lahat. Good morning, everyone. Thank you for joining us in this pre-convention of the Philippine Society for Microbiology in partnership with the Philippine Network of Microbial Culture Collections. At PNMCC, we are mostly concerned with the preservation of microorganisms in culture collections. And of course, if we wish to preserve the microorganisms in culture collections, then we want to identify them properly, or else we will be committing a serious mistake in cataloging our microorganisms. And so for this pre-convention, we thought about delivering or looking back at the techniques that we use in identifying microorganisms. In this lecture, we are going to talk about the molecular techniques for microbial identification. Earlier, you were able to witness or you were able to hear the lectures, the previous lectures, which focus on techniques on identifying microorganisms. And those techniques are actually based on examination of expressed characteristics. So earlier, you were able to see the Vitek system, the Biolog system, and even the MALDI-TOF. So in those previous lectures, you were able to see how these systems are usually used in identifying microorganisms. And if you will notice, as I have mentioned, the identification principles of these systems are usually based on expressed characteristics, which means that these are characteristics that are based on the genes of the organisms that have been expressed. So it's mostly based on biochemical reactions or metabolism or based on proteins. Okay. So aside from these data that we get from expressed characteristics, we can actually add another source of information in order to identify the microorganisms. And since its revolutionary start in the sciences, this had actually been somewhat required already as data to add in identifying the microorganism. And I'm talking about the use of molecular techniques. So, because, for example, in the usual tests that we have for bacteria, for example, for the catalase test, which is a very common test in the bacteria, which differentiates Staphylococcus from Streptococcus, you'll see that it is an enzyme that we are testing, the work of the catalase enzyme that we are testing. And catalase is an expressed, is a protein. So it is actually based on a particular gene in order to be expressed. Also, we use a lot of biochemical tests such as the citrate utilization test, which can determine if the bacteria uses citrate as a source of energy. And the bacteria is able to do that because of the presence of citrate permease, which is again another enzyme and a protein. And again, it's based on a gene. So basically, our identification is mostly based on expressed characteristics and also, of course, on morphocultural characteristics. But in medical microbiology, for example, the algorithm for identifying bacteria is quite simple because we follow rules. For example, if it's Enterobacteriaceae, we use IMVIC, and then we use some more other tests after that. So it's quite, it's already a step-by-step process because we are somewhat limited to the usual microorganisms that we can isolate from humans. However, for others who are working on microorganisms from the environment, we don't usually have, a step-by-step process to do identification based on biochemical tests and based on metabolic properties. But rather, we use our best friend. I'm sure you're very familiar with Burgess' Manual of Determinative Bacteriology. We look at the properties of a particular group of bacteria or a specific bacteria, and we do all those tests that Burgess recommends in order for us to be able to identify the particular microorganism. Now, unfortunately for fungi, we don't have many biochemical tests, so our identification usually depends on the morphocultural characteristics, as you can see from the picture, and then how it appears on the plate, and then hyphal characteristics of the conidia. Those are very important, and we do measurements in order to know, in order to compare it to what is found in the literature, and that is how we identify fungi. So actually, identification of fungi is much more challenging than bacteria because we don't usually have lots of tests that we can do for fungi. So we usually base it on their morphocultural characteristics. Okay, so it doesn't mean to say that these are inferior techniques when we identify microorganisms. Of course, these are all very important techniques also that we have. However, we can further supplement it by adding another information, and that information will now come from the genetic material, which is the basis for all of those expressed characteristics. So I'm now talking about using DNA for identification of the microorganisms. So we can now use DNA, and that is what we will be talking about in this lecture. Earlier, you saw how the different tests are performed in order to see the identification, the identity of a particular, or the species of a particular microorganism. This time, let us look at how DNA is used in identifying microorganisms. So The use of DNA is included in what we call as a polyphasic approach. And when we say polyphasic approach, this means that the data from DNA is not a standalone data that you can use to identify the microorganism. You cannot say that you will not characterize the microorganism anymore because anyway, there's a DNA and then I can just identify it using that. So when we say polyphasic approach, this is a combination of genotypic, chemotaxonomic, and phenotypic. Earlier, we saw phenotypic and chemotaxonomic properties being used in identifying bacteria. And so in this lecture, we focus on the other part of polyphasic approach, which is the genotypic, which means that we will now be focusing on the use of the DNA in order to identify the microorganism. So, in using molecular techniques, I was saying earlier that we need to have a polyphasic approach. But let me just say that there are some methods, especially in medical microbiology, where we don't even need culture. The samples do not even need to be cultured. So that will not be our focus for this talk, because these samples that need not be cultured actually belong to a specific set of microorganisms. So it's easy for them to identify it even without culturing. But for most of the bacteria or microorganisms that we can isolate from the environment or from other sources, we would usually want to culture them. And of course, since we're talking about culture collections, then we are focusing more on culture-based methods. But just to give you an example of how molecular methods are used in medical microbiology, the most famous example that I can give is actually the use of the GeneXpert system. So the GeneXpert system is widely used in the Philippines to detect Mycobacterium tuberculosis infection. So it detects the RPOB gene of the Mycobacterium tuberculosis. So you just have to place the sputum into the sample container, into this cartridge, and then place it on the machine, and then it goes on to do an extraction, then PCR, then matching of that particular gene. And of course, it can also detect rifampicin resistance. So it actually uses 5 molecular beacons in order to achieve that. So there's no culture that is required there, but only because this is very specific for a particular bacteria. So the GeneXpert system can actually be used for other microorganisms and other purposes, even in fact for COVID. But in the Philippines, its more famous use is in the detection of Mycobacterium tuberculosis. There's also the BioFire film array, where you just also place your sample inside the sample holder in the cartridge, and then it goes into a nested PCR, and it can identify common microorganisms that are associated with this, with the respiratory, blood cultures, gastrointestinal. But then again, this is a limited database of microorganisms. It's not something like, something when we isolate from the environment. So we cannot really limit the database for those microorganisms. But this is just an example of some systems that do not use culture, but they also use molecular techniques. So in this case, there's no polyphasic identification because there's even no culture at all. So this really depends on molecular techniques, because again, the database or the group of organisms that are identified are only limited to a particular subgroup of the microorganisms that we are studying. So for our purpose, we are going to discuss the use of DNA for identification of any culture, any microbial culture that we have. So how do we do it? So first, we need to have a pure culture and isolate. It must be a pure culture, of course, so that, we will not have problems in identifying it. And then we do DNA extraction on that culture. So we can have different methods. We can use kits for that. Fungi, for example, are quite difficult to extract because they have a different cell wall, so it's challenging. But in my practice, I've always found that if you use a homogenizer, it's a machine that vigorously shakes the setup together with small glass beads, it would always work for fungi. It would usually work for fungi. Hindi kami nagkakaproblema pag may homogenizer. Pag wala, malaking problema sa fungi because the DNA would not usually be extracted. Then we do PCR. Then after PCR, we test the products if they have amplified in a gel, and then in a gel electrophoresis, and then if they're okay, then we send it for sequencing. So you can have it sequenced here in the Philippines via the Philippine Genome Center, or kung kami, we usually send it pa to Korea for sequencing. And then after that, you'll receive the DNA sequence, and then you can now work on the DNA sequence. So our focus now is not really on the PCR or gel electrophoresis, but what happens when you receive the sequence, because this is where most of the practices actually differ. So we'll discuss how they are actually analyzed. So when we do PCR, what are our target regions? For bacteria, our target region is usually the 16S rRNA. For fungi, our target region is the ITS, or the internal transcribed spacer. So these are universal markers for barcoding of bacteria and fungi. So we usually start with those first. I'm saying first because sometimes it doesn't end there. The analysis does not end there. But if we can get the identification just by using 16S and ITS, then we are very thankful. And for most instances, we can actually do that identification using these genes alone. Okay. So for our primers, for 16S rRNA, our most or baka famous lang ito sa akin because this is the one that I use. I use 27F and 1492R. For ITS, I usually use ITS1 for the forward and ITS4 for the reverse primer. So remember, in in PCR, we use two primers, one forward and one reverse. And so we get two sequences, one forward and one reverse for the reverse primer. So, why do we choose 16S and why do we choose ITS? Actually, hindi naman tayo ang pumili sa kanya. This had been agreed as a barcoding gene for those microorganisms. And why were they chosen as the barcoding gene? Because they satisfy the three characteristics for a gene region that is used for identification. One is that there should be species level genetic variability, which means that that gene region should have differences across different species. However, that same region should also have conserved flanking sites, which means that the region should not be mutating all the time, because if it will mutate most of the time, then that means that the species will not have the same the same DNA sequences already if that is a hypermutable region. So we want a conserved region to serve as our barcoding gene. And 16S, ITS, because they are housekeeping genes, they are usually conserved. Kaya maganda silang marker. In plants and in algae, we also use RBCL. Sa animals, iba naman sa vertebrates, iba sa invertebrates. So we just actually change the marker or the gene marker, and that's when we start to use DNA for identification. And they should have relatively short sequence length in order to be friendly to us para hindi malaki ang data na maproduce as long as it can already help in the identification. For 16S, we usually get 1,000 or 1,000 plus base pairs. For ITS, we usually get about 750 to 800 base pairs when we have it sequenced. Okay, so that's basically the reason why we use those gene regions. So again, 16S for bacteria, ITS for fungi, but these are just our starting genes. Pwede kasi na hindi natin makuha ang identification using those genes alone. Pinapaunahan ko na ang kwento. Okay. So, what you'll get from the sequencing facility is a file. So, they don't need to send anything. Mag-email lang sila and you are given a file, and that file will have a .ab1 extension. So, dahil dalawa yon, meron kang ITS1, meron kang ITS4, so you get 2 files for 1 sample. So, dalawa sila. So, if you're asked for 1 sample, you need to have 2 reactions. Isang forward, isang reverse. So, yon. So, this is what we usually see in order to check the quality of our sequencing. Pag maganda ang sequencing, ganyan ang nakikita mo, which you'll see that there are distinct peaks in your chromatogram. So, pag ito ay magulo na may nag-overlap dito, like in this part, that's not a good sequence. But, it's always normal na hindi maganda ang beginning and end ng sequencing. It's normal. If you'll notice, the body of this sequencing is quite clean. So, masaya na kami dyan. Okay. Now, so that means that if you see that in your chromatogram, then that means you can already work on that. If it's bad, then you have to repeat the process. So there's always prayer involved in doing this because, pagkatapos mo siyang ipa-sequence, it will take about 5 days, and then pag hindi maganda ang sequence, ulitin mo na naman. So that is a challenge. So we have to do it very well always. Then we can now analyze the sequence. So the steps in sequence analysis would include: we assemble the DNA, we do a blast search, and most people will stop here, but that is not the correct way to do things. Then we do sequence alignment, and we finish it up with phylogenetic analysis. So we always recommend to do a phylogenetic analysis on our data and not just stop at BLAST, because the BLAST would only give you the closest matches to your sequence, and sometimes it even gives you different species in your search. So, and sometimes they have the same e-value. So how do you choose? Of course, you can use the biochemical characteristics. Yes, it's possible, but we always have to subject the sequences to an analysis. Para bang meron tayong data, hindi natin masabi kung significant ba ang difference. What do? We have to use statistics. It's the same with working with DNA. We have to subject this DNA data into statistics. Hindi lang pairwise comparisons, right? So let's start. These are the softwares that we need in order to fulfill those tasks. So for DNA assembly, I would recommend DNA Baser kahit hindi siya free. Hindi siya free, pero pwede siyang magamit ng free basta pa isa-isang assembly lang ang gagawin. Fine. Then for blast search, you just need a browser for that. For sequence alignment and phylogenetic analysis, I would be recommending already ngPhylogeny, although in the past I would usually recommend Mega. I do not also use Mega. I do not recommend Mega now, even for beginners, because I believe that the software has become slower, or I don't know if my processor is the one that is slow, basta in my experience it had become slower, and medyo maraming beses na rin siyang nagcrash. Well, Mega had been like that naman. Although, nagagamit siya. Pero ngayon, I have found NG phylogeny to be simpler actually. Kasi ang ginagamit talaga namin for maximum likelihood is the phi-mil pipeline. So, NG phylogeny, it uses the phi-mil pipeline. I will show you that later. So, let's start with DNA assembly. So, sa DNA assembly, technically, ang ginagawa natin ay ina-assemble lang natin ang forward at ang reverse na sequences. We're just assembling them. So we use DNA baser. So this is what we do. When you have DNA baser, you simply click single config assembly, then ilalagay mo ang dalawang sequences, ang forward and the reverse, and then you place kung saan mo siya ilalagay sa desktop, and then you place the file name, and then click start sequence assembly, and then tapos na ang assembly. Okay? You can do that in BioEdit, pero medyo manual siya. So here you will see that you have the reverse sequence here, and this is the forward sequence. ITS5 ang ginamit for forward in this particular example. So you'll see that it's the one that decides na i-discard niya ang pangit na sequences, i-discard niya ito, and it will match it to the bases on the other side, kung match ba ang yung forward and reverse. At magrereklamo yan pag hindi matched ang yung forward and reverse, because they should match, okay? So, pag hindi siya na-assemble ng DNA baser, that means that your sequence is not good. So, now, ito na ang ating config, yung na-assemble na DNA ng DNA baser. And we can use that in the next step of the analysis. Okay? So, basically, our output for DNA assembly is a contiguous DNA sequence na nasa FASTA format. Pasta, hindi po pasta kasi spaghetti yon. Okay? So, FASTA. So, once we have our sequence in FASTA format, then we can now do a BLAST search. What is a BLAST search? We will search a database, usually the NCBI database. So, BLAST stands for Basic Local Alignment Search Tool. So, this is basically what we do. Kung hindi mo alam kung nasaan ang BLAST, i-enter mo sa Google. Dahil sikat ang BLAST, siya ang mauunang mag-appear. Or mauuna lang siyang nag-appear sa akin kasi palagi ko siyang vinivisit. Click nucleotide BLAST, and then you're going to upload here, dito. I-upload nyo ang inyong file. Yung na-assemble kanina from the DNA baser. You upload it there. And then you click nucleotide collection, and then you choose highly similar sequences or megablast, and then click blast. And so the search will start, and the time it will take for the results to appear will depend on the traffic in the blast database. Kung marami ang gumagamit, matagal. So after that, you will now see the samples or the species that are closely related to your sequence. Okay? So, basically, kami, ginagamit lang namin ito to see to which genus our sample will belong. So, for example, ito, then we are sure that it belongs to Phyllosticta. As to the species, it is something that should be solved by using phylogenetic analysis. So we do not choose the number one species there because sometimes it's not the correct identification, or should I say most of the time it is not the correct ID after doing the phylogenetic analysis. So you can click on this part here in order for you to see the information about that sequence, kung sino ang nag-upload sa kanya, because it's very important that we only use reliable sequences. I will talk about it later. So, after that, we can now do sequence alignment. Ano ang sequence alignment? Meaning, i-align mo na ang yung sequence to known sequences. And in order to do a sequence alignment, a good backbone tree is needed. A good backbone tree is needed. Anong ibig sabihin ng backbone tree? It is a tree that contains the DNA sequences of different species. Pero these are very reliable sequences. Kaya kailangan ng good backbone tree. A good backbone tree is very important. If we miss a good backbone tree, pag kumuha ka lang ng kahit anong sequence in BLAST, in the NCBI database, this will not result in a good analysis. This had always been my, when I was starting my phylogenetic analysis journey, I would usually just pick anything from the database, and then hindi maganda ang kinalalabasan. We should use reliable sequences. Now, fortunately for us who are working in fungi, there we have may gumawa na ng backbone trees for important phytopathogenic genera. So for example, if we're looking at Curvularia, we're looking at Coleotricum, we now have good backbone trees. Dun kami usually pumupunta sa backbone trees ng publication na to by Dr. Hyde and company. So kasama dito ang professor ko at IMCAS, si Dr. Lei Chai. So this is one secret. Now, and this is one secret that I've also learned when I started my phylogenetic analysis when I was at the in Beijing, at the IMCAS in Beijing. So my lab mates there would usually give me the backbone tree. For example, if I have Coleotricum, they say, "Oh, Carlo, here are the files for Coleotricum." So all I need to do is place my unknown into their backbone tree, and then I can do the analysis. Actually, it takes time to make a backbone tree. Ito ang medyo mahirap. Now, if you don't have friends who will give you backbone trees, to find a good backbone tree, then you need to look for publications with a backbone, with a tree with your unknown. For example, your unknown is, let's say, Grignardia. Maghanap ka ng publication na may phylogenetic tree ng Grignardia, because those would be usually that would be a good backbone tree. Those would be reliable sequences. Again, I discourage the use of just any sequence picked from the database. Kailangan backbone tree should be a published backbone tree. That is I think the most important thing that we should do. Now, once you have a backbone tree, download mo lahat ng sequences ng nasa backbone tree and place it in one file. So you can just place it in a notepad file for those using Windows, or in a text edit file for those using Mac. This is an example of an aligned sequence. Okay, this is from Mega, so it's colorful. Okay, ito ang maganda sa Mega, it's colorful. So you'll see that the sequences are aligned. Yung pare-parehong bases are aligned. That is sequence alignment. But since we will be using NG phylogeny later, we will not do the alignment ourselves. Isasama na siya sa NG phylogeny. So the last thing to do is do a phylogenetic analysis. Now, you will use NG phylogeny for this, and we will also use Mr. Bayes kung kailangan natin ng posterior probability. So what is a phylogenetic analysis? This is actually a study of the evolutionary development of a species or a group of organisms. So basically, our output is a phylogenetic tree, as what you can see from this presentation, no? So basically, if the organisms are placed in one clade, that means that they have evolved in the same manner based on their DNA, ano? So nandun sila sa the same position. So that means that they must belong to the same species. So yun lang ang tinitingnan natin. So we have here numbers on the branch. It gives you the probability that that particular node is actually present. So for example, here, you're given a probability of 1. So parang 100% sure ang software, ang statistical test, that the branch really exists. And if you notice, the unknown here is this M2.6.3. It is grouped with Silaria cubensis in just one clade. So it is safe to say that molecularly, that unknown is Silaria cubensis because it is supported by a good maximum likelihood. Okay? So ganon ang ating interpretation ng phylogenetic analysis. Now, in phylogenetic analysis, we can use different types of statistics. Usually, we recommend paired ang gamit. So kaya you'll see dalawang numbers in every node. Okay? So one is maximum likelihood and the other one is posterior probability or Bayesian posterior probability. To compute the maximum likelihood, gamit tayo ng NG phylogeny, which I will show you in this presentation. And to compute the Bayesian posterior probability, gagamit tayo ng Mr. Bayes, which I will not be showing you in this presentation. Kasi medyo boring ang Mr. Bayes kasi it's a word, tinatype mo ang command mo. It does not have a colorful interface. Okay, so I'll be showing you only maximum likelihood. We can also have a partnership of maximum parsimony and Bayesian posterior probability. So it's either ML, MB, or MP, MB. Okay? So, but we don't usually use maximum parsimony kasi ang ginagamit naman nun ay ang POPE or parsimony analysis, phylogenetic analysis using parsimony na paid software. So, dun tayo sa free. Okay? So, we use maximum likelihood and Bayesian probability. So, this is how we use NG phylogeny, no? All you need to do is click this FIMIL, SMS one click, masusundan nyo naman. Then we load the file with our backbone tree and our unknown. Then math is for alignment. I suggest that you adjust the direction so that the software can see kung may baliktad na naka-reverse na sequence. And then let us, you can use Bootstrap. We're supposed to use Bootstrap, but in this presentation, I did not use Bootstrap because it takes time pag gumagamit tayo ng Bootstrap computation. So, para mabilis lang siya, I just used the default. So, you will see that it says analysis is being analyzed on the Galaxy server. That is another advantage of using NG phylogeny because the analysis does not occur on your computer. So, pwede kahit medyo mabagal ang computer mo kasi nasa server nila ang analysis. This is our issue with making phylogenetic trees because some of the softwares are localized on your computer. Kailangan maganda ang processor mo or else it will take a long time for it to finish the computations. In fact, sometimes we really leave the computer for about overnight or minsan 24 hours. In our lab in IMCAS, merong computer lang na dedicated for phylogenetic analysis, but it's better if it is analyzed on a server, on their server para mas mabilis. So hindi talaga to real time because I actually cut that video to make it shorter. It takes a considerable time before this is actually finished. So now it's already finished. You will see here, this is the cleaned sequences. Okay, so yung sequences na, yung alignment na ito na na-clean up na so that only the more important data are retained. So if you'll notice, hindi siya colored, it's black and white, hindi kagaya kanina na, pero it still shows you the alignment. Nakikita pa rin naman ang alignment. Okay, so after that, there's also a test for the best model because there are many different models of evolution that are applicable for your data. So, tinitest din ng software kung ano ang best model. In this one, ang lumabas na best model is GTR or General Time Reversible, and then the distribution is gamma. So, GTR plus G. So, yan ang ginamit niya in the analysis. In Mega, tayo pa rin ang nagsiselect ng model na yon. And then after that, you will now have your phylogenetic tree. So this is the phylogenetic tree that was derived from that analysis. You will see that, okay, so let's just wait for the maximum likelihood values to appear. Okay? So it's already there. Enlarge lang natin ng konti. Okay. So you will now see that this is our unknown here. This is M1.1.3. And that is found in a clade. Itong clade na ito na binubuo ng Grignardia mangiferae, and their maximum likelihood is 1. Which means again that the computation says that the probability of that node being there is a 1. So we are sure, based on the phylogenetic analysis, that that particular DNA sequence is mostly identical with Grignardia mangiferae. And so it is safe for us to say that it is indeed M1.1.3 is Grignardia mangiferae. That is how we do the analysis for a DNA sequence. Hindi hanggang blast lang. And then after that, we just match it with the morphocultural characteristics. Again, babalik tayo sa morphocultural characteristics because we will look at the publications or the taxonomic book, taxonomic description ng Grignardia mangiferae, and then we have to match it to our morphocultural characteristics. Unfortunately, when we're working with fungal endophytes, wala kaming masyadong mahirap mag-sporulate ang mga samples. So sometimes all we can match are the hyphal characteristics, the cultural characteristics. So in the NG phylogeny, ito ang kanyang mga publications na nandun. So hindi siya parang ano lang, practice yung NG phylogeny because this is really based on real studies. So I will just disappear for a while so that you can see the entire text. Okay, so you'll see that the one that is used for visualizing the tree is the Newick Utilities, published in 2010. And 2013, ang MAFT or Multiple Sequence Alignment Software, that's the one that is used for alignment. Then you have new algorithms and methods to estimate maximum likelihood of phylogenies, okay, using PhyMil. So yan ang ginagamit to compute the maximum likelihood. And then phylogenetic bootstrap, if you use bootstrapping, and then smart model selection in PhyMil in order to select the best model, then block mapping and gathering with entropy or BMGE to clean up the alignment. Okay, this is used for selection of phylogenetic informative regions from multiple sequence alignments. And of course, yung buong collection niya, which is the NG phylogeny, pinagsama-sama niya ang mga tools na ito so that we will not be stressed when we're doing phylogenetic analysis. So this is for non-specialists because di ba, parang nakikigamit lang tayo, ginagamit lang natin ang tool na yan, but we're not really good at the statistics behind that. Parang nakikigamit tayo ng ANOVA, pero pag sinabi sa'yo, ano ang assumptions ng ANOVA? Bakit nagagamit ang ANOVA? Bakit pag ang p-value is less than 0.05, that is significant? So, something like that. So parang alam natin paano gamitin, but we can not actually usually discuss what's going on behind what computations are going on behind it. Parang isang semester ulit na course yun. So that is the basis of doing the phylogenetic analysis. So pag natapos na yon, again, babalik na naman ulit. You have to match your results in the phylogenetic analysis with your morphocultural or biochemical kung meron kang biochemical, and they should all match. Okay? Pag hindi sila mag-match, then you have to resolve it. Now, what if identification is not resolved after phylogenetic analysis? You need to add more information by using other genes. For example, very sa fungi, the Coleotrichum is a big species complex. So, usually hindi siya nasusolve by only using ITS. So what we do is we use GAPDH, CHS, HIS, ACT, tubulin, and APMAT. Although, I think 2 years or a year ago, may publication na lumabas na APMAT is enough to differentiate the Coleotrichum from all the species. Okay? But we have not yet used this na AP Mat lang ang ginagamit. Usually, pag Coledotricum, magastos talaga siya kasi marami ang genes na dinadagdag. So, if you have these sequences, idadagdag mo lang yun sa sequence mo at idadagdag mo rin ang kanilang sequences dito sa baba ng other species in the analysis. So, isang analysis lang technically ang lalabas in that phylogenetic analysis. So, this is an example of Coledotricum where we used ITS, CalTube, GAPDH, APMAT, ACT. So, you will see here itong B4.6.1, B4.6.2 is in one clade with Coledotricum fructicula. So the outermost clade is here. So it's 0.97 in maximum likelihood, 0.93 sa Bayesian. So our cutoff for that pala is 0.75. So if it's higher than 0.75, then it's good. But you know, for maximum likelihood, it's 0.75. For Bayesian, it's 0.9. Yun ang parang arbitrary cutoff that we have set. But we would always want it to be 0.9 and above as much as possible. So, and these are the morphocultural characteristics of the isolate. Then, another example for Pestalozopsis, if you cannot solve it by ITS, then magdagdag ka ng TEF at tubulin. Okay? This is our example for Pestalozopsis. You'll see that they have unique conidia. Maganda siya. Okay, this is the culture. And then, it's Pestalozopsis adusta 1-1. So, you are molecularly sure that this Pestalozopsis adusta. Then you have here you see here a very big tree because it contains all the species of Diaporthe. But despite being matched to all the species of Diaporthe, you will see that our sample here makes a clade of its own. It does not group with other species, even if you already have all the species of Diaporthe in that analysis. And that tells us that, you know, this is a possible novel species. This is a molecular proof that it's a novel species. Now, molecular proof is still very far from reporting a novel species, although the molecular proof is needed to report a novel species, pero kailangan mo rin lahat ng characteristics ng organisms, you should be able to describe it. So, yan ang hindi nagawa for this. So hanggang dyan na lang siya sa aking presentation. So, plus of course, this is a fungal endophyte. So it's quite difficult for them to sporulate most of the time. So, and then, kailangan mo siyang i-deposit in 3 culture collections in different locations. So, parang hindi ito na-report as a novel species. So, together with my students, we've also been working on fungal endophytes in mangroves. So these are some of the isolates that they were able to report. So makikita nyo, palaging may cultural characteristic, and we also report the host. So here it's coming from Soneracia alba. This is Arthrinium malaysianum, 0.97 and 1. So we're kind of good with that identification already. Although, if you'll notice, walang masyadong conidia to kasi this is the time na naabutan ng pandemic. So, they were not able to finish this. Molecular lang. So, parang ito ay parang blind na identification because we only have the culture, the cultural characteristics, and then molecular. So, kailangan mataas palagi ang values. Then we have Philinos bicuspidatus, isolated from Soneracia alba. This is 0.99 and 1. And you'll see na convincing naman ang kanyang tree. Okay? This is from Avicennia officinalis. You have Pithomyces mydicus, 1 and 1. It's also, so you're also confident with that ID. This is Coledotricum queenslandicum. We used many genes here, kaya na-solve namin ang position nito. We were able to report Coledotricum queenslandicum, 0.99 and 1. then we're not lucky all the time. Look at the schizophylum. We could not solve it because there are no schizophylum sequences in. There are only I think 3 species which has sequences in the NCBI, so we could not report them. So since we could not finish characterization up to spores and conidia kasi kailangan silang i-feed para mag-sporulate, so hanggang dyan na lang siya, Schizophylum sp. Kasi yun ang blast, Schizophylum siya. So we're safe that it's Schizophylum. And then, despite the use of many genes, itong Coledotricum namin, hindi pa rin talaga siya na-identify. So, things happen like that. So, kailangan pa ng more data. And since nga, ito silang students na na-cut during the pandemic, hindi na rin natapos. And there are samples na hindi namin ma-extract ang DNA, hindi rin namin alam kung bakit. So, naging Mycelia sterilia na lang siya. Okay? So, hindi na rin kasi ito na-solve kasi nga na-cut during the pandemic. So, those are some of the applications that we have in identifying fungi using ITS and other genes. We're also working on bacteria. So, my students have isolated bacteria from the soil. Ito ay also ang walang masyadong test na ginamit dito, molecular lang, and then gram stain and cultural characteristics. So we were able to have Lysinibiacillus macroides 0.92, so mataas naman. So we're and then it matches naman the expected shape and gram reaction. So we're quite good with that. Ito ang aming hindi talaga sure. So we're not yet done with this. Kasi ito ay bacillus and they are difficult to identify just by using 16S. So you'll notice this 1.8 lang ang kanyang pasok na rin siya kasi 0.75 lang naman ang requirement, but we would need more morphocultural, biochemical properties to match that. Kailangan may more proof kasi medyo mababa ang kanyang maximum likelihood. Ito naman na isolates ay Bacillus megatherium 0.88, Bacillus paraflexus 0.89, medyo pwede na. Ang medyo mataas lang dito is Bacillus stichilensis 0.99. So kung parang walang ibang basis, at least we're confident with this already because it's a 0.99 na on maximum likelihood. So yung iba, we really need to match it with other properties. So that's how we use the DNA to identify these microorganisms. So basically, what we discussed is multi-gene sequencing. Okay, the use of DNA and sequencing DNA and doing phylogenetic analysis. So again, we boil down to the same thing, that this is part only of the polyphasic approaches. Okay, so it cannot stand on its own. Hindi pwedeng siya lang ang data for your identification. It should always be matched with morphology, of course, morphocultural characteristics, physiology na nakita nyo kanina. Pwedeng ang gamitan dito is your Vitec and your Biolog. And also, kung merong kang equipment, use MALDI-TOF pa rin to match what you have in your DNA sequencing. So that would be the best approach to identify a microorganism. That is why in culture collections, we always also recommend a polyphasic approach. So maraming data ang ginagamit para malaman natin na ito talagang ID, ito talaga ang identification ng microorganism that you are submitting to the culture collection. Okay? So I hope that you learned new inputs in this talk. And if there are questions, I would gladly answer your questions. So the talk on molecular identification will complete the whole package of identifying microorganisms. So from the express traits to the genetic traits, we should collect those data so that we are sure about the identification of the microorganism that we're working on. Again, on the part of PNMCC, we are pushing for always the right identification of our microorganisms, because we cannot keep microorganisms with a name and that name is a wrong name. So that is a very serious mistake on our part. And this is the reason why we decided to again review the identification of microorganisms in this pre-convention. Again, maraming salamat po. If you have questions, we would surely answer your question after this talk. Thank you very much.
The 51st Annual Convention and Scientific Meeting is made possible.
Thank you very much, Chris, for that very extensive and informative lecture on molecular techniques in microbial identification. I know everybody has a lot of questions, but before we go into the open forum, may we request everybody to first turn on their cam for our photo op. And then after that, we'll have a 5-minute break. And then we will then proceed with the open forum to answer some of the questions for today. Okay, so can you please turn on your cams? Tech team from Trinity, how will you do this? Okay, we have several frames. Miss Tin. Who will take the picture from Trinity? Miss Tin. Just tell us when to smile and when to stop smiling. Everybody, can you please turn on your cam?
Hello. Pages. So
Cannot open the cam.
Kindly hold your smile.
Disabled po.
Wait lang.
Disabled.
Okay.
Disabled daw yung iba.
On it. Yes,
Okay
po,
na.
we're working on it.
Okay.
So we have 8 pages right now. So kindly hold your smiles. Ready for first page. 1, 2, 3, smile. Second page. 1, 2, 3, smile. Third page. Ready? 1, 2, 3, smile. We're going on the fourth page. Ready? 1, 2, 3, smile. Fifth page. Okay. Just taking the 6, 7, and 8. Seventh page. And last page. Thank you very much.
Okay, thank you very much, tech team. Okay, we will be having a 5-minute bathroom break. Can you please come back after 5 minutes? We will then be starting our open forum. Time check, it's 11:12.
The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors.
To please turn on their cameras, and then we will also request the tech team to put our speakers on spotlight. Okay, so we have a lot of questions. Right now, we'll try to limit and make sure that it's equally distributed from our speakers. So we will be starting. We have a question here for our first speaker, Dr. Siringan. In comparison with other methods of phenotypic characterization, such as analytical profile index kits, conventional preparation of media, such as carbohydrate oxidation fermentation tubes, how does the biolog system compare against these procedures in terms of, number one, cost and labor efficiency, number two, available number of species described in the databases, and number three, reliability and reproducibility of the results?
Thank you for that question. It's a big question, and I don't really have a clear answer on this. If you will think about preparing for the different culture media, no? And then you will also use the biolog. Of course, the biolog, you will have like 3 days to finish it. But there's a limitation on the shelf life. Of course, the equipment is expensive. Used to be 3 million, and if you have the Omnilog, that will be about 12 to 15 million, and then the maintenance is also expensive. But if you also do the conventional biochemical tests, of course, you have to buy the different culture media, which are also expensive. So maybe 5 to 6,000, depending on the culture media per bottle. And there's expiration, and you have to use different. You need also the incubators. So I don't have a comparative analysis on this in terms of the species, the databases. Biolog says that it has more than 2,900 species in there, and that's including the mesophilic, aerobic, gram-negative, and gram-positive bacteria, and the filamentous fungi, and the yeast and anaerobic bacteria. They are actually updating it, but for you to get the updated database, you have to buy the database. So everything has
the 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble,
so in terms of
ESCO,
reliability and
3M Philippines, and
again, these are
and our minor
methods. For example,
sponsors. The 51st Annual Convention and Scientific
We cannot say that this is best or what. That's why we have emphasized, I think, in most of the lectures that the way for us to identify is polyphasic approach for identifying microorganisms. And I think you have to look into several characteristics: the phenotypic, the chemotaxonomic, the genotypic. Let me just put in perspective. When I was in Chiba in the Biological Resource Center, it says that first they do the molecular identification. So you have S or ITS or ATS or whatever marker for phylogenetic analysis. Once they determine the genus or the species, then they go with Malditos and biolog. So it's like a reverse approach, so that when you publish for a new species, all of this information are needed. Okay, if you have new species, for example, you go to the say International Journal of Environmental and Systematic Microbiology, and it will tell you like what are the minimum traits that you have to report. And so that's why you use different methods. So again, for this particular question, I don't have any answer. There's another question about for microorganisms. So can I just read it?
Yeah, please. Yes, please, ma'am.
From the environment, is ambient temperature not sufficient for the incubation? These are optimized protocols. So if Gen 3 for aerobic mesophilic bacteria, if it says 33 degrees Celsius, then do so. If there are variations, you have to validate it in the laboratory. Actually, when I asked about using biolog for marine isolates. They don't have any clear-cut protocol for that. So they said that you have to grow it in the marine bacteria, the medium for the marine bacteria, and then just apply it to gently microplate. So it's actually tailor-made for all of the species that they have studied, and metabolic fingerprints are stored in the database. So to remember whether you use Malditov or Vitec or Biolog for this microbial identification system with database, the limitation is in the size of the database. So whatever they have inputted there, that will be their reference. So you will just have to look for the right match. For the third question, that's about if you have.
It's one about pigments, yeah.
Producing pigment. This question. So I look into the database of the gentry and Serratia marcescens is here. So if you follow the protocol and it's prescribed, you have to use the PUG. Normally we use the PSA. Maybe we need to use the PUG. Maybe you have to purchase from Biolog also. Maybe there will be no interference in the color reaction. So if they have in the database Serratia marcescens, then I think there should be no problem with the pigmented bacterium. So actually, I have here an example of the result of Serratia marcescens. But my Serratia marcescens does not have any pigment, so I did not encounter any problem. Now, for the using dual wavelength reading for the FF and the yeast, I don't know why. What's the question? What are the possible?
The question is, in using dual wavelength reading for filamentous fungi and yeast, what are the possible sources of errors that might be encountered in such wavelengths, and how are they being addressed? The errors.
For the filamentous microplate, filamentous fungi and yeast, there are two wavelengths. The 490 for the filamentous fungi, and for the yeast is 590. That's for the utilization of the carbon source. Then for the 750 nanometer is for the growth. So you are reading two characteristics of the organism: the ability to utilize the carbon source and the ability for it to grow. So in that particular substrate. So there's a software there, it's an algorithm that analyzes, and they will just present the fingerprint, the metabolic fingerprint, and that's what you get. And the wavelength actually is adjusted in the, you have to select that in your microstation when you read it. So I don't know if, well, possible errors, maybe you need to have preventive maintenance for the filter. You have to have annual preventive maintenance for the filter and for the reading. If there's error actually in reading which we encounter, it will say in the display, and you have to contact the technician and they will advise on some troubleshooting. Otherwise, you have to have check very your problems.
Okay.
Now, this in general, just the generation of bacteria.
Yeah, that's the last question.
There are two more after this.
Two more.
Phenotypic characterization. You mean if you have several passage of the organism, do you have changes in the phenotypic traits? Is that the question? Well, we haven't really, because it's too expensive to really authenticate. We're now trying to authenticate if funds are available. It means that when you isolate this organism, do the biolog, and then maybe after several subculture passage, you can do the biolog. But I don't have any data. They have changes in the phenotypic traits for now. Now for the ground flu, can I share screen? There's a question about the carbon substrates.
Example of the carbon substrate.
Yes.
Yeah.
Okay.
Can I share screen? It's disabled. Okay na?
I think is it disabled at this point?
Oh,
The share screen?
Yes.
Hello, ma'am. You can now share your screen.
I
Yeah.
can. Okay. Okay.
Yes,
Can you
po.
see my screen?
Yes, we
Okay.
can see your screen, ma'am.
So this is the Gen 3 microplate for aerobic mesophilic bacteria. So I'm emphasizing mesophilic because your temperature, incubation temperature is 33 degrees Celsius. So if you look at this, there are the extreme, maltose, trihalose, cellular bios. And what you're measuring is actually the ability of the organism to utilize. There are no enzymatic traits that are being detected here. So it's just the ability to utilize. So it's respiring, so it's energy producing. So you have raffinose, you have glucose, you have lactose. These are from column 1 to 9. For column 10 to 12, you have some sensitivity assay where you have pH 6 and pH 5, and then different concentrations of sodium chloride. You have 1% sodium lactate and some antibiotics. Okay, so let me share with you this data set for an isolate, which is a serratia process. These are the carbon sources that the organism utilized. And from this, if you are going into drug discovery or metabolite production, you can utilize this information to determine which carbon sources can be used by the organism. So we have here some information on the sensitivity assay and the antimicrobial resistance. So the set of antibiotics there in the gentle plate is not that extensive, no, but you have some here. There's a question about the similarity index. So I'm showing here this actual data. So for you to say that the identification could be acceptable, you have to see that the ID number 1 to 4 should be belonging to the same genus or related. So all of these have serratia, right? If you look at the same, that's acceptable, that's greater than 5. But the distance, when you subtract the distance of ID number 2 to ID number 1, it's not or higher. So your decision here is just accept serratia SP. So if you will not really accept the whole the species identification.
Okay.
In the interpretation, for example, there are because of limited time, I do not but I can show you. Stop share ko to, no?
Ma'am, in the interest of time, I'm sorry. So that we'll have, we'll go back to the other questions probably later on. We'll just have the other speakers answer some of the questions. Yeah.
All right.
Okay. So
I would just say that our troubleshooting guidelines for biolog users. Okay. Thank you.
All right. Okay, so thank you very much, Dr. Siringan. Okay, so we have some questions for Ma'am Rose Capanzana. Okay, how often does Biomeru update its database for identifying new microorganisms? This is for the Vitec.
All right, thank you, sir, for that question. I have also consulted with the Biomeru technical team. So they usually update the database or the software every 2 years. So according to them, it takes around 18 months for FDA to validate the new software. In our case, in PNCM, we had our update last 2020, and we have already been informed that they have their updates for this year. So we haven't had our Vitec to check. So we'll work on that soon.
Okay.
Yeah.
Yeah. Another question is, Is Vitek 2 particular on the culture medium from which the colonies are isolated prior to its identification?
Okay, so for that, yes, Vitec2 has this, they have this list of specific culture media to use for each group. So they have also advised in their manual that the results may be affected if the user is tend to use another type of culture media. But it is also said there that the user can do validation of the culture media that they wanted to use outside of what was recommended by the UME team. So in case the different culture media works for them, they have validated it, and the ID is okay, then they can use that. They can continue using that culture media. Thank you very
Just like
much.
the API, you need to do the gram staining first and oxidase or catalase test. Is it the same for Vitek? Do you need, well, of course you need to do the gram staining, but do you need to do additional biochemical tests first before running your samples?
Yes, we have to make sure that the gram reaction has been determined before we subject it to the Vitek2 system.
I have a personal question.
Yes.
Normally, Vitek is used by hospitals, so therefore their cartridge are actually used for pathogens, for pathogenic
Yes,
organisms.
sir.
Is there a possibility of coming up with more, say, for environmental samples or any other samples, food samples? And is it also possible if you want to decide on the specific antibiotics that you would like to be tested together with, is there on the process of upgrading this possibility of, you know, specializing a cartridge for a special group of microorganisms to be detected?
I'm not very much familiar if they can customize like a reagent card for a specific purpose. But all I am familiar is with their AST card. We haven't used it in the laboratory, but in the AST card, you can test your isolate to different antimicrobials. So that, yes, so we can, I can connect you, sir, with the biomerit team, so we can inquire about that, if we can customize reagent cards.
Reagents. In one run, how many samples simultaneously could be identified?
For like for one run, we can like subject 10 suspension, but we can do like up to 60 cards.
60 cards
per batch.
Okay, thank you very much for
Thank you very
answering the
much,
questions. Sir. Okay, so we will then proceed now to the some of the questions for Dr. Lu and Nicole. Okay, so this is for the Malditov. Okay, is the Ceramis database proprietary to Shimadzu and can only be used by the Shimadzu equipment users? Is there exclusivity of the database only to the Shimadzu users? I think that's the question.
Oh, yes, sure. Thanks. Dr. Lu, I'll just take this question first. I think at this moment, yes, it only works with the Shimadzu data. You only work with the data which is generated by the Shimadzu multi-top technology. Yeah.
So there's another question which says, can you further elaborate on how the super spectra can directly identify components of a mixed samples? For instance, does this mean that mixed samples such as environmental samples, food samples, clinical samples, among many others, can be analyzed as it is? Or is this in reference only to mixed colonies in culture plates? You're talking about mixed samples. Can they be analyzed as it is?
Okay,
or you need to purify first?
Yeah, I think we have to clarify this first, because usually we do not mix different types of sample together. We're definitely, like when you have environment samples, you have clinical samples, or even food samples, we will definitely have to separate them. Yeah, so so whenever we are doing the multi-top technology or the analysis on the microbial identifications, we always trying to pick the single cloning from the culture meat rather than the mixture. Yeah, because you know, the multi-top technology, so-called, there is only so-called one type of the separation, which is based on the molecular weight.
Thank you very much.
Thank you.
Here is a very interesting question for a laboratory. Okay, which Axima ID Plus multi-TOF model do you recommend for schools or students starting or even starting researchers? I know this is very expensive. This is a very expensive technology, but they are just asking which one you could probably recommend for starters, a laboratories just starting research.
Okay, maybe I'll start first, and then later, Dr. Lu, if you want to add on, please feel free to add on. I would say if you are really starting to go into the multi-top technology, the benchtop will be a good choice because it's easy to operate, easy to maintain, as well as we have this software which allows the so-called the junior researchers or even the students to work on different projects. Of course, it depends, or it really depends on the specific applications the person or the researcher wants to go in. So if you really want to go in just for specific microbial identification with certain software, then the big piece of the eczema will be the one to go. But of course, there are 3 different models. If the customer really need to go to really the fragmentation stage, that means you need to look at the MS information. And then will be the performance will be really the combining of the top and top technology. That will be the best idea. But of course, considering the financial support, so that's going to be another concern as well. Yeah.
Okay.
Yeah.
Dr. Lu, would you like to add something?
Actually, I think Nico already covered most of the points. So actually, we have a very broad range for the multi-top product. Depending on the application, the skills, and so you can select different product. Yeah, this is a point. Yeah.
Okay. There are questions here which I know you already have answered in our chat box, but for the benefit of the other participants, because we are also seen live and we have participants outside of the Zoom.
Okay.
Okay, so there's a question. I would like to ask if mutations in the AA sequences are also detected and shown as variations in pics. I think this is answered. Maybe just reiterate again.
Pardon, please. I didn't follow with this question because my background is noisy. I'm in the lab. Sorry. Can you just repeat the question?
Yeah. Okay. He would like to ask if mutations in the AA sequences are also detected and shown in variations in pics.
Yes. Actually, if you sum the mutational variation in the amino acid sequence, definitely the whole mass will be changed. So by the multi-top, you can measure the mass shift from the wild type. You can definitely know what kind of the mutation or the amino acid sequence change or the variation. You can identify based on the mass, the difference, the mass shift. Yeah, definitely can do. Yeah.
Okay. And the last question is, where the different strains of particular species always have variation in the ribosomal protein molecules that will allow their identification using their PMF patterns?
Yes, for the strength level, the kind of the differentiation and identification for different species at the strength level is mainly based on the biomarkers, the peptide specific peaks. So this basically based on the theoretical molecular weight calculated based on the amino acid sequence, and compared to the measured one from your real sample in the strength solution, the software, you can identify the biomarkers and used for the clustering analysis. You can definitely to separate the different strengths. Yeah.
Okay, thank you very much, Dr. Lu and Ms. Nicole Zhang, for answering the questions of our participants. At this point, we'll move on to questions for Mr. Chris Apurillo. Okay, we have a lot in here. Would you recommend research publications which utilize BLAST alone and their species identification? Repeat the process of identification using the one you have discussed earlier or a while ago.
I would not recommend anymore. If they're already published, then they're already good. But going on into the future, I hope that we will use the method that I just described when we're using molecular techniques for identification. Plus, of course, I think these were able to work, the ones that only used BLAST, because these are also correlated to the other properties or other characteristics of the microorganisms. I'm sure that they are not just based on the molecular identification.
Okay, another question is, what is the most challenging part using molecular techniques and microbial identification? What would be the limitation as far as your country is concerned? As far as the Philippines is concerned,
I think the limitation is is equipping the lab itself. But once you have the equipment, or you can work in a lab with an equipment, we have actually optimized a method for DNA extraction that does not require kits or require lots of funding. So we also have suppliers for master mixes and primers. They are not really that expensive anymore. So I think molecular ID is there are not really that many challenges anymore for molecular identification. Plus, of course, we have a sequencing facility in the Philippines. If you do not want to use the PGC in the Philippines, you can also send it abroad, and it just takes about 5 days, and the price is just almost the same. So I think it's really easier now to do molecular techniques because there are more laboratories that have capability for molecular biology.
And molecular techniques now is the gold standard identification. So that's why
Yes.
we really need to go into this.
True. And I also picked up what Ma'am Oxy said earlier, that in the identification, they usually use molecular first and then they just backtrack to the morphological, because basically that's what we use also for fungi. So for fungi, we use morphocultural first, then molecular, then that's when we examine already the other properties of the isolates.
Okay. We have a very interesting question in here. This is regarding novel species. Okay, so currently, what is the percent similarity that is needed for species to be considered novel? Do you need to subject the organism to other tests before we report it to be novel, or is it molecular barcode enough to report it as a new species?
Okay, this had also been asked in the chat, and of course, there are different schools of thought in declaring novel species. However, for fungi, we do not look at a level of similarity, kung 75 or 98 or 99. Rather, we do the whole phylogenetic analysis, and you should see, as I have shown earlier, it should form its own clade with a high value on its nodes, and ML or MB or MP or MB. Plus, that is just the first step. You need to describe, you need to document, you need to measure the different parts of the isolate that you are describing, and you need to compare it with existing and closest species of the organism before you're able to actually declare a novel species. So the molecular data is just one of the many data that you need in order to declare a novel species. So you cannot declare a novel species just because you have a phylogenetic tree. It will not be accepted. So kaya medyo there's a lot of work to be done when you are declaring a novel species. And seeing it in a phylogenetic tree is just one of the very many steps in declaring it as a novel species.
And also, not just because it's not deposited in the database, it already means that it's novel.
True, because for example, we have to make sure that at least all of the species in that particular genera are included in your analysis. Because if not, then that means it may just be a species that is not in the database, but it's actually existing already. So that's also one thing. That's why I said that in order to do a phylogenetic analysis, you must have a good backbone tree, and a good backbone tree is a complete tree. So that you'll know that your isolate is a novel species, or at the very least, you will know the identification of your isolates.
We still have a lot of questions, but we'll probably have one more. And then for the other questions, you may contact Chris through his email later on. Okay, so the last question would be, is ML and PP the only three constructing methods that can be used for all studies? What might be the possible data sets or expected results needed to use other phylogenetic tree building methods?
Actually, there are other tree building methods. No, Doc Lin in the chat had actually mentioned neighbor joining trees, NJ. There are actually many algorithms that can be used to build phylogenetic trees. However, what I have described are the ones that we have already used in practice, and these are also the ones that have been kumbaga, naipamana, handed over to me in my training. So we usually observe that by using these trees, we actually get a robust result. So it's usually a paired analysis, so maximum likelihood and Mr. Bayes, because sometimes it appears, for example, it has a good statistics in maximum likelihood, but the node does not appear in Mr. Bayes or in Bayesian probability. So we have to resolve that. So using two statistics, using also two software is for us a very safe way to analyze the DNA sequence. But again, there are many publications that are using other algorithms. So that means that this also works. However, I cannot really comment on them now because I do not personally use them, but they are still the same. They have the same statistics that are being applied to analyzing DNA sequences.
Okay, we wish we had more time, but unfortunately, it's almost 11:50 now. So at this point, may I call on the president of PNMCC, Dr. Ursula Bigol, and the president of PSM, Dr. Donna Papa, for the awarding of certificates to our speakers. Okay. Can I please have a copy of the certificate? Okay, so the certificate reads, the Philippine Network of Microbial Culture Collection, incorporated in partnership with the Philippine Society for Microbiology, awards a certificate of appreciation to Dr. Auxilia T. Siringan, okay, for having delivered a lecture on phenotypic identification using the Biolog microbial identification system during the Preconvention webinar of the 51st Annual Convention and Scientific Meeting of the Philippine Society for Microbiology in cooperation with Trinity University of Asia. Okay, so this is signed by Dr. Howell Ho, the Preconvention chair, and Dr. Donna Papa, the president of PSM. Dr. Siringan, thank you very much po. Okay. So the same certificate, thank you ma'am. The same certificate is given to Ms. Rosemary Ann Capanzana for having delivered her lecture on microbial identification made easier, the Vitek 2 identification system for bacteria and yeast. Okay. Ma'am Capanzana, thank you very much for your time.
Thank you po.
Okay. We also have the same certificate that reads Certificate of Appreciation given to Dr. Lu Yung Hai for having delivered the lecture on Rapid Microbial Identification by Maldi Toff with Ceramis Database during the Preconvention Seminar of the 51st Annual Convention and Scientific Meeting of PSM in cooperation with the Trinity University of Asia. Dr. Lu Yung Hai and Ms. Nicole, thank you very much.
Thank you.
Okay, so the same certificate is given to Ms. Nicole Zhang Li for delivering a lecture on the phenotypic identification. Well, this is, sorry, this is wrong. They have the same title. This is on Malditov, okay? So Ms. Nicole Zhang, thank you very much for your time.
Thank you.
And lastly, the same certificate is given to Mr. Carlo Chris S. Aparillo for having delivered a lecture on molecular techniques for microbial identification during the pre-convention workshop of the 51st Annual Convention of PSM. Thank you very much,
Thank you po.
Mr. Aparillo.
Thank you.
Okay, so with that, okay, so we are almost at the end of our webinar for today. So today we were able to see the different methods of possibly identifying your microbial isolates in the laboratory. So we have discussed first biolog. Dr. Siringan discussed on biolog. Ms. Capanzana discussed on Vitec. We have speakers from Singapore who have discussed, Dr. Lu and Nicole on Malditov, and of course the molecular techniques by Mr. Chris Aparillo. So indeed, this is a very interesting lecture, and this actually the different techniques are there for you to choose which probably is the one that fits the need of the laboratory or the need of your research. All of this are very helpful in our research, and of course there are several considerations that you need to see which one might be the best one that will fit the research that we are actually doing. Given the possible advantages and disadvantages, our speakers were able to lay down to us what are disadvantages and the limitations of the different techniques that were discussed today or this morning. So we really hope that you were able to get some insights of the different techniques that were presented today, and I hope it was very helpful for you in deciding in terms of which one do you need, depending on the cost, depending on the availability of the material, and depending on, you know, the need of the laboratory and the research. Okay, so with that, may I call on our Vice President of PSM and the incoming President, as well as the Vice President for Academic Affairs of the Trinity University of Asia, Dr. Howell Ho, for the closing remarks.
Thank you very much, Sir Joel. Thank you very much to our esteemed speakers and organizers, especially to Dr. Siringan for the crash course on Biolog, and Ms. Capansana for the uses of BTEC, and Dr. Lu and Ms. Sang for the applications of Malditov, and Mr. Aparillo for the micro-identification concepts that were shared to us. For the informative and comprehensive treatment of the topics on the trends in microbial identification that we learned today, we are very thankful to our speakers and organizers. And I'm also grateful to our participants who took the time to attend our pre-conference session this morning. Hoping you learned a lot and appreciated the sharings of our speakers. Enjoy the rest of the day, and encouraging all to attend the other pre-convention activities that we have this afternoon and this early evening. And if you have the time, please participate as well in our exhibits. Visit our exhibit area, visit our poster sessions. And I hope to see you in the two-day conference that we have starting tomorrow, and listen to our plenary session speakers. Have a hearty lunch. Thank you very much to everybody.
Thank you very much, Dr. Howell Ho. With that, thank you
The
very much to
51st Annual
our
Convention and
participants
Scientific
and hope to
Meeting
see
is
you
made possible
in
by
the activities of
our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors.
Good afternoon, everyone. I hope everyone had a hearty lunch and energized for this afternoon's pre-con or pre-convention workshop offered to you by the Philippine Society for Microbiology. So this morning, we had our first workshop. We had several speakers who had very interesting, informative, and extensive presentation about the current trends in microbial identification. This afternoon, we are very fortunate to be joined by two certified biosafety officers who will be presenting to you the biosafety and biosecurity in research and education, pandemic and post-pandemic scenarios. This is Marian De Leon. I'm the current liaison officer of the Philippine Society for Microbiology and a certified biosafety officer. And I will be one of your hosts for this afternoon's workshop. So to formally welcome us all, may I call on the President of the Philippine Society for Microbiology, Dr. Donna May D. Papa, to give us her welcoming remarks.
To our esteemed speakers and participants, good afternoon. On behalf of the Board of Directors of the Philippine Society for Microbiology, it is my great honor to welcome our members, esteemed speakers, and participants to this pre-conference workshop, one of the three pre-conference activities to jumpstart the PSM 51st Annual Convention and Scientific Meeting. This afternoon, the workshop entitled Biosafety and Biosecurity in Research and Education, Pandemic and Post-Pandemic Scenarios is a very timely and a very relevant theme, most especially because of the challenges that this pandemic has confronted or has brought upon us. And with the world now slowly opening again, this workshop is just one of the things that we need to update ourselves and learn. We have a lineup of experts who are trained and are certified biosafety officers who will enlighten us this afternoon with the principles of biosafety and biosecurity. Specifically, we have Dr. Redel Jonathan Vitor II, who will be delivering his talk on program birth or biorisk management in research, teaching, and health, and also on tools to engage and check biosafety. We also have Dr. Fedelin Estrella, who will educate us about code breach, biosecurity in research, education, and community health. I am honestly very eager to learn from this afternoon's talk. My sincerest and heartfelt thanks to Dr. Howell Ho, PSM Vice President and Chair of the Program Committee, and to Dr. Marian Pulido de Leon, PSM Liaison Officer, who is also a certified biosafety officer. Thank you to the National Training Center for Biosafety and Biosecurity of the National Institutes of Health, UP Manila, the focal training center for biosafety and biosecurity in the country for today's workshop. To all the participants, may this virtual event serve as a venue for all of us to know more, learn more, and ask more. We also look forward to your participation in the PSM 51st Annual Convention and Scientific Meeting tomorrow and on Friday. Good afternoon, everyone, and thank you so much.
Thank you very much, President Donna May Papa, for that, for the welcome remarks. And indeed, no, it's very timely that we will be having this lecture presentations from our certified biosafety officers as we slowly opening our research and academic institutions to our students, our researchers, and our partners. So let's start with our first lecturer. Actually, our lecturer will be having two lectures. So our lecturer is Dr. Rodel Jonathan S. Vitor Diseca. He is an assistant professorial lecturer at De La Salle University, Manila, and an adjunct research assistant professor of the National Training Center for Biosafety and Biosecurity. So let us know more about Dr. Vitor. Dr. Vitor is a lifelong learner that strives to upskill in the fields that he is interested in. Currently, he is a postgraduate researcher at the University of Tokyo, specializing in veterinary pathophysiology and animal health. Dr. Vitor is currently involved as adjunct faculty in different academic institutions in the Philippines, namely in the fields of biorisk management with the University of the Philippines, Manila, biology teaching and education with the University of the Philippines Open University, and biological sciences with De La Salle University. His research interests are in the field of veterinary pathophysiology and animal health, laboratory animal sciences, biorisk management, and biology education. These involvements allow him to share the knowledge that he has acquired, ensuring that he is likewise updating his knowledge and skills, not only in content, but also with technology and pedagogy. So, ladies and gentlemen, Dr. Rodel Vitor. Sir?
Thank you very much, Dr. Marian, for that introduction. So as Dr. Marian mentioned, we'll be having I will be discussing two parts for this afternoon. The first one would be on program birth. And basically, we will be trying to introduce what is biorisk management in research, teaching, and health. And the second lecture will be focusing more on biosafety aspects of research and teaching. So our objectives for this afternoon will be defining what is laboratory biosafety, what is laboratory biosecurity, and laboratory management or biorisk management, and outlining their scope. We will be also introducing you to the AMP model, which is assessment, mitigation, and performance. And I will try to explain how this AMP model can be used in biorisk management and subsequently using it into laboratory biosafety and biosecurity. So first is hazards are everywhere. So as you can see, this is a typical laboratory that we have. Probably you might have seen some of your students handle microscope, hopefully not this, but probably they're not handling it properly. They're not holding it at the base. Or some students are observing the beaker and its contents too much or too close to their faces. So probably it can cause some problems, or it can cause accidents in the laboratory. Or in this case, we can say that if this is a hot plate, the student is not paying too much attention to that. And as such, these can lead to accidents and thereby probably having an incident in the laboratory. So in 2021, Bill Gates said that together with climate change, bioterrorism might be the next biggest threat after this pandemic. So he said that related to pandemic is something people don't like to talk about much, which is bioterrorism. So it is true that in the history of bioterrorism, there has only been 4 documented, and one of which was the anthrax scare in New York. And as they're saying that because biologicals or the biological assets needs to have ample time to replicate, to multiply, then it's not as they're saying, sufficient enough or good enough compared with your chemical and nuclear weapons. However, still, it is something that we should be talking about. And this is what Dean Estrella or Dean Feds will be talking about in the biosecurity part this afternoon. So, biosafety concerns for labs in the developing world. And what is these concerns? It says here, biocontainment labs across the Asia-Pacific region all too often fail to live up to the term. Why? Because basically, our laboratories are not conformed to what the West manuals are saying. And as such, there is a need to localize the biosafety manuals, the biosafety guides for us to be able to have a biosafety management or the biorisk management in our laboratories. So here are some guidance documents, and you can scan the QR codes, but I discuss them individually. So the first one here is the ISO 35001, and it is for the biorisk management for laboratories and other related organizations. This is the newest one, or the guidance documents that we have. Unfortunately, this is not free to download. You need to pay, if I remember right, around 318 francs, which is around 6,000 pesos, for you to have this one. The next one will be the CWA 15793. This is already obsolete because we now have the ISO standards for biorisk management, but both of them are dealing with laboratory biorisk management. Another one would be from the WHO, which is the Laboratory Biosafety Manual. This is where they outline the different biosafety levels. We have biosafety levels 1 to 4, and I'm sure you've heard it during the onset of the pandemic, where the Philippines is not able to diagnose fast because we have lack of BSL-2 laboratories in the Philippines capable of using the different diagnostic test kits. And another one, we have the BMBL, or the Biosafety in Microbiological and Biomedical Laboratories by the National Institutes of Health in the United States as well. This one also outlines the biosafety levels together with the ABSL, or the animal biosafety levels, for those that are handling animals aside from for the microbiologicals. And in the Philippines, we have the Manual of Standards. Unfortunately, this manual is specific currently for the clinical laboratories because this is from the OH. As such, you will notice that also I wasn't able to find a downloadable file for this one, but hopefully the WHO and the BMBL will be sufficient enough for you to be able to assess your laboratories on what are the specific needs that you should be having when you go back to face and try to upgrade your laboratories to make it safer. So some terms that we need to clarify is, first, we have a biological agent. So a biological agent would be any microbiological entity, whether it is cellular or non-cellular, that can be engineered, it can be replicated, there is genetic material, and can cause infection, allergy, or toxicity, whether it be in humans, animals, or plants. And basically, these are the bacteria, the fungi, viruses, viroids, endo and ectoparasites. So they're all biological agents. What is a biological material then? So a biological material may contain biological agents and their harmful products, such as the toxins and allergens, as such that we also take into consideration toxins and allergens when we are going to handle this in our laboratories. And as such, as faculty, as researchers, we need to understand what should be present in the laboratory. Should there be contaminants? Should there be spills? Should there be incidents, etc.? So that we can have first aid immediately in the laboratory. So a biological hazard is only a potential source of harm. So basically for this one, it is potential because even though if it is in the laboratory, let's say you are handling Mycobacterium tuberculosis, for example, in your research laboratory, it's still only a potential source of harm because as long as there is no situation that will have this hazard and become more hazardous, can lead to a laboratory acquired infection, then it's only still going to be potential. So let's discuss, or let's try to define laboratory biosafety and laboratory biosecurity. So laboratory biosafety are containment principles, they are technologies, they are practices that you can do inside your laboratory to prevent unintentional exposure. And basically what you are trying to do is to prevent yourself as the researcher, as the teacher, to become infected, and not only you, including the community that your laboratory is, your institution is located, and also the environment. So basically, everything is being protected from the biological material. Laboratory biosecurity, on the other hand, is still there principles, technologies, and practices wherein we do want to protect the biological material and also the equipment, the data that comes from researching that biological material to prevent unauthorized access, loss, theft, diversion, or release. Or in other terms, we do want to protect the pathogen, the biological materials from people who would want to intentionally use the data, the biological material for malice. So if we will be summarizing it, biosafety is accidental or unintentional release of biological agents. Biosecurity, on the other hand, would be intentional, deliberate, or unauthorized release of the same agent. And usually we try to simplify it more that for biosafety, we protect people from bad bugs. Wherein in biosecurity, we are protecting these bugs from bad people. So there are a lot of issues, and we are going to look here if whether it is biosecurity or biosafety concern. So recapping of needle can be or we can say that it's a biosafety concern. Your lab technologies, though we don't want it to be happening, is a member of a terrorist group, and it's a biosecurity concern. Laboratory personnel not provided with PPEs, it's a biosafety concern. Disgruntled staff, angry staff, together with work overload, it's a biosecurity concern. Waste from the lab are not segregated, it's a biosafety concern. Lack of staff training, this can actually be both. It can be a biosafety concern or a biosecurity concern. Why? If the laboratory staff lacks training, they will not know how to protect the pathogen, the biological material from outsider threat. And staff with a huge debt can actually be a biosecurity concern, because if your research is of high market value, let's say, they can actually be sold in the black market if in case it will happen. Hopefully not. So, but basically, biosecurity is something that it's more or less a behavioral concern, and I will not preempt Dean Fed's lecture, so she will discuss more about this biosecurity issues. But there are a lot of overlaps, and here are some of them. So in the biosafety control or biosafety concerns, we do have some engineering controls. Probably your biosafety cabinets were purchased 10 years ago and were not recalibrated. The directional airflow is no longer unidirectional. There's no more negative air pressure. Good laboratory work practices are not being checked. So hand washing technique is not being checked. We're not sure if the people are doing hand washing properly. The PPEs are purchased because it's cheap, but the quality is not being checked. So those are biosafety concerns. Biosecurity concerns, on the other hand, are all of this, and they are those that you are supposed to introduce in your facility or in your institution to ensure that people will be deterred from entering your laboratory. Like, for example, if the door towards the biological material holding room would have a biometric using fingerprints, then only those that will be enrolled in the system can access them. So you can now narrow down if there would be some loss in your pathogen or biological material supply. And there are some that overlaps, like access control, who can only access the biological material, personal management or reliability. Did you cross-check the background of the people? Did you check whether they are saying the truth, if they are really trained in molecular biology, in microbiology? How do you create an inventory of biological hazards? Especially for microbiology, it's very difficult. Let's say for a swab, you're going to say, how many ml? It's easy if it would be ml, but if it would be a plate, are you counting how many colonies are still there in the plate? So you need to have a proper inventory of biological materials. How are you disposing of the waste materials or decontaminating the laboratory after each microbiology class? Or how are you shipping? How are you sending your samples to other facilities if you are a research facility? And what are the practices and procedures that you are doing in your laboratory to ensure that you are safe and secure? So biorisk is an effect of uncertainty. So again, it's only a potential. And it is a combination of consequences and likelihood, where biological material is the source of harm. That's why we are adding the term bio, because risk would almost have the same definition. So this is a usual misconception when we are talking about biorisk. So likelihood of occurrence caused by a biological material and the consequence of harm caused by the same biological material is biorisk. Biosecurity and biosafety is not equal to biorisk. So only this first line is biorisk. So please take note of this one, because a lot of times people are confusing that when you merge biosafety and biosecurity, that is biorisk. But actually, biosafety and biosecurity would lead to your biorisk management processes. So what are the likelihood? So probably you have seen this in one way or another. So the likelihood would have a level of 1 to 5, and we have descriptors of rare, unlikely, possible, likely, and almost certain. So here, you can use the description to help you more qualify whether the risk will occur. And if indeed the risk will occur, what are the consequences to the institution, to the laboratory, to the school, to the research group? Is it insignificant? Is it minor? Is it moderate? Is it major or catastrophic? And again, you can still use the description. So for example, let's say that laboratory acquired infection was found in students in a microbiology laboratory. So are there no injuries? So let's say that the LAI is due to when students were inoculating Staphylococcus. So is it insignificant? Were there no injuries? Were there low financial loss? Or would you say it's minor because there are first aid treatment and on-site release immediately contained? So those are things that you need to check or arbitrarily qualify. And you will know if you will take more training on biorisk management in the assessment phase, that risk assessment should not be done only by one person. There should be at least a team of risk assessors so that you can find a common ground so that you are going to establish the risk assessment properly. So biorisk management then is a system or process to control safety and security risks. So basically, we do have a lot of this, starting from handling the pathogen, going to the storage and disposal of biological agents, including the toxins. So again, from purchasing to storage, to handling, to manipulating them, and again, disposing of them. So the biorisk management system is a system wherein you would now have policies, objectives, and processes. And what are the objectives of your biorisk management system? So you would want to limit the presence of risk, or you would want to have a risk that is acceptable for you to work safely in the laboratory. So again, this is a misconception with risk. Sometimes people will say, we don't want risks, but unfortunately, the way we are doing our jobs, whether we are teachers teaching microbiology or researchers in the laboratory, there would always be inherent risks in our tasks. And as such, what we do want to do is we want to have the risk be attained at a level that is where we can control it, where we can work safely. So the biorisk management then is what we want to do is provide the highest practical protection and the lowest practical exposure. So we do want to protect, again, ourselves or the pathogen and having the exposure very low. And for the practical protection, we are saying practical because sometimes, and this is actually true during the pandemic, where a lot of people were in a hurry to purchasing PPEs because they say that PPE will protect. Yes, PPE does protect you, but later in this lecture, you will see that PPE is the least effective among the different mitigation steps or mitigation processes. So again, we have the biosafety risk and the biosecurity risk. The biosafety risk is the risk of accidental exposure. Biosecurity risk is the intentional removal or the intentional misuse. And again, these risks are only potential because without a situation, the hazards or the threats for biosecurity will only be hazards and threats. Or if you have a situation without the hazard or threat, then there will also be no risk. So why do we need then biorisk management? So first and foremost, we do want to conform to biosafety and biosecurity practices. These are defined in manuals, in standard operating procedures that you can have in your facility. Next is we do want to expand the research community's awareness of biological safety in congruence with good laboratory practices. And this is going to be reached through effective communication. So for example, if in your research facility you are handling a pathogen, a biological material that is potentially dangerous because it can be causing infection through inhalation, you need to have an effective communication to the stakeholders. What are you going to say? What you need to tell them so that they can also protect themselves. And this is where prevention, or to prevent employees and their families from acquiring laboratory-associated infectious diseases. So what are the mitigation steps that you can provide so that, for example, this happened during the peak of the pandemic, where the healthcare workers are alternating in their duties, and during the time that they are on duty, they will be staying in a dormitory and they will be going home after 2 weeks. So those are things that can be mitigation steps. And again, we do want to prevent contamination of the environment and promote environmental quality. Others is we do want to have applicable complying with applicable national, international, and local guidelines. And as mentioned a while ago, the current national guidelines is only for clinical laboratories. The biosafety primer in the Philippines, if you will be looking for it, is only for GMOs. And as such, hopefully in the very, very near future, especially with the Virology Institute of the Philippines being constructed, we will now have our own national guidelines so that we can regulate the use of these biohazards. We do want to prevent the loss of microorganisms, biological materials, and specifically the data that we are having in our laboratory, together with protecting proprietary materials. And again, imagine the reputation of your institution if there will be theft of the data and this data will be misused. So imagine how much it can cause problems for your institution. And we do want to enhance emergency preparedness and response when we do have, and hopefully not in the very future, when there will be a new disease that will be emerging. So the goal of the biorisk management or the system is to identify and reduce the risks at the lowest level before they evolve into becoming near miss or harmful incidents. So for example, if we do identify that we do have inadequate biorisk management conditions, then we need to have our training immediately. If we do find that there are already now improper use of PPE, let's correct them. We're not yet at the harmful incident, but once we are already at the harmful incident level, then that is more difficult to fix. So hopefully, when we are still at the lowest level, we are trying to prevent them, we are trying to reduce the risks, and we are trying to correct the mistakes in the laboratory. So now to introduce you with the famous AMP model. So the AMP model is consisting of assessment, mitigation, and performance. And it is being said that it is like a stool. Each one is essential. And if you remove one, biorisk management, which is the seat, will not stand. So you need to have all of them for it to properly work. So 3 vital questions that we do have. At the assessment level, your questions to ask, or the question to ask yourself is, what are the risks? The next one, at the mitigation level, your next concern would be, how can I reduce or, again, possibly eliminate the risk if, again, possible? And last, at the performance, how can I confirm that I have reduced or eliminated the risk? So in here, so this is assessment, apologies. So it's a process of identifying the hazards, threats, and evaluating the risks associated with biological agents and toxins, and taking into account the adequacy of any existing controls, and deciding whether or not the risks are acceptable. So we do have a risk assessment. And for this one, the approach for biorisk management is we are always risk-based. So whatever things we are doing in the laboratory, we need to ensure that they are always risk-based. So for example, if you say to your students, you need to use this particular type of gloves, if in any case, it needs to be backed up by risk assessment. So in the risk assessment framework, we try to gather information about the risk. And then we try to evaluate. And then after evaluating the risks, we develop a risk control strategy, selecting and implementing this risk control measures, and then going back to reviewing the risks and whether they were eliminated, they were decreased. And if still not, then we still gather again information. And as such, it is a circular process. So the hazards and threats, I have been mentioning them a while ago. Hazards is the potential cause of harm for biosafety. Threat is the potential cause of the incident in biosecurity. And the threat is usually the individual, and usually it is the group of people that would want to steal from you. And again, I would be focusing on the hazards in the next lecture. Dean Feds will be focusing on the threats in her lecture later in the afternoon. So risk is only a potential. So again, this is what I was telling. If you remove the hazard and you only have the situation, risk may not occur. If you do have the hazard or the threat and there is no situation, then risk will always or will also not occur. And as such, you need to have these two at the same time to have a risk. So this is a risk matrix. And what are these numbers? We are very good at multiplying numbers. So as you can see, this is 1, 2, 3, 4, and 5. This is 1, 2, 3, 4, and 5. So you just multiply it. 1 times 5 is 5. 3 times 3 is 9. So that's how you assess. And this risk rating of low, medium, high, and extreme, as you can see with the green, the yellow, the orange, and the red, are arbitrary. this would take more than this 3 hours to discuss that the risk matrix is arbitrary. Probably if I'm a risk averse, ayaw ko ng risk at all times, I would say that these are all extreme. Pwede kong gawin yon. But for the purpose of this lecture, let's just use this basic risk matrix. So this is a very, very good scenario. You're in an open field next to a very hungry, aggressive adult tiger. So this is the adult tiger. And the tiger is unrestrained and sees you as food. So I do know I don't have much time, but let's say, what's the hazard here? So the hazard is, again, the biological material, and the hazard is the tiger. And let's describe more the tiger. So the tiger is an adult. It's hungry and aggressive, unrestrained as well. The situation, I'm in an open field. I don't have any protection, and I'm out basically in the open. The tiger is seeing me, and that's my situation. So now, what is the risk? The risk here is me being eaten or becoming the food of the tiger. So if we will be looking here, this is one problem when doing a risk assessment. They automatically look at the matrix. Don't look at the matrix immediately. Separately assess the likelihood and the consequence. So the likelihood of me being eaten by a hungry adult tiger, given the situation, is it's almost certain. I cannot run. Obviously, the tiger will be faster than me. So it's automatically 5. What would be the consequence of me being bitten by a tiger? Obviously, it's catastrophic. So it's 5 times 5, it's 25, and it's extreme. So that is how you are supposed to do a risk assessment. Do not automatically look here. Assess separately the likelihood and the consequence. So let's say here, suppose you are working with the seasonal influenza virus, conducting aerosol challenge studies on an animal host with little respiratory protection. So for this one, your hazard, I'm not sure if you can actually type your answers, but in here, the hazard will be your virus. So basically, it's the seasonal influenza virus. And what is your situation? You are conducting aerosol challenge studies on animal hosts and with little respiratory protection. Okay, so let's try to assess what's the likelihood. So again, you have very little respiratory protection. Probably you're only using just a normal mask. So let's say it's possible or likely, or probably almost certain, if you would say. But what is the consequence of being infected? So of resulting to the risk. And again, I forgot to say the risk would be laboratory acquired infection. So what is the consequence if you will be having LAIs, laboratory acquired infection, from seasonal influenza virus? So again, the term here is seasonal. It's the usual influenza virus that you get yearly. So it's not going to be catastrophic. It's not going to be even major. Probably it's minor. Or probably some would say it's even significant or insignificant. So you can only have a 5 to 10, high, medium to high. So again, you need to really characterize your hazard. It's not only an influenza virus, it's a seasonal influenza virus that you are always, or probably you are always exposed to. So now the question is, are you satisfied with the assessment that you did? If you are satisfied, then you can stop and proceed with your work. But if you're not yet satisfied, then you have your mitigation. So mitigation are actions and control measures that you put into place to reduce or eliminate the risks associated with biological agents and toxins. And what are these control measures? There are 5 control measures. So we have elimination or substitution. We have engineering controls. We have administrative controls. We have practices and procedures. And we do have personal protective equipment. In elimination or substitution, you are trying to remove the hazard or changing the agent that you are using or replacing the hazard with something less dangerous. So for example, if possible, you would try to replace glass flasks with polypropylene flasks. But for example, in the laboratory, if you are going to inoculate bacteria, you need to find a suitable replacement. So for example, if you are using E. coli, can you replace E. coli with another less pathogenic bacteria, for example? So those are substitution. But most of the time, this is very difficult to do because if we do eliminate the hazard or the bacteria that we are working with, then we are left with no work. So basically, this is sometimes very difficult to do. And as such, we do have the engineering controls. So engineering controls are facilities, your biosafety cabinets that would allow you to work safely. So you can change the workstation. You need to assess how your tables are made. Are they porous materials? Are they easy to clean? What else? The lighting, the angle of the floor, it should be a C and not an L. Going to administrative controls, are you putting a biohazard symbol in your microbiology laboratories? Have you designated a biosafety level for them? And again, this picture is a biosafety picture. However, in biosecurity, sometimes they would need you to write what are the pathogens inside your laboratory. But it's a biosecurity concern because you are telling the world that you are handling these pathogens. And as such, this might be the only thing that are essential. Biosafety level, level 2, who's the responsible investigator, the teacher? In case of emergency, contact immediately. Policies and procedures are behavioral things that you need to do. So these are processes and activities that have been shown effective to reducing the risks because changes in the behavior of a person would lead to reducing the risk. And lastly, as mentioned, PPEs are devices worn by the workers. And in here, we are trying to protect the workers against the hazards. But in the hierarchy of control, the most effective is actually the elimination and substitution, then going to PPEs. The problem with PPEs is it can easily break. Administrative control is basically behavioral. Engineering control is too expensive to implement. So here are some of the advantages and disadvantages. So advantages, again, administrative, it's authority approach. The main problem here in administrative is we are addressing human factor and not the hazard. In practices and procedures, we need to have a lot of trainings. For PPE, it doesn't eliminate the hazard at all. It's just making you uncomfortable. I do know it's very much uncomfortable wearing masks. Then it limits the ability of the user when you are handling pathogens, specifically if you are using the PAPR, wherein there is an air purifying system attached to your astronaut level protection. And then engineering, as mentioned, it's costly, it's complex, needs yearly maintenance. So in here, let's say we are revisiting scenario 1. What changes will you make to lessen your risk of being eaten? So basically, if you are a veterinarian, let's say studying this tiger, probably you will be going into a car that is tiger proof, let's say, or you will also have like restraining materials for the tiger. So let's say you can have a anesthetic to actually dose down the tiger so that you can manipulate it easily. Or in this scenario 2, you can have more respiratory protection to ensure that the risks will be decreased. And that is what we call the residual risk. Residual risk is the risk that will be left after instituting implementing mitigation processes. So probably since you have more protection, then the likelihood is rare and the consequence is still insignificant because it's seasonal, then you can now have a risk rating of low. And if your facilities say that only risks that are low will be allowed to proceed, then you can proceed with your research. So lastly, we do have the performance evaluation. And performance evaluation is the implementation of the entire biorisk management system, including the assessment or evaluation whether you are indeed lowering the risks. So in summary, this is the last slide for the first lecture. We cannot lock up all the bugs or the pathogens. We cannot control all technologies. We cannot contain all the knowledge, and we cannot isolate all the laboratorians. But we can build awareness and educate, similarly with what we are doing now. We can seek to communicate and build trust among the stakeholders. We can apply leadership in the laboratory by ensuring that there is a person responsible, and we can create an impact in the culture of the laboratory. So with that, thank you very much for listening, and I'll just stop this for a while. And I think I would be going straight to the next lecture, which is on the biosafety levels. Or are we going to have first a 5-minute break? Well, what do our moderators would say? Let's check. Okay. Okay, so there's no announcement. So I think I would go straight to our... Yes, Paul.
Oh, would you like to have a quick photo op before we proceed with your next talk so everybody can have a breather?
Okay,
and time to absorb. Thank you. Team, the tech team.
Good afternoon po. Akin.
We request everyone to kindly turn on their camera for a quick photo opportunity with our speakers.
Ma'am Marian, ang pwede lang mag-photo op is yung mga panelists. So we have 20, less than 20 panelists po.
Okay.
Pa-turn on na lang po ng camera. Salamat po. Meron pa bang magjo-join?
Okay. We request all the panelists to kindly turn on their camera so you can join us. And document this event. Thank you.
Okay, so I think we should be good now. So in the count of 3, so 1, 2, 3. One shot done. One more. 1, 2, 3. Thank you.
Okay. So yeah, I think I did over time, but I'll try to have this second lecture faster because this is we're focusing now on biosafety. So the next topic will be on teaching or teach biosafety. Sorry. So teach biosafety, we are looking at tools to engage and check biosafety in your institutions. So in here, we are going to only focus on the role of biosafety in keeping the laboratory environment safe and explaining the principles of biosafety and how it works hand in hand with biorisk management. So laboratory biosafety, as a review, is again to prevent this unintentional exposure of biological agents or materials to the environment to infect the different people that are working in your laboratory. And laboratory biosafety, again, protect the users, those outside the laboratories in the immediate vicinity, and again, the environment. So principles of biosafety is, as you can see, similar with what we have or discussed in the biorisk management system, which would be the control or the mitigation steps. So there would be specific practices and procedures that you can do inside your laboratory. There are safety equipment that you need to purchase, and you need to have a specific facility design and be constructed. And what are the increasing levels of protection depending on the pathogen or the biological material that you are handling? The first one would be the standard microbiological practices. And again, I do know that the teachers here would be very strict to have their students adhere. Again, this is the most important. And usually the first day of any microbiology class, even if you are taking microbiology for the 5th, the 6th time, not the same subject, but in subsequent subjects, they are making you be aware of the potential hazards. And our teachers, our faculty here are trained and proficient in the specific techniques. And they should be responsible to have the appropriate laboratory facilities. And again, they should have the proper or the trained personnel, and they are trained specifically for microbiology. And there are also occupational health programs that they should be aware of, so that in any case there will be accidents in the laboratory, they should be or they will be able to do a first aid for these accidents. The second one would be safety equipment. And these are the primary containment barrier. So for example, the primary containment barrier is where you are placing your biological materials. So in a test tube, or if we are using animals, we are putting them in individually ventilated cages. We also try to minimize exposure to hazards by having PPEs, biological safety cabinets, and other engineering controls or equipment that will ensure that we are delineating where people can work, where people can safely go and not go. And the next one is facility design and construction, which is usually the most expensive. It's not easy to attain. As mentioned, the biosafety levels is not that easy, specifically if you are retrofitting a laboratory. So for example, let's say that during the pandemic, so they say that for you to have the go signal to have a face-to-face class, you need to retrofit your laboratory. And sometimes retrofitting is more expensive than having a room made from scratch. So for here, you would want to protect the outside environment. So you need to have the ventilation if in any case you would be having a specific negative air pressure room, for example, in the research facility. For the teaching laboratories, it's simpler, though we still need to have the autoclaves. And sometimes if, again, for animal facilities, we need to have the cage wash facilities. And lastly, we do have the increasing level of protection. And this is where the guidelines for having biosafety levels 1, 2, 3, and 4 comes into place. So we do have the biosafety level 1. It's the foundation, meaning if you are working on biosafety level 2, you just add up on the biosafety level 1 and add on more protection. And as such, if you are going to work in a BSL4, you do have all of this. So it doesn't mean that if you are working on a BSL4, you are only going to have this protection. You will have all of the protection to ensure that you are protected, the environment is protected from the biological material. So biosafety levels are a combination of laboratory practices and procedures, the safety equipment, and the laboratory facilities. So for example, laboratory practices and procedures in biosafety level 1 would be your hand washing technique. However, as you go up, they would be requiring that the faucet should have a knee lever so that the hands will not touch the faucet itself. So those are changes with the laboratory practices. Sometimes you would find in other textbooks or in other references that they are saying that the biosafety levels are also known as containment levels because we are trying to contain a certain material in that level. So here are some of the biosafety levels. So we have the basic biosafety level 1, level 2, the containment level 3, and maximum containment level 4. So the laboratory type, usually for biosafety level 1, we have the basic teaching and research laboratories. And we do have the good microbiology techniques. Safety equipment, usually we just use an open bench and work on it. However, for biosafety level 2, sometimes even research and teaching laboratories may have a biosafety level 2 if they would have biological materials that would need a biosafety level 2 laboratory. So aside from the GMT, you need to now have protective clothing and biohazard sign outside your facility. And then we now have biosafety cabinets. In any case, we are having potential aerosols. So when we're opening the test tube or let's say the container and it's possibly aerosolized, we need to open it in a biosafety cabinet. Then for level 3, this is a special diagnostic service and still for research. Again, all of this one plus controlled access, directional airflow, and other primary devices that is needed to contain the biological material. And level 4, we do have airlock entry. There's already a shower exit and a different biosafety cabinet. And for this one, usually biosafety level 3 and 4 are those that needs to be operated 24 hours because we do have a directional airflow. We do want to maintain that, so this might be too costly. And as such, most of the time, we only have biosafety level 1 and 2 in our teaching and our research institutions. So similarly, so for biosafety level 1, the agents here are well characterized, not known to consistently cause disease in immunocompetent adult humans, and present minimal potential hazards to laboratory personnel and the environment. So these are usually those that even if you manipulate them and there would be some accidents, most of the time you will not have any infection. However, if you are working on biological materials that may cause human diseases, and if there would be some escape in the environment, can cause hazards as well, then you probably should have a biosafety level 2. So again, there's already a limited access. The sink located near exits, there are already autoclaves. So again, you are only building from one level to another and not forgetting what is in the lower level. So here's an example of a picture for biosafety level 1 and 2. I did not include 3 and 4 because basically for teaching and research, the basic researches would only be up until level 2 unless you will be working in the Virology Institute of the Philippines. So as you can see, they already have a controlled access, meaning they already put the biohazard symbol. They also have a biohazard can that is colored red for pathogenic waste. They have the sink. They have the policy on the hazards. Then the main difference here is, as you can see, is the presence of this biosafety cabinet, which is the physical containment device. Otherwise, they also have the autoclave. They have the laboratory bench that would be easy to clean. Probably this is metal and non-corrosive. So there are some properties inherent to biological materials that you need to understand and to ensure that you will be able to take into account when you are going to assess and have the mitigation processes for your facility. The first one is importance of the disease or severity of the infection, if in any case it will happen. How much infective it is. If the strain that you are using virulent, how much of this pathogen, this biological material you need to take before you can be infected? What's the mode of transmission? And what is the natural route of infection? The host range of the microorganism, are you using a pathogen of humans, or is it only for animals? Or for those veterinarians here, are you using a zoonotic pathogen? And if in any case it is in the environment, will it survive in the environment, or it can be easily cleaned? And what are the prophylactic measures available? Can you have antimicrobials to take in? Are there any vaccines to prevent you from having the disease? So we do have risk groups, and don't think that if you are working in a risk group 1, you are going to use it in a biosafety level 1, because they have the same number, RG 1, 2, 3, and 4. But for risk groups, it will tell you the pathogenicity of the organism. The mode of transmission is depending. So risk groups is also depending on whether there are preventive measures, treatment, and all of this can be seen in the pathogen safety data sheet. So if we do have the MSDS, we also have the PSDS. And the risk groups based on the NIH guidelines on research are, again, reviewing. Risk group 1 would be those not associated with disease in adult healthy humans. Risk group 2, associated with human disease. Risk group 3 would be serious or lethal human disease. And 4 would be those that would be having serious or lethal human diseases. So where can you find this risk group? You can download as an application the pathogen safety data sheets, so PSDS, or you can also look into the risk group database. And this one will be able to give you what are the different RGs for each pathogen that you are working with. And again, since we are already here, we do have LAIs. So LAIs, again, are infections and can be symptomatic or asymptomatic. And they are acquired when you are working in the laboratory. So it can be primary or secondary. Primary, you are the one working and you are infected. Secondary, you got it from the one working in the laboratory. So LAIs are continuing despite control practices and measures because even the pathogens, even the microbes are changing their modes to infect people. And again, it's getting worse and worse every year with it being emerging and re-emerging. So routes of exposure and transmission in a laboratory is important. So again, we are trying to prevent the pathogen from being ingested. We always tell the students not to bring in food, or again, we try to make them wear masks so they would also be prevented for the inhalation. The main issue now is usually the inoculation and contamination. Inoculation is, for example, if using a syringe and you accidentally pierce yourself. And contamination is where there will be unwanted substance on your skin. And if you do have a break in the skin, then you can be contaminated. So the causes of laboratory exposures currently is 20% due to equipment failure. And again, 80% is caused by human factors. Why? First, we are using the wrong or inadequate PPE, or most of the time, again, behavior, non-compliance to policies. If you are saying hand wash before and after, but they're saying, why I need to do hand washing before? So again, it's for contaminating your stock sample, for example. And sometimes no eating and drinking in the laboratories, but some students will say, I had classes from 7 a.m. to 7 p.m. and my break is only 10 o'clock to 10:30. And yes, it was me. I had a class before that I only had a break from 10 to 10:30, and I need to sneak my food in my bag and eat outside. But again, the teacher will say even eating outside may bring in the particles inside and can contaminate. So you need to find ways to actually say that how to protect your laboratory. So the top 4 accidents resulting to infections are the spills and splashes, specifically when you are inoculating, probably even with Staphylococcus, there will be an outbreak of pimples in the laboratory the next time. Then your students probably did not do proper GMP. Then the needle and syringe pricks for our medical technology colleagues here. Sharp objects, broken glass lacerations if you break glasses and you try to pick them up in the laboratory. And lastly, for those handling animals, animal scratch or bites. And this top 4 accidents, and as you can see, can easily be prevented. For spills and splashes, if you are wearing proper PPEs, then probably your mucous membranes will be safe. For needle and syringe pricks, if you are not recapping the needle and just directly throwing them in the receptacle, then you can prevent from being pricked. For sharp objects and broken glass, if you will not pick them, you would have a specific policy on how you will be cleaning up the sharp objects. Or for those handling animals, if you train properly in handling animals, then you can actually prevent this accident as well. So in 2019, the American Society for Microbiology released these guidelines for biosafety in teaching laboratories. So basically what is present here is a concise, a condensed version of the BMBL and the WHO. So basically the BMBL and WHO came out 2020, 2021. So it's newer, but basically this guidelines is saying that what are the different biosafety levels that you should have, what are the good microbiological practices and techniques that you should do, what are the safety precautions, what are the facility, what are the equipment that you should have to be able to have a safe teaching laboratory. So the last few slides will be all about scenarios. So the first one is a researcher wants to start using influenza A virus in his or her laboratories. You, being a top-notch researcher in influenza, she calls you to discuss some of the possible hazards or risks associated with introducing influenza virus into the laboratory. She tells you that they want to work with a non-pandemic, low pathogenic avian influenza virus, H9N2 or H7N7. So for those not familiar, LPAIs can still cause probably human diseases. So first one is when you are going to analyze this kind of scenario, if in any case, this is somehow similar in your teaching scenario or in your research scenario, you need to analyze what are the hazards of performing influenza research. So the hazards here are the influenza itself. And because it's LPAI, probably there would already be a treatment prophylaxis for this one. Now, does the risk change of getting infection if it is highly pathogenic avian influenza versus low? So basically, there's a much difference between this. Highly pathogenic avian influenza can cause an epidemic for the poultry industry. And this is actually a problem in the Philippines because we do have AI. So what are some mitigation measures that can be taken to prevent illness? So you need to have probably the biosafety cabinets. What's the biosafety level to safely work with LPAI? What are the PPEs that you will be ordering? What are the practices and procedures that you will have? So moving on, you need to use again the risk matrix to find the inherent risk. And again, if your inherent risk is still in the extreme or high, then probably you need to implement more mitigation measures. Meaning, aside from what is currently in place, you need to add more so that you can safely work with the pathogen inside the laboratory. Continuing with the scenario, so the researcher is now working with the low pathogenic influenza, and then you got a call and they say probably there was a possible exposure in the lab. So in here, someone in the laboratory came down with flu-like symptoms and fever, and they think it could be linked to the research in the laboratory. So being one of the top-notch researcher, you were charged with investigating and determining what happened. So They explained the situation that they were centrifuging the influenza samples and not using the safety cup and the samples break. So they were using a glass tube. So unsure what to do, the researchers opened the failed centrifuge without any protection. And basically, the virus has aerosolized. So in here, you need to now analyze what are the hazards present. So the hazards here is the aerosolized virus. The potential outcome is they would be infected. So that's the risk. So probability is the likelihood. Severity is the consequence. So you can now use the risk matrix. Now, you would now think, is the risk acceptable to you? And obviously, a lot of you will say, no, that's not acceptable. That's not, or it's highly not acceptable even in the laboratory to open a failed centrifuge without protection. So how will you mitigate this situation in the future? So you need to have specific SOPs, specific practices and procedures if there would be incidents and accidents like this. So you will now try to have a residual matrix that would be low so that you can work safely, or you can remove the samples safely from the failed centrifuge. So finally, we are reopening classes, and in here, these are the COVID-19 school health and safety protocols. This is a policy paper that was released this 2022. And this one is, if I remember right, this is from Brazil. So what they're saying is, if there are no cases, all schools open. In sporadic cases, all schools are still open. But if there is already local transmission, they don't have a blanket policy, but rather they have a risk-based policy that says that most schools may still open, but local authorities may close schools where there are clusters. And if there is already community transmission, again, it would be risk-based approach. And all this would ensure that no one is left behind, and still we are ensuring the safety of the people and the stakeholders. So in summary, so in biosafety, we are mostly looking at protecting the people, the environment, by utilizing mitigation procedures that are based on risk assessment. As mentioned a while ago, this introductory pre-conference workshop is introducing you to the topics of biosafety, biosecurity, and biorisk management. Always remember that the mitigation that you will provide should be proportional to the risk. Do not over mitigate, meaning sometimes they would say over is good. Yes, but probably you are not needing them. And as such, you are just incurring costs. And again, this will be very difficult to explain to the top management, to the deciding body, why you need to have a biosafety cabinet class 3 that is ducted when you are only using staphylococcus in your laboratory. So again, the mitigation should be proportional. And again, I have been mentioning this, do not overcompensate. So with that, thank you very much for listening. And I think Ma'am Marian has transferred the hosting to Sir O'Neill. So Sir O'Neill.
Thank you very much, Dr. Vitor, for that wonderful lectures. No, not just one, but 2 lectures on biosafety and biosecurity. Okay pa ba yung ating mga participants? Can, if you are still okay, please raise a thumbs up or a heart reaction sa ating chat box. Ayan. So at this point, we will be pausing for a few moments, for a few minutes for our health break. And we will be back after 5 minutes. So you may use your time to go to the toilet, to stretch, to eat some food, and to relax. We'll see you in a bit.
The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors.
multi-case scenario. All right. Okay, so for the biorisk management, we have heard earlier from Dr. Ron Vitor that there should be an interplay of biosafety and biosecurity practices because of there is really an overlapping roles of these two components. And with that, we also been oriented that when we talk about biosafety, we are protecting people from bad bugs. And when we talk about biosecurity in layman's term, we are protecting bad bugs from bad people. So just to focus on the biosecurity, protecting bad bugs from bad people. So I have here an illustration. Okay, so you can see now the image of these two scientists, the lady scientist on the left and another scientist on the right. Hey, so if you can observe this lady scientist or laboratory researchers on the left, she is wearing the right PPE. She is also equipped with the materials that she will be needing for the research or for the experimentations or whatever laboratory activities she will be doing. And she has also a protocol, okay, a protocol that she will be following during the course of the activities. And seems that she is really knowledgeable about the microorganisms that she is catering and thinking a lot of things on how she can really put benefits on the things that she is doing, especially in terms of research. Okay, on the right part of your screen, you have here another laboratory scientist. Okay, he is wearing a decent clothings right now, and but you can observe that he is also wearing an inappropriate PPE. Okay, and then inappropriate gloves, the goggles, or I think this is a shade, eyeglasses, and then you can see the face expression. Yeah, we're not judgmental here, but I just want to represent how biosecurity that we are talking about here is being conceptualized and is being put into a context of those people with malicious intents. So this person is probably thinking on the possibility on how he can have the opportunity to get into the details of these microorganisms, to get also access with the equipment, and of course, to get into the details of the entire laboratory activities. So within that context, we will be working on that and we will be talking all the way up to the last part of this presentation. And how are we going to mitigate these malicious intents that we are talking about? Okay. So just to address the red flag, then we have the laboratory biosafety level. So it has already been presented a while ago, and this is one of the measures that we can do with our laboratories. So it has been categorized as to different activities and practices that is being conducted in the laboratory or in a research laboratory. And of course, the necessary safety equipment that we will be needing, and it has a corresponding risk group level. But I just want to emphasize that the risk group level here and the biosafety level of the laboratories, if we are talking about the current state of our emergence and re-emergence of infectious diseases, these are just guidance. All right, so this is not a strict guide or guidelines that if it is risk group 1, then it should only be level 1, or 3, then it must be 3. All right, so we should always be thinking that it always depends upon our risk assessment. Okay, so in this laboratory biosafety levels, we are also looking into the containment facility. So it's important that we know very well the specific pathogen that we are working with. Aside from this, as I have mentioned, we have the risk group categorization. So we have to be familiar with the risk group categorization as to RG1 or risk group 1 up to RG4. And by the leveling or by the numerical value, you can see here that RG1 are those agents that are not associated with disease in healthy adult humans, while going down to RG4, these are agents that are likely to cause serious or lethal human disease. Okay, so if you will be thinking, okay, biosecurity is only applicable with RG3 and RG4. You are right that it is applicable, but the word only applicable should also be corrected because as we are working, especially in teaching laboratories, if we are working with RG1 or RG2 microorganisms, although they are not causing any diseases or associated with diseases, then they can be used. All right, they can be used for some malicious intents, like in the case of mutations, or they can be put into something new or recreate into something new out of this. So as long as you have a pathogen in your laboratories, then the biosecurity measures should also be integrated. Right. So moving forward, to elaborate and to put a stringent guarding in our biosecurity measures in our laboratories, in our teaching lab, whether it is a research or teaching laboratories, then we also have the 8 pillars of biosecurity. So just to mention these 8 pillars, we have the physical security, the personal management, the material control, transport security, the information security, emergency incident response plan, emerging technology, biotechnology, and dual-use research of concern. So in the past, we only have 5 pillars. But of course, as I have said, due to evolution in the technology and all of those things around us, especially with the coming of genetically modified organisms, then addition of the three pillars on emergency and incident response, emerging biotechnology, and dual use research of concern has been put into place. Okay, so just to have a brief overview of these 8 pillars of biosecurity, just also to have a guide for our risk assessment later. So let's go one by one for each pillar. So the first one is on physical security. So yes, we are now within the context that we have important pathogens in our laboratory. So it's very important that we have the physical barriers, and that starts from the fencing, from the gate, and it should be made up not necessarily as meron siyang specs, but it should be something that can really guard and sturdy enough to prevent illegal entry into your laboratories. And you also have here, you can also include the features of a CCTV camera. So with this one, based on my experience, if I may share with you, when we are doing some laboratory assessments in different laboratories in the country, it is just sad to note that there are still laboratories which did not have this kind of features. So they do not have physical barriers, they did not have CCTV camera. And another remarkable incident here is they have the CCTV camera, but no one is monitoring it. Okay, so that is useless. And then you can also put in an additional measure like the RFID equipment or the biometrics equipment, or in some other cases, if you have extra resources, then you can also put in the lens sensor. Okay, so eye lens sensor just before the persons may enter in your laboratory. And we're talking about that specific area in which all of those important pathogens and equipments are being kept. So there is a graded protection in terms of the physical security, and it has also different levels of security based on our risk assessment. And most of the time, higher security area is on the exclusion area. So exclusion because there's only few people or limited number of people who can enter here and who can come in and out here, but on a specified date and time. Okay, and then as you can see here, you should also be observing the concentric rings of increasing security spanning from outside to inside the facility. So you should know where is the limitation or the limited area for our visitors, for our contractors, if there will be some internship, until which part of the laboratory facility that our interns or our OJTs Okay, we'll be moving around, as well as if there's some sort of educational tour. So you should also be able to determine that limited area. And another important features that we have to ensure is that when we install physical security equipment or any mitigation measures related to it, is that it could be able to facilitate detection of any illegal access to our laboratories, and that it could also cause some delay. Yeah, there should be some alarm, for example, and response in such a way that you can readily have an immediate response, an urgent response from the authority, internal authority, and of course, those coming from the external agencies. And lastly, the access control. Yeah, it should be limited. So there are some features that whenever you enter a secured area, you will be trapped inside. So instead that you will be getting those materials inside that facility, what will happen is that before you leave the facility, and if you are intending to leave the facility, then there is an automatic closing of the doors. There's automatic closing of the gates. Okay, so there are some features of that, but of course we know the cost of that. So we can always look for a tailored fit access control system for our physical security. And then the next one is on the personnel management. So the personnel management, this one includes starting from the recruitment, the hiring of our personnel. Are they reliable? Are they trustworthy? Are they honest? You should also be considering checking their educational backgrounds, checking their experiences in other laboratories. All of those you have to explore, because you know very well that they will be involved in the manipulation of these important pathogens. And of course, in the personnel management, we cannot get away with the proper orientation of our personnel. We also have to safeguard their mental health and to safeguard their relationship with each other. So there should be harmony also, because as we know, if the employee is disgruntled or experiencing mental health issues, then that could be a threat in our organization. So teamwork, harmonious relationship is also very important that you can integrate in your mitigation measures. Okay, so aside from that, this personal management also talks about the insider and the outsider. So when we say insider, these are threats. These are personnel, these are students or employees of our institution who have the access in everything that we do, in everything that we cater in the laboratory. And if at times they will be like coerced, or for some reasons they will be rewarded for breaking the rules, then that could be a red flag for everyone. So we should always be watchful with that kind of incidents. And we also have the outsiders. But for outsiders, yeah, it's very easy to determine, because, of course, from the physical security, we can really determine if they are coming in from outside environment, that they are not part of the organization. But then again, we should always be careful on gaining access or giving access to these outsiders who might want to look at our facility. And there are also instances that those people who are doing benchmarking of our facility, of our equipment, of our protocols, you should also check on the real intentions. So those are the outsiders, no, that we are talking about. Okay, and the next pillar is on the material control. For the material control, Dr. Ron is right. You have to count how many vials of pathogens are kept in your laboratory. How many of those are RG1 up to RG4? You have to label that. How many are fresh in preparation? How many are frozen in preparation? And where are they being kept? What are the specific equipment that you are using for to safeguard them? And is that equipment having the right let's say access or the right lock or alarms? So we should also be integrating that in our safekeeping protocols. And of course, the list, the inventory list of all of these materials This biological material should also be kept in a secured area. If this is like a soft copy, no, if you're using a software or database, then you have to make sure that there's also limited person who has access to it. Hey, so with this material control, you should be able to determine if something is missing. Because first and foremost, we do not want to report to our administrators that one vial of SARS-CoV-2 is missing. Okay, so we know very well that some scientists have the not so good intention, but I do not say all, but there are really some cases, as presented earlier, that some scientists are being motivated to go beyond what is expected of them. So they might be using this for personal gain or monetary reasons or something else. Okay, and then another pillar is on the transport security. So in here, we're talking about what we call COC, or the chain of custody. In our laboratory, we usually acquire microorganisms. That is very common among our students, especially those doing research activities. Starting from identifying the suppliers up to the part that the students will bring in the pathogens or the microorganisms in the respective laboratories, you should be involved, or the academicians, the researchers, the faculty should be involved, or somebody must be in charge. Okay, so you have to check the accreditation of the suppliers. You have to check if they have the permit to really release those kind of microorganisms. And then if they have the transport license to do that, it could be traveled by land, by air, or even by the sea. So you have to check if they have all those legal documents to do that. Otherwise, if you have the biosecurity measures in your own laboratory, then you're violating already your own biosecurity protocols, because you are accepting pathogens or microorganisms coming from unidentified source. Okay, so you are tolerating those, and we do not want that to happen. And along the processes, we should be involved, that we should be informed what is the exact time and date that this kind of pathogen will be arriving in our institution. That's very important, because we do not know that someone else will be receiving it. And also, we have to make sure that there is a designated person who will be receiving that specific delivery of pathogens. So you should make sure that one of your employees should be in charge of it. And then, of course, you have to make sure just before the microorganisms or the pathogens arrive in your laboratory, then you have the proper storage equipment for that, and of course, the protocols for that. Then, as soon as the package reach your laboratory, and please tell your students not to open the packaging anywhere else. There should be a designated area in your institution where they can only open those packaging. Because the first thing that you have to do to is to check on the labels. Yeah, para tayo nag-grocery. You have to check on the labels if what is indicated is that, is it infectious? Is it fatal? Because we do not know what is really inside the canister. We do not know what will be surprising us later on. So we have to check the labels. We have to check the identity of the pathogens inside, and of course, everything about the shipper. So in here, we also have to check on the packaging. Usually when we are transporting pathogens, then we have the triple packaging. So the primary, the secondary, and the tertiary. So it has 3 leveling. And in between this packaging, we can also put in some absorbent, okay, and cushioning materials, just to facilitate if there will be some events of spillage or leakage, then we can really avoid unintentional release of these microorganisms in the environment. Okay. The next pillar is on the information security. So on the information security, whether you are using the conventional or the traditional way of recording your pathogens, recording the list of your researches, recording the name of your employees with their specific designation and all the other things, the protocols that you are doing in the laboratory, whether is it the high technology using software or database, then you have to make sure that they are being kept in a very well secured area. And if it is confined in a software, then you have also to make sure that you have the necessary features. Okay, so medyo technology aspects ito of your information security. Aside from the common thing that we use password, specific password for each staff or for each employee, we also have the installation of the different softwares that really we want to make sure na they will not be hacked, that they will not be entered by anyone who's not authorized to gain access with our information. Okay, so that is on the information security. So having also a very good firewall could help. And of course, in the information security, we have also to allot or to allocate a specific area of the server. So this information area, okay, or recording area of everything that we are doing or everything that we are recording in the laboratory must be situated in such a place that not everybody, especially the yung mga tenants natin, the OJTs, no, yung ating mga contractual employees, that they really cannot go into that areas or specific rooms. So the server that we will be using for this should also be checked on the aspects of that there will be no breach on any part of this information, and there will also be observing of the data privacy aspect. Okay, and the additional pillar is on the emergency and incident response plan. So yeah, it's very common. We have incident or emergency response plan in our laboratories, but the next question is, are we really following what is written in our response plan? Do we have the training? Do we have the regular drills of this emergency incident response plan of all the procedures that's written? And how do we do when it comes to spillage? And how do we do if we have new employees? How do we do if we have retiring employees? So all of those things should be incorporated in our emergency and incident response plan. But in here, we can also integrate the aspect on natural disasters, because as we are thinking of deliberate attack or deliberate or intentional release of these pathogens, we should also be thinking that there should be some sort of indirect, okay, attacks. Okay, so these indirect attacks could be a natural disasters, like there will be some flooding, or there will be some earthquake, yeah, and then bomb threat, and then everybody will be vacating the laboratory or the research lab, and then somebody else will be going inside without your permission and will just be stealing one of the vials of whatever organism is kept there, and then we'll be telling everybody that that is because of the natural disaster. So you should also be keen on that when you are preparing the incident response plan. So being a pillar, so this is important within the aspect of our biosecurity measures. Okay, so another one is the emerging biotechnology. So when we do the laboratory experimentations in our educational labs, then we have to orient our students. Yeah, we are knowledgeable already, and we know that the students are very competitive nowadays when it comes to experiments or researchers involving biotechnology. But the only or the best thing that we can integrate here is to give them the right attitude, to give them the right or the proper orientation on how they can really contribute with this emerging biotechnology, because we know that in this evolving environment, we can do a lot of things with just one DNA or RNA. We can do a lot of things. So it's very important that we impart to our students, to our researchers, the most important thing, and that is keeping the integrity and keeping their expertise along with the right attitude when it comes to handling pathogens in relation to emerging biotechnologies. Okay, and I think this is the last one. For the dual use research of concern, this is more on the malicious use of these microorganisms of the protocols that we have in the laboratory. So we have to be observant of these incidents in the laboratory, because some of our researchers might be joining our team because they are spying or because they are being coerced. And there are also some students who are forced to do that, to be involved in that kind of group or organization. So we have to be very observant of the behavior of the movement, especially when it's break time. You have to make sure that everybody is out of the laboratory. So in the dual use research of concern, most of the incidents here are relevant to using those microorganisms into another purpose, into a malicious purpose. Okay. So to be able, I'm just checking the time, so it's 3:10. So to be able to come up with a better mitigation measures, so we have to follow a systematic approach of doing it. So it's important, it is necessary, and it is a must that we have a biosecurity plan. And to be able to come up with a biosecurity plan, then you must have a robust risk assessment, biosecurity risk assessment. So this is somehow the same with biosafety. Aside from the hazards or the threats that we are talking about now, are those people who has malicious intent. So these are the adversaries. So the adversary's motive, the adversary's capability, and of course the historical frequency. If there is some history in the past that these people are spying or stealing or getting all of those important materials in your laboratory without any permission. So these things will be checked based on the likelihood of these events. So the risk will also be checked on these adversaries. And then alongside with that is to check on the consequence. But this time, the consequence is more focused on the impact and the vulnerability. Vulnerability of the existing mitigation measures and the impact not only to the laboratory personnel, but more importantly to the community. So let's move on with the risk assessment. I have here 3 guidelines that you can use as your guidance when you perform a biosecurity risk assessment. So we have the ISO 31000, the Canadian Biosafety Handbook, and conducting a biosecurity risk assessment guideline. But for the purpose of the presentation, I will be using the conducting a biosecurity risk assessment guideline from the Canadian Biosecurity Guide. So in here, it's almost the same, but in this reference, it is more detailed, as you can see in the highlighted part. So you take each item individually. Okay, so let's move on with this one by one. Okay, so let us try to assume that we are already on the mindset of conducting a biosecurity risk assessment in our laboratories. So the first thing, of course, in all other things as well, so we need to have a very good preparation. And in preparing, you have to gather all the documents that you have, your current documents, your previous documents, if you have a record of your risk assessment in the past. And then you also have to check on the organization's mandate. Also have to check on the list of your employees. You have also to check on the regulatory guidelines. So all of those relevant information or documentations that you have in the laboratory must be gathered together for reference. And then you need to have a brainstorming of the threat environment. So you just have to look into the possibility, what could be the threat in this kind of laboratory, or in the laboratory I am in, considering that we are working with this kind of pathogen, for specifically the RG3 or the RG4 pathogen. And who could be the possible adversaries? So you will be listing all of those, and afterward, you will be coming up with a narrative summary, okay, of this threat environment. And you should also be checking on the assessment team who will be included in the biosecurity risk assessment. So in here, you have to make sure that you're choosing the right people. Okay, so in the assessment team, you have to check who are the people involved. Specifically, these are the laboratory personnel who has knowledge on the daily basis activity of the laboratories. You should also include, of course, the BSO, or the biosecurity or the biosafety officer, being the lead person. And then the senior management, representative from the senior management, because they will be the one who will be deciding on the risk tolerance later on. Then, and of course, of the budgetary requirements, they will be involved on that if ever there will be some mitigation measures that you will be identifying. And you can also include in your assessment team your IT personnel, security personnel, the finance personnel, and all the other people that you think has something to contribute and you think is necessary for the operation of your laboratory. Okay, and then the risk assessment schedule, you must have a very good project plan, including all the timeline and flexibility of the schedule. And from the review, from the assessment until the part that you are performing or drafting the final report, up to the part that you are transferring the reports, or you are communicating or disseminating the reports to your superior. So that should also be included in your risk assessment schedule. Okay. And now you can proceed with the asset inventory. So yeah, the assets here are the biologicals, the materials, the equipment that are all related in that pathogen. Okay, so in asset identification, you can check on the specific identity of your pathogen. So again, you can go back to the RG groupings, okay, the risk group groupings, RG 1, 2, 3, and 4. And then you try to check which are the tangibles, the intangibles, and the people, because all of these are assets. So tangibles are your biologicals, materials, and equipment, while your intangibles are your scientific knowledge, your logistics in terms of the plans. And then you could also include in your intangibles all of those protocols. So for the people, these are the people who has really an understanding of the mandate of the organization. Okay, so you should also include them. So you just list all of those assets that you have in your laboratory, and then try to come up with prioritization. And when you do prioritization, you have to refer to which item could be used for malicious intents, or which item has the potential to be of dual use. So that is how you do the asset prioritization. So I have here an example of the asset prioritization. So I have mentioned, for example, the tangible. These are the biological, the virus, and then once you have identified them that they can be used for malicious intent, then you can tag them as SSPA, or security sensitive biological agents, and then you just put in the risk group. And by the way, this chart, you can use this chart as your guide coming from the Canadian Biosecurity Guide. So you can use this to facilitate, no? So we have a template already. So I'm sharing this template to you so you can use this in your own laboratory. So you just have to fill up the necessary details here to help you or to guide you in the identification of the assets. Okay, and then you have to come up with priority value, and you can use this component values. So this is mix of qualitative and quantitative numerical values. So based on the assessment, so this is just an example. So take, for instance, you got 3 or medium based on the existing situation in the laboratory. Okay, and then afterward, you will try to come up with the checking on the likelihood of the risk. So yeah, the threat here are those adversaries. Always remember that. So the likelihood is the deliberate loss, deliberate release of these important pathogens or equipment. So you have to identify biosecurity events, deliberate biosecurity events, determine the motive. Do they have enough motive? Some people are very expressive when it comes to motive. So you have to be keen on that. And some people will be silent for this. So you just have to observe the behaviors. For the means and capability, this is very common among our insiders, among our employees. So they have the means and the capability. And the historical frequency, these are the history that is taking place while they are involved in other laboratories as well. So you could also look on those aspects when you are checking on the likelihood. Okay, I have 10 minutes. Okay, so for the adversary assessment, you can use this chart again. So this is open to all. So for the motivation, you can have, or you can put in a scale here. If the motivation is very motivated, somewhat motivated, or low motivation, or very low motivation, then you just have to put in the corresponding value. So that will depend upon, of course, on your assessment. So that is why it's important that you involve all the people in that laboratory. And then for the means and capability, do they have a very sophisticated means, sophisticated, somewhat sophisticated, limited, or very limited? And then you just have to put in the numerical value. For the biosecurity event frequency assessment, you have to check, is this risk could happen, or the loss of this important pathogen could happen in less than 1 month, or is it possible that it will happen in less than 1 month to 1 year, or 1 year or to less than 5 years? So you just have to choose here, which do you think? And then for the proximity, you just have to think on, will it be happening in other laboratories just near my laboratories, but outside of your own laboratory? So it is very frequent. So is it happening also in other laboratories just near your own laboratory? And then you just have, again, to use this component value for the proximity value. And for the distance, you will be thinking if this theft or loss of this pathogen could happen in a national level, in a regional level, or in a nearby community. So that is about the distance. So again, it is very frequent. So sometimes you also have to ask your colleagues or your friends from other laboratories, from other regions. So that could also be a basis for the distance assessment. So again, you just have to use the component value. Okay. So this is very tedious if this is your first time to see all of these things. But then again, these are justifiable in a sense that if you want to really pursue mitigation measures with your superiors or with the senior management, then it is you can use this as your reference to really justify, okay, your intention to put up or to install those mitigation measures. Okay, for the likelihood assessment, then you just have to put in the adversary and the frequency, and you just have to get the average of the 4 element values. So example, you have here 5, 5, 5. So 4, 5, you get the average. So that's very high, or 5. All right. Okay, so this is just an example. So first is we have the disgruntled employee uses assets to infect other personnel. So in here, disgruntled yung ating employee. For whatever reasons, that employee is so stressed and disgruntled at all times. So yeah, the category of the biosecurity event will be deliberate, intentional, and the group is, there could be some misuse. And yes, that employee is an insider personnel, and that could also be a student. And then the targeted assets, most likely, if that employee wants to infect other personnel, that would be an RG3 organism. Okay, and the target also are the personnel. And then for the likelihood assessment, the one that I presented to you earlier, you just have to put it in here. Yeah, and then you just look at the average of these 4 elements. So that will be your likelihood value. Okay, then you should also use again the component value that I presented to you a while ago. Okay, so this one, the 1 is very low, 2 is low, 3 is medium, 4 is high, and 5 is very high. So these component values will be used in all parts of your assessment. Okay, so paulit-ulit lang natin siyang gagamitin as a reference. Okay, and then the next one is looking on the consequence. So when we check on the consequence assessment, we want to see the impact to public health, animal health, and the organization. We want to check the vulnerabilities and the effectiveness of our mitigation measures. And in the example that I have here, you can check the impact by looking at the negligibility, limitations of there will be several cases or minimal deaths. So this will be your guide, this one. And it has also the corresponding component value. Okay, so in this example, with our disgruntled employee, so it has been identified that the mitigation measure is on security screening, there is insider threat training and access control system concerns. But after assessing on the impact value and the vulnerability value, because this might not be really be implemented accordingly, so you should put in here if it is effective, very effective, somewhat ineffective, very ineffective, or no mitigations at all. And then you just put the corresponding score. Okay, for the risk level or risk analysis, you just have to come up with a summary of everything, likelihood times the consequence. So just multiply it, the impact and the vulnerability, and then you will come up with this risk level results. So for example, you have the medium 18, high 36. Just multiply the numbers here. And the one that is the highest number that you will obtain will be your final risk level. So in here, the highest is 36. So your final risk level is high. For the risk tolerance, it always depends upon the senior management on the willingness of the organization to accept or reject a given level of residual risk. So this is the next step that you have to really talk about among yourselves, especially with the senior management. And you can use also a risk tolerance threshold that, for example, if it falls between very low to medium, then the risk level can be acceptable. But if it falls between high to very high, then you will not accept the risk. Okay, so this is just again an example of our disgruntled employee. So because this is high and 36, and as I have mentioned, we use that tolerance chart, risk tolerance chart, and it indicated there that if it is high, then it will not be accepted. Okay, and then afterward, we will try to employ the possible mitigation measures. So possible mitigation measures will not only end on identifying, but also it will be helpful if you will come up with a cost-benefit analysis. So of course, we always want to take into considerations all the resources that we have. So we will have to perform cost-benefit analysis. So that's why we have also our procurement officers, our finance officers to help us on this aspect. And we can also use the different international standards on coming up with better mitigations. We have the ISO 35001, which is the biorisk management for laboratories and other related organizations. Okay, and then finally, you have to report your findings. It should not be kept inside the cabinet, or it should not be kept just for accreditation purposes. So you have to review and do continual improvement by preparing a biosecurity risk assessment report. And in that report, it should include all of these things. So it should be comprehensive. It should be written in such a manner that you will be justifying each part of the biosecurity risk assessment. Okay, so the next big question is, are we ready to take small steps onto bigger leaps to implement code breach? So yeah, that is our takeaway questions that you really have to ask among yourselves when you get home, or if you are in your laboratories, if you're coming back to your laboratories. Okay, so going back to our objective, so I believe we're able to explain the basic concept of biosecurity. We're able to discuss the 8 pillars of biosecurity. We're able to elaborate the biosecurity risk assessment and do some analysis, a sample analysis of biosecurity case scenario. So I think that would be all for my presentation for this afternoon. So I hope you gain something out of this presentation to complete the biorisk management for this pre-con. So thank you very much everyone for listening and for your attention. Always be safe, always be biosafe, and be biosecure. So thank you once again.
Thank you very much, Dr. Estrella, Dean Feds, as we call her. Um, Marami pong salamat. Thank you very much to both of our speakers this afternoon for the very complete, from birth to breach, discussions and elucidations and asseverations regarding biosafety and biosecurity. Jumping off from what Dean Feds had just said, we should strive further to become biosafe and biosecure. At this point, we'd like to invite our audience, our participants this afternoon, to type in your questions in the chat box, both here in the Zoom chat box and in our website, in our conference platforms. Our guests this afternoon, Dr. Ron and Doc Feds, are both eager to answer your questions regarding biosafety and biosecurity, from birth to teach to breach. Ayan, so simulan na natin. So again, before we start, can we have a show of virtual thumbs up para makita natin if you're still alive, awake, and enthusiastic? Ayan, thumbs up. Thumbs up tayo. Okay. May nagta-thumbs up pa. Naku, wala nagta-thumbs up. Ayan, okay. All right. So okay, so let's start our first ayan, so may mahabol pa na nagta-thumbs up. All right, ayan. Okay, so please feel free to abuse the reaction button so that we know, that's actually the difficulty of the online setup, and hindi tayo mag face to face, but nevertheless we're still here and we are awaiting some of the questions. We'll start off with a question. Ayan ha. Titingnan ko po yung kodigo ko eh. Sige. Ayan. I think this is a question for Dr. Ron. No? So the question goes like this. I'm sorry hindi identified yung nagtanong, pero the question is, should we expect significant changes in biorisk and biosafety protocols in schools, as a response to, possible emerging or re-emerging diseases?
Well, personally, I would say that in the Philippines, like, the concept is relatively new. Dean Feds can attest to this, specifically in the medical technology field. And as such, that as we become more aware of the concept, we are more adamant to implement it. So, the moment that we know, okay, these are the biosafety levels, what's our biosafety level in the school? Afterwards, after knowing the biosafety level, okay, according to this biosafety level, what are the equipment? What are the facilities? What are the only procedures? What are the only laboratory experiments that we are supposed to do? So I think with this kind of knowledge that we are getting more every day, and biorisk management is actually evolving every day, we are responding to it. So it's very good that the universities now, and hopefully those specialized senior high schools having specialized STEM tracks, would also try to implement the biosafety, biosecurity, biorisk management system in their own institutions. But yes, for personally, I would say that eventually we will really see more and more people becoming more aware about this one.
So, well, uh, because of this pandemic, I would say no, that the awareness or the level of awareness on biosecurity has has jump started as of we speak. Dean Feds, I'd like to pose this question to you know, kasi although all of us here are from the academy, so in cases that the schools, for example, are not well equipped, for microbiological studies, how can uh, proper biosafety or biosecurity protocols be implemented even without the equipment needed? I think both of you can answer. So, kahit sino.
Okay. So yeah, first and foremost, as I have mentioned earlier, we have to get to know your biological materials. So that is your basis for everything. So if you think you cannot support the requirements of handling those kind of biological materials, then might as well the mitigation measure on elimination can be can be implemented. Yeah. That's my personal opinion, because if we will insist on it, then we're just putting more risk with our students, with our faculty members, and the outside environment.
Especially in designing perhaps educational activities or laboratory activities and even research. Kailangan alam mo talaga kasi you don't want to expose yourselves.
Yes. And actually, Doc, Ron, you're also Ron.
Yeah, pares kami.
Actually, we have also in our institutions, in our academic institutions, we have our safety officers. So
Yes.
just before we have the actual facility or laboratory, so somebody is already guarding on it. So I think they should be doing their task on checking the specific facility design.
And since you brought to the table the safety officer, yung mga, I know in the university that I work at, merong parang occupational health and safety committee. I'm not so sure if that is the same with the biosafety and biosecurity committee. I don't know the setup. But in terms of... Yes, Sir Ron.
Sorry po. Kasi ano lang po, since babalik po tayo dun sa bagong concept po kasi siya, currently it's under the Occupational Health and Safety Administration or the office. But hopefully, like for example, in other schools, they have the environmental, occupational, and biological safety offices. So hiwa-hiwalay po. So yun yung nagkakaroon. But currently, it's good na, yung sabi nga ni Dean Feds, may safety officer na even though ang originally niya lang was yung occupational safety, binibigay na rin yung biological safety with them. So yun yung currently set up, but hopefully in the near future, baka magkaroon ng increased need for biosafety officers for different institutions. So that would be one aspect where our microbiologists, our registered microbiologists, our specialist microbiologists from PSM can actually be working with.
Yes.
Ayun po.
I may add to that, Sir Ronilo. Ang pangalan mo si Bobo. Okay. If I may add to that, actually the Department of Health has already acknowledged the necessity of biosafety officers in their new administrative order. But that is for clinical laboratories. So that could be a good jumpstart for all of us to look into the possibility of integrating that kind of requirement among our laboratory institutions in the schools. Because one of our objectives is to expose our students with what is actually happening in the workplace. So if they will be going, like in our instance, to the medical technology program, if they will be immersing in such kind of environment from the school laboratories that they are already equipped with the awareness and all the information about being a biosafety officer, then it will not be really difficult for them when they reach the time that they will be really working as medical technologists, with all the other professions or discipline.
Even microbiologists nga, katulad din ng sabi ko. That's really nice. So I was going to ask, and since you mentioned already that DOH had already some sort of adapted this kind of paradigm where they recognize by safety as a component of the operations of different hospitals. Would there be a need to, let's say, for HEIs, even for HEIs who handle or who offer courses in microbiology, or even conduct research on pathogens, to really institutionalize by way of, let's say, a law, or by way of a circular from, let's say, CHED or regulatory offices. I'm not so sure about how it is done, but do you know? Would you know if that is perhaps necessary? If that is something that needs to be considered.
Basically for this one, Sir O'Neill, with the law, actually that's one thing that we are actually missing right now, yung talagang mai-institutionalize, or there would be a national law saying that biosafety is an integral component of the different HEIs. But currently, like for example, with the medical technology, again, I'm going to use Dean Feds. They have institutionalized the biosafety component in their curriculum. So sa curriculum pa lang po siya nagsa-start. But then eventually, I think what will happen is once we have this, for example, a law that says that for those HEIs that has a microbiology course or anything that would be having biologicals, they should have a biosafety officer. So I think without the law, dun po tayo kasi nagkakaroon ng parang deadlock na hanggat walang law, hindi tayo kumikilos because this is the bare minimum. So when we do have the next step, I think this is being pushed in the House of Representatives in the senators level, that to really have the program, the professionalization of the biosafety and the biorisk management. So yun yung currently set up talaga sa Philippines. We lack the law to mandate the biosafety implementation in HEIs. Dean Feds.
Yes. And actually, with the coming of COVID-19 pandemic, as we go back to our institutions for our limited face-to-face, if you can observe the guidelines that was released by CHED, almost all the mitigation measures within the context of biosafety and biosecurities are already integrated there. So it's also a good start for all of us. However, we are still hoping that it will be put into a national level legislative aspect so that everybody will really be compelled to follow. So I think that will be the next move of whoever who wants to initiate on that. We will be supporting on that because we can do better if we are very good in number. So for all the advocates for biosafety and biosecurity, we are calling out all of you to please join us in this journey of putting in all of these things into legislations.
Kumbaga, let's join hands to win. Parang isip tayo ng acronym doon. Kasi may birth, teach, and breach na eh. To reach. To reach. So there's a question here for Dr. Estrella po. This is from Danica Volpane. Can you provide best practices or tips on how we can assess the frequency of incidences of biosecurity breaches near our laboratory, because usually other labs would not divulge this information due to confidentiality.
Yes, that is right. So I also observed that some laboratories will really not be releasing those kind of information. But if you are in the process of doing your biosecurity risk assessment, I have mentioned that in my presentation, that you can also seek the help of the agencies, the outside agencies, like, for example, the defense, the PNP, because they have all the records and they have all the logs of the crimes, of the events that's happening in the surrounding community. So I think that is open as long as open to public, as long as, because they have the statistics for that, as long as you will not divulge the name of the persons involved. So I think that's one way. And another way is you have to be observant of your colleagues, of the people who are doing researches, of the people who are working in the laboratory. As I have said, background checking of the people who's going inside your laboratory. So that's another concrete example that you can do. So yeah, we're very careful with the information because we already have the Data Privacy Act. So I think we can get all of those statistics from the regulatory agencies or from the police agencies that we have in the community.
Yeah, thank you very much. I think Dean Feds already answered it very well. O di ba, Dean yung sumasagot sa mga tanong. Normally, Dean siya nagtatanong. But yan, I think Danica is very satisfied with the answer. There's another question that was earlier posed here. I'm just going to read it. It concerns about databases. Sabi niya, DNA and protein sequences are available in many online databases as we know it. While these sequences are used primarily for research purposes, their potential for misuse is possible with recombinant DNA technology and other technologies which are readily and commercially available. Ngayon, his question is, how do these databases prevent the possible misuse of these sequences, or is it even possible for these materials with potential dual use? Now, in terms of biosecurity advice, how do you address databases like that, or the use of databases?
Okay, so to continue my thesis defense. Okay, so for that question, yeah, I know, I'm aware of that. There's a lot of protocols around. We can just ask Mr. Google about that. But the next big question is, it will only reside in the minds of those people who wants to go into the misuse of those protocols, because they will be needing a laboratory, they will be needing equipment. They cannot really proceed on that without really diligent planning on how to go about those processes. And I hope that those information, those protocols will be used for academic purposes. Diba? So for the learning, the benefit of our students, of our researchers, future researchers, as a guide for them on how to do certain things. So to answer again the question, yeah, you can to that point, but the next part of it is a question mark. Okay, so that is my personal opinion. How about you, Dr. Ron? What can you say about it?
Well, I was to actually post in on the chat, but apparently I cannot send to everyone. But probably you can check cyber biosecurity challenges of pathogen genome databases. And basically, this was published in 2019. They actually know that the main problem is the access. Because as we do know, like for example, NCBI, where most databases are present, the access is free for all. So as Dean mentioned, usually where we have a stopgap measure is when the research is legitimate, meaning it will undergo an ethical board review. And this is where the ethical board will actually determine whether the research is a dual use research of concern.
Yeah.
So usually there are two different terms. We have the dual use research and the dual use research of concern. Basically, they would lead to the same thing, but the dual use research of concerns are only those that are, or those pathogens, those biological materials that have been tagged by your government, for example. So for example, in the US, they have 15. So for example, including here would be the 1912 influenza, the smallpox, because it has been eradicated. And for animals, this would include rinderpest. So those are the pathogens that once you will be doing a research for that, the first would automatically ring a bell. Why are you going to do a research, for example, on smallpox?
Yeah.
So usually it's going to reside, as Dean Feds mentioned, to the minds of those people who would want to really do malicious intent for this. And in the training that I recently attended, they said that currently biological threats are lower compared with chemical and nuclear threats because it's very difficult to manipulate the pathogen to become highly pathogenic. For example, if you have like, how can you make it aerosolized? But again, there is the possibility, again, laging may potential. So in legitimate institutions research, dito papasok yung ating review board. And dito na papasok ulit na magkakaroon ng Institutional Biosafety and Biosecurity Committee aside from the Research Ethics Committee. So dumadami yung hindi natin sinasabi na gusto natin na magkaroon ng maraming stop, but rather hindi siya red tape, but rather we do want to legitimize the research. We do want to ensure that the research that we are doing are safe. They are going to provide us with good results, and eventually we can publish them. Kasi baka yung maging research natin ma-flag down. So hindi lang ma-publish. So sayang lang din. So yun yung biosecurity aspect for that.
Thank you very much, Sir Ron. And that brings us really to the original juxtaposition on that there is a need to institutionalize and to not even popularize, but operationalize even biosafety and biosecurity, not just in schools that are offering medicine or medtech courses, but also in every other institution that has the potential for these things. And surely one way is by the efforts that the NTCBB and the PSN have been doing, and we are very appreciative of your time and efforts in joining us this afternoon even. But before we let you go, parang kanta yun, before we let you go, what do you think? Parang takeaway messages. How do you see the fate or the future of biosafety and biosecurity in the Philippines now? Now that we are, starting to live with the virus, and perhaps in the future, in a few months' time, we'll probably declare a post-pandemic situation in our country. Doc Ron, and then Doc Fitz.
Okay. So for the biosafety aspect, so basically similar with biosecurity, I would want to introduce you to ADORA. So ADORA is an abbreviation for All Depends on Risk Assessment. So all of your steps, whether you choose to have a biosafety cabinet, whether you choose to have a certain kind of PPE, a certain kind of specific protocols, you need to ensure that they are based on risk assessment. So lagi tayong babalik sa risk-based nating mga procedures to ensure that we are giving only the mitigation that is equated to the risk already present. Kasi baka meron tayong mga binibigay and na hindi naman pala dapat, so sobrang napapamahalang yung costing natin. So that would be for me on the biosafety. So Dean Feds.
Okay, so just a little takeaway for everyone. Biosafety, the concept of biosafety and biosecurity is now really relevant. We have been introduced to it little by little, and we cannot get away from it. We are living with a complex society. We are living with evolving technology and evolving environment where everything is there. So in just a snap, we do not know what will happen next, if there will be emergence or re-emergence of anything. So with that, I think it's the best time that we all join hands. Sabi nga natin kanina, we all join hands in raising awareness about the principles of biosafety and biosecurity. And also, we should integrate among our students how to be a responsible researcher, how to be a credible researcher in which you will really be upkeeping integrity and excellence in everything you do, might be a research or it might be in your workplace. So that would be all. Thank you very much, Doc.
Thank you very much, Doc Ron and Doc Feds, for being with us this afternoon. A warm round of the warmest round of virtual applause once again for our guest this afternoon. And at this point, we would like to award our guests certificates, no? Wait lang. Hold on. Ayan, okay. So it reads, Philippine Society for Microbiology Incorporated, in cooperation with Trinity University of Asia, awards this certificate of appreciation to Dr. Lodell Johnson S. Vitor for having served as speaker during the pre-convention activity of the 50th Annual Convention and Scientific Meeting of the Philippine Society for Microbiology Incorporated, in cooperation with the National Training Center for Biosafety and Biosecurity. With the title Biosafety and Biosecurity in Research and Education, Pandemic and Post-Pandemic Scenarios, given this 20th day of July 2022, signed Howell T. Hall, Pre-Convention Chair, and Donna May Papa, PSM President. We'd like to invite on screen our dear President and Vice President, Dr. Donna Papa and Dr. Howell Hall. Ayan, yan si Sir. Good afternoon po.
Good afternoon.
Ayan. And we will be awarding our next certificate to Certificate of Appreciation to Dr. Fedeline P. Estrella for having served as speaker during the pre-convention activity of the 51st Annual Convention and Scientific Meeting of the Philippine Society for Microbiology Incorporated, in cooperation with the National Training Center for Biosafety and Biosecurity, with the theme Biosafety and Biosecurity in Research and Education, Pandemic and Post-Pandemic Scenarios, given this 20th day of July 2022, signed Howell T. Ho, Pre-Convention Chair, and Donna May D. Papa, PSM President. Congratulations po, and thank you very much to Dr. Vitor and Dr. Estrella. Palakpakan po natin sila. Thank you very much. And at this point, I would like to invite on the virtual floor our dear Pre-Convention Chair and Vice President of PSM, and also the Vice President of Trinity University of Asia, Dr. Howell T. Ho.
Thank you, Dr. Flores. So I thank very much our esteemed speakers and organizers. This was a very informative and comprehensive webinar. I'm especially thankful for Dr. Vitor for sharing to us his thoughts about biorisk, and to Dean Estrella for her insights into biosecurity. I'm grateful to our participants who took the time to attend our pre-conference session this afternoon, and hoping you learned a lot and appreciated the sharings of our speakers. And please help us to push to institutionalize biosafety and biosecurity in your institutions. I know our speakers and all the other CBOs, they are more than willing to help you in this aspect. And enjoy the rest of the day, and encouraging all to attend the other pre-convention activities and to participate as well in the exhibits and poster sessions, which are also already available in the website of the PSM. And the plenary sessions that are slated for tomorrow in our two-day conference. I'm really very excited for our speakers. They have prepared a very nice talk for us, and have a great and hearty dinner tonight. So thank you very much for sharing your time.
Thank you very much, Dr. Howell Hall, for that
The
closing remark.
51st Annual
And
Convention
we've
and
come
Scientific
to
Meeting
the
is
end
made possible
of
by
our
our
pre-convention
majors.
workshop session this afternoon. We'd like to thank again all the committee members for their hard work, to all our guests, and to you, our participants, for making this pre-convention on biosecurity and biosafety session a resounding success. We also like to thank our dear President, President Donna May Papa, and our Vice President, Dr. Howard Hall, for being with us this afternoon, as well as all the PSM members who are here with us today. And as Dr. Hall just said, we would like to see you tomorrow as we officially open the PSM 51st Annual Convention virtually. virtually. So see you in our plenary sessions tomorrow, and good afternoon, everybody. Bye-bye.
The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors.
Good afternoon, everybody. So I hope you had a very fruitful day, and we have more to show or to experience today in our pre-convention activities. So today, our last of our pre-convention activities, we have the Mu Talk on microbiology education, a panel discussion on the current state of online microbiology education. So let me introduce our panelists for today. Next slide, please. So, let me introduce myself first. I'm sorry. So I'm the
You introduce yourself.
Oh, so I will be moderating this discussion. Okay, so I'm Dr. Howell Ho. For those that would not know me, I am a graduate of De La Salle University, PhD in biology, and I earned my BS Biology at the University of the Philippines, and my research interests include, among other things, physical and mental health, environmental and genetic biology, bacterial and viral and vector disease dynamics. I'm currently the Vice President for Academic Affairs at Trinity University Asia, and also the Vice President of the Philippine Society for Microbiology. So let me introduce our esteemed panelists. Foremost, of course, is Dr. Esperanza C. Cabrera. Okay. So yan, so yan si ma'am, my mentor.
Yes, good
colleague
afternoon, my
and very good
mentee.
friend.
Yes, that's true.
Yes. So she's a fellow of the Philippine Academy for Microbiology. I know this a formal introduction, but this is a very informal talk. But let me give you some background of the introduction. I had a very difficult time cramming all their all their information in very few sentences. So this is really a very much abridged version of the actual, background of my esteemed panelists. So Dr. Cabrera is a fellow of the Philippine Academy of Microbiology. She earned her MS Medical Technology degree from the University of Santo Tomas, summa
Sir
cum
Howell,
laude.
that is BS.
Ay, sorry.
BS.
Yes, BS. Nagulat din nga po ako. Iniisip ko nga kanina. Sorry for the
It's
mistyping.
okay.
Yes. And then she was awarded by the Philippine Professional Regulation Commission as the 2018 Outstanding Professional in the Field of Medical Technology, and a recipient of the Dr. Alfredo Pio de Roda Excellence Lifetime Achievement Award given by the Philippine Association of Medical Technologies, among other awards. She was also awarded the Outstanding Microbiologist and Professor William L. Fernandez Excellence in Microbiology Teaching Award by, of course, the Philippine Society for Microbiology. So we have here Dr. Cabrera.
Thank you, Sir Howell.
Yes, and then also joining us will be another a very close friend of mine, also and colleague and mentor. So, Dr. Thomas Edison de la Cruz. Sasabihin ko sana teacher ko kaya lang di kayo maniniwala.
Oo naman, hindi sila maniniwala.
But si Dr. Tom, very young pa yan, but he established the Fungal Biodiversity Ecogenomics and Systematics Group at the UST Research Center for the Natural and Applied Sciences. Yan. Dr. De La Cruz completed his Doctor of Natural Sciences at the Technical University. Naku, hindi ko alam paano i-pronounce to. Braunschweig? Tama ba, sir?
Braunschweig.
Braunschweig in Germany.
Yeah.
Okay, so Dr. De La Cruz, interestingly, is also one of the 10 outstanding teachers in the Philippines in 2015 and one of the 10 outstanding young scientists of the Philippines in 2012. The American Society for Microbiology also awarded him with the ASM Graduate Microbiology Teaching Award in 2017. And he also received the Philippine Society for Microbiology Outstanding Microbiologist Award in 2019. He served as the Philippine representative to the Asian Mycology Committee from 2007 to the present. And he is also a fellow of the Philippine Academy of Microbiology and as well as the Indian Mycological Society and the Global Young Academy. Yan, si Ma'am Sir Tom.
Thank you
Thank you,
very much,
sir.
Joel.
Thank you, sir, for joining us. And then third in our panelists, we have Dr. Supachay Basit, also a very good friend, mentor din, and colleague. Dr. Basit also has a very CV, so we had to cut it very, very short. So Dr. Supachay Basit is the founding dean of the School of Medical Technology of Emilio Aguinaldo College in Dasmariñas, Cavite. He finished his Bachelor of Science in Medical Technology from Centro Escolar University, and he received numerous accolades, including the Outstanding Medical Technologist Award given by the Professional Regulatory Commission and the Philippine Association of Medical Technologists, PAMET, both in 2017, and the Crisanto Almario Memorial Award for Research given also by PAMET in 2011. He was the former president of the Philippine Society for Microbiology and an international fellow for microbiology education given by the American Society for Microbiology. Dr. Basit?
So, good afternoon, and it's always nice to be back in every PSM gathering, so it feels like home.
Okay,
Thank you for inviting me.
So thank you very much, Dr. Basit. So maybe I can invite all our panelists in together, technical team. Yes, and then may I have this next slide? So this discussion is a really very informal discussion. We actually debated on this several months on how to do this, and we realized that it would be, I think, more appreciated by everybody if we just have a candid conversation about microbiology education, because it was so difficult for us to pick what topic to choose. And the reality is that education right now is really fast changing. If we create a PowerPoint today, tomorrow the content will already be different. So we sort of decided to go for a very casual, straight to the point, discussion, okay, instead of a prepared, but we will have some prepared materials for you as they become relevant. Okay, so the discussion for today will follow the following outline, parang lecture lang, no? So the first part will have some ideas from all of our panelists on the current state of microbiology education, just a bird's eye view, okay? And then we will start our discussions on keeping updated and things that we need for retooling. And of course, afterwards, we will talk about the delivery of the lessons and content online, which is, I think, a major issue right now of many people. And then we will, excuse me, nawala yung view ko, sorry. Maliit na, lumiit yung hindi ko makita. Naka-glasses na, di pa makita yung screen. Then we will go into laboratory skills, and then we will end our talk about assessment and evaluation. And then, of course, we will recap this with our main takeaways, okay? So I'm very excited for this because our panel has a very diverse, set of skills and experiences in microbiology education, okay? So, the first question is, this is for all our panelists, is what are your general thoughts on the current state of microbiology education, especially in the past 2 years? Maybe we will start with Ma'am Cabrera, Doc Cabrera. What are your thoughts about this? It's a general idea.
For the past 2 years, we're going on 3 already,
Yes.
and everything had been online in delivery. It really was very chaotic at first. Even up till now, we still have the birth pains, we still have the transition pains. And me personally, I have questions about the effectivity of this online education. We don't have any choice, we just have to do it. But then the thing is, there are so many adjustments we have to make, so that if you are not a dedicated teacher, then do not teach, because you're going to ruin the education system further. And so what we're now more concerned with, aside from the formative learnings of our students, would also be, aside from the summative learnings, would also be the formative learnings. So many aspects that we have to take care of, not just the course contents, but also how the students are taking them in, not only their physical health, but very importantly, and this is coming out so often, their mental health. Ang daming mga issues dyan. And so we really have to be on our toes as educators, so that we don't do a disservice to our students. We owe it to our educational system.
Thank you
So
very
far, that's
much.
it.
Thank you very much, Dr. Brera. So very interesting the point. No? So it's not a matter of degree in what you know. It's more of the dedication,
That is true.
the human side of the teaching. No? So
Yeah.
this is a very, very interesting point. So let us hear from the ideas of Dr. De La Cruz on the current state of microbiology education.
Yeah, I actually agree with Ma'am SP regarding her observation. And for me, I think the last 2 years is very challenging for those who are teaching microbiology and probably for other natural science programs. This is because microbiology is a skill-based program. So we need to develop technical skills, and that's very important. And we usually do that through the laboratory courses. And since
Yeah.
we don't have any laboratory activities, or we don't have laboratory facilities available at the moment, since we do our classes at home, I can only imagine we have now graduates of microbiology who are probably well versed with the principles of microbiology, but with have very limited technical skills. No, so they probably experienced only 2 years of lab work, and they probably had that with their basic biology and basic microbiology courses. So the technical skills for the higher microbiology courses, they never experienced it. So I can only imagine the anxiety that they will feel on their first day at work, knowing that they have the concept, but they have this limited experience in doing microbiology experiments or microbiology work.
Yeah.
So it's very
That's
challenging.
true.
Thank you very much, Sir Tom. Yes, very, very nice insight. So the anxiety level is not only on the teacher, but also on the students, that when they graduate or when they finish their course, uh, are they ready for actual microbiology work? So that's a very interesting take. So it's really, the mental state of people is really something that is of being focused right now. Interestingly, because what we're questioning here not the knowledge or the technicalities of the skill, but it's more on the, do you feel ready? Do you feel capable? Something like that. So very interesting idea. Thank you, Dr. De La Cruz, for sharing this. So let's hear from Dr. Basit on the current state of microbiology education.
Hi, good afternoon, everyone. So before I say my piece, I would like to quote a quotable quote from Dr. Cabrera saying, if you're not dedicated, then do not teach at all. Well, that's very true, because in the past years, I've experienced faculty member, if there is such thing as an absentee voter, there's also such thing as absentee teacher, particularly online class, because it's really challenging, especially for us administrator
Yeah.
to monitor their attendance. But I would say that the current state of microbiology education, if I will be using a secular term, it's like we are in the limbo. It means that We're already out perhaps of a burning wood, but we're not yet on the pinnacle of paradise where everything is perfect, right? So to translate it in a more comprehensive manner, I think microbiology right now is having the best of both worlds. I would say both world because looking back, it's quite difficult to invite international lecturers, quite difficult to invite professorial lecturers from a remote area or different part of the world. Let's say, for example, Tom right now is in Germany and he's teaching in the Philippines, right? And I would also say the worst thing is, of course, I would like to quote Dr. De La Cruz on that matter because it's quite difficult to assess the psychomotor capability of our students, right? We do not know if is it really the basic stuff such as proper pipetting or inoculation into petri dishes. I'm not yet sure if they're really capable of that because all I hear now is, of course, the click of the mouse whenever they're doing the virtual simulation. So I would say the best and the worst of both worlds. So yeah, it's like we're still in the limbo.
Thank you,
And,
thank you.
and if I may
Yes, ma'am.
add,
Yes, ma'am. Yes,
Sir
ma'am.
Howell,
Yes,
following
ma'am.
up on what Sir Tom mentioned a while ago, because of this limitation in the development of laboratory skills, then it also affects their research capability.
Yes, yes. That's
So
right.
it's not only passing the course because you had the laboratory course, you were able to do the different activities, but you just cannot apply whatever you have learned through online classes to your particular line of work. So in the future, what kind of researchers can we have? So they have to retool themselves on the actual work. And not only on the skills, but also how research designs should be framed, how studies should be designed. So it's really a long, long thing. But the thing is, let me just tell you honestly, I was caught flat-footed when the COVID uh, lockdown happened. Even before that, our university, De La Salle, had already been so active in imbibing in us the need to teach online. Like you can have hybrid course, whatever you call it, flex course, whatever. But I just always felt that the best is really the presence of warm bodies, in-person classes, face-to-face classes. You interact more, you learn more, etc., etc., and you get to know if they're honest during exams, etc., etc. But the thing is, there's no choice, no. So I finally had to embrace it, having to admit, accept that online classes had been offered already decades ago. Meron ng ongoing na mga talagang kahit na walang lockdown, even without the lockdown, you have distance learning courses, you have online classes. And whether COVID will be here to stay or not, I'm pretty sure we will still have this type of platform. So we just have to embrace it. Yun ang unang step ko eh. I have to embrace it. I have to learn all the technologies. Ano bang LMS, Learning Management System? Ano ba yun? Ano ba yung Canvas? I just have to take it in. Nakakapanik. Di ba? Doon ka magbigay ng exam na hindi mo alam anong button ipipindot mo. Baka lumabas yung answers pag mali ang ginawa niyo. Hindi niyo hinide yung file. So that's my sharing. Ang difficulty ko. Yun.
Well, Dr.
Pero masarap
Cabrera,
ang feeling, no? Masarap ang feeling na finally, marunong na ako. Kahit na marami pa rin dapat aralin. Oh, pero
Dr.
marami
Cabrera,
pa rin
may I
dapat.
just share something? Actually, my experience is the other way around because even before the pandemic, I was the e-learning coordinator of our institution. So I've been exploring this LMS and I even have my YouTube channel even before the pandemic. So, so when the government when the government announces ECQ, I have the materials that would last me for the entire SEM. So the adjustment was actually very minimal on my part, but then there were still challenges. So you're correct. This online modality should have been taught way before the pandemic, so that when it struck you, then you'll not be caught flat-footed.
Thank you, thank you. Thank you for that, Doc Bassett. I would like to read a comment from one of our
attendees, participants. So please do. I forgot to mention this a while ago. We're not just here to talk to each other. You can interact with us. You can type in the chat box, and then our moderators will forward your messages or your questions. But I would like to just read one of the comments put here by Mom Eloreta. Uh, she said that a microbiology teacher should be innovative enough to teach the skills with fun, to encourage students to love the subject. And she says that is her opinion, but I guess that is what Dr. Brera and Sir Tom and Sir Basit is saying, right? There were changes, whether we were prepared or not, we have to cope with it. And the focus was our students, right? And all our fears or our misgivings, we just had to set them aside and then, accept the fact and then look at the positive, like Doc Basit said a while ago. We can now invite speakers from foreign countries to come to our class, which before you cannot do that because you have to, pay for the plane ticket, put them in a hotel, right? And then feed them. But now, like if I want to invite Tom to my class, I can do it anytime.
That's true.
That's true.
Grabe
Libre yun, ano?
naman,
Libre.
grabe
Libre.
naman yung feed them no? Feed them.
Oh, yes, yes. Now, if they want to be fed, you can say that, oh, I'll send it through you through GCash. You do not have to worry about the caterer and everything. So, but kidding aside, but that is true, no? Being innovative is actually very important right now
Yeah.
as a teacher, di ba? So you need to be able to adapt and to go with the times, right? So very interesting, no? So as you can see, though, it's a very multifaceted problem. So let us go through it one step at a time, so that we really look at all the aspects. So first, let's look at the current challenges, okay, of microbiology education. So maybe I would like to ask Doc Basit to maybe expound a little bit about the modalities of microbiology teaching right now.
Okay. So actually, there are several modalities that can be done when teaching microbiology online. So nowadays, we have several terminologies that we have become familiar with. Let's say, for example, are you doing it synchronously or are you doing it asynchronously? But before, that might sound Greek to us, right? So there are several modalities. Let's say, for example, personally speaking, I have actually made my learning management system attractive to my student. If I may share my screen, I
Yes,
just
sir.
want to share my LMS. Teka lang, baka ako ano ma-share ko.
Wow, may
Yan.
sariling LMS.
Can you
Wow.
see? This is
Wow.
my learning management. We're using Brightspace, and this is
Okay.
my learning management system. See, I have a banner, a customized banner, and then I have I have announcement, and then if you will be looking at my content, yan. So I have actually organized all of the contents. And in order for the students, in order for the bandwidth, so the would be minimized so that the current problem is internet connectivity. I have actually linked my 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 lecture, for example, to my personal YouTube channel. So that way, if you will be looking at my YouTube channel, so this is now my content. So all of my lectures are actually here, and you know what? It's not that I'm boasting about it, but somehow I was able to share also my lecture to students from other universities and also students who will be taking board examinations or licensure examinations. So sometimes I'm elated that they are reviewing my YouTube channel before taking the board exam. So somehow I'm also elated as well. So you know, because we have to share knowledge, right? And so there are several modalities that we can actually do. Well, of course, you have to check with the data privacy mechanics of your institution. But after being cleared with what I'm going to do, then they give a go signal to go about it. Then that's what I did. So minimize the bandwidth. So instead of, let's say, for example, uploading the raw video, then a link would do, because that would somehow minimize the bandwidth consumption, the data consumption of the students, and most importantly, it is not enough that you post the content without facilitating a discussion, because that is very important, because many of the faculty members think that a synchronous session alone is enough. Okay, so just place the content. However, if you are not going to process it, it's just a basic principle of IPO. You have the input, then you have to process it. How would you process it? It's like a scaffolding. You have to build the knowledge, you have to build the content so that you'll be able to remove any tenacious prior knowledge or any alternative conception, so that the output would be aligned to the intended learning outcome of that particular topic. Then you can say that somehow you did your job as a faculty member, even in a digital online modality. I think that's my thought on that matter.
Thank you very much, Doc Basit. So just as a side question, should one utilize or know all kinds of teaching modalities, or can just one just master one kind and focus on that?
Is that for me, Sir Howell?
Yes, sir.
Actually, you have to choose modalities that has been validated. That's very important.
Okay.
Or content that has been validated. And then, you have to ask, you have to self-reflect, is that particular modality appropriate and applicable for you, for your students, for your particular setup? Because if you remember, CHED has actually, at the early onset of the pandemic, CHED has actually classified students, those who are capable of online, those who are not really quite capable of online, and those who are not totally capable of having an online class. So for the third category, so the modality is purely a modular type of instruction. However, majority of us are in the HEIs, and we have opted for the online learning modality. So you don't have to master everything. So the main thing that you have to remember is think what is appropriate and applicable to you, to your institution, and to the stakeholders that you are catering. However, very important also to choose the content properly. Even before the pandemic, that's actually the basic role of a faculty member.
Thank you very much. So I think Dr.
Sir
Raimundo
Howell?
yes, Dr.
If I may, I just add to what
Yes,
he was
yes.
saying. No,
Yes, ma'am.
maybe
Sir Tom. Yes.
another thing that you should consider is think of choosing something that you really like to do. For me, this is very important. There's a saying that suffering is a choice. So you might be doing a modality that you have difficulty doing it, and then it becomes a burden on your part as a teacher, and it reflects actually when you teach, because if you feel that it's a burden on your end, then you don't do it well, and it also goes trickle down to your students. So I think if we are to choose a modality or a strategy that we would like to apply in our class, make sure this is something that we like to do or we are enjoying in doing or in preparing. So for me, that's my take on that.
Oh, thank you very much.
If I may, before
Yes, ma'am.
we give the floor to our distinguished Dr. AKR.
Yes, ma'am.
Because I'd like to follow up on what Sir Tom mentioned.
Yeah.
In addition to what you are more comfortable in handling, in doing, it's also very important to consider the maturity of the students. I have, I for whatever reason, sometimes you have students who are mature grouped together, and you have students who are not so mature grouped together in one section. For whatever, di ba? Minsan meron tayong paboritong section, meron tayong kinakainisan na section. Anyway, so yun, no? So in some classes, in one of my classes, they did very well with more of the asynchronous, and then we made less often synchronously, whereas the other one had really to be met synchronously every time. And that was a graduate class compared to the other one, which was an undergrad class. So it really also depends on the student. And I agree, we just do what we are comfortable with. Kundi magkakamali tayo, bubu yun. So, Dr. AKR, please share with us.
Yeah, Dr. Raimundo? Yes, ma'am.
I'll butt in, but then I think actually
you should. We appreciate that, ma'am.
About
We
the
appreciate that.
Microbiology Consortium of the Philippines. because the start of the pandemic, we were faced really what you have been discussing, the challenge of teaching the course online. So we went through several webinars, several workshops, and we came up with a series of laboratory exercises that can be taught online. And we shared that with especially those from the so-called underdeveloped universities where they do not have the capacity to develop their own materials. So those were made available in our website, the Microvalue Consortium in the Philippines, which is available for any member of the consortium. Plus, I would also like to share the effort now of UPLB. I was asking Millet Carlos to speak out, but she's busy. How to bridge the lack of how to bridge the gap of the lack of skills. This summer, they had a bridging course. They offered a short course for those graduates who went through junior and senior during the pandemic and did not have the opportunity to do the actual laboratory exercises. So they shortened the different exercises and jump-packed it into I think how many weeks of courses, and the students had the opportunity to actually do the laboratory exercises. So I think that at least when they go there, they're not mayroon na silang alam kang kaunti.
Thank you, Dr. Raimundo. You know, actually, Ma'am Millet actually messaged me about. I was going to mention it after you, but it's good that you mentioned it already. Yes, the bridging is a very important thing. Learning to know what the students know or do not know. But I'd like to read a shout out here. I think Dr. Cabrera is a fan, no? Shout out to Dr. Cabrera, the best teacher ever.
I think July is now a student at UPLB, ma'am.
Okay.
Yeah, alagaan niyo, ma'am.
So siguro go to the lighter side of it though. I know this is a very serious discussion, but I think maybe our guests could share to us, you know, any favorite moments in teaching microbiology online. Or do you have any favorite moments? Kasi sabi nga ni Tom, di ba? Sometimes parang pilit na pilit tayo doon sa method na hindi natin kabisado, di ba? So, do you have any favorite moments?
Well, obviously, I'm so happy I don't have to set the alarm clock anymore. I have a leisurely breakfast, but I still see to it that I'm well groomed before I appear to my students. At kaya, alam mo, ang maganda sa online lang, you get to see everybody up close. Kahit na sabihin mong hindi siya warm bodies, no? But you get to see each one up close. That is if they will say yes to turning on their webcams. Kasi madalas sasabihin nila, wala silang mabagal ang internet connections, etc. So they will not turn on their webcams. Tapos sasabihin, okay class, let's have our photos taken. Ayan, bukasan na ng webcams. Biglang pwede na magbukasan. Pero ang hirap talagang mag-isip ng isang moment na siguro masasabi ko ang na-appreciate ko sa online classes, challenging siya. Na-challenge ako. At lahat gagawin mo, song and dance, talagang gagawin mo na sa klase yun eh. Tapos may break ka, magpapa-exercise ka, lahat gagawin mo as what Ma'am Minette was saying. So nagiging mas innovative ka para kunin mo yung attention nila. And I think that helps you develop yourself as a teacher further. So hindi lang content ang focus mo, pati na yung classroom management, ibang klase na rin. I think that's what I like, the challenge in it. Pero nakakapagod talaga.
Very nice, ma'am. Thank you, ma'am. How about Doc Bassett or Sir Tom? Do you have any favorite moments?
Sige. Sige. My favorite moment during online class, so we are of the same height.
Isama mo ako dyan, isama mo
ako dyan. Height, and then, what else? I can play around, so I can wear hat like this. Alam, di ko na maalis. Okay, so what else? And then,
most importantly, ka nun.
Grabe. And most importantly, mas gwapo ba pag nakamas? Most importantly, I'm able to interact with my students. So I find a way to interact with my students at the comfort of my home. That was before, but now I'm reporting on site. At the comfort of my home, and then while, of course, while playing with my son, for example. So that's actually my favorite moment. So there was even time that my son joined my online class, but that wouldn't be possible if it's in-person class, right? So yeah, you have to find a way to interact with your student. Let's say, for example, a simple word such as "Kamusta kayo?"
Oh,
That
yeah.
would really mean a lot. That
Yeah.
would really mean a lot, no? Kamusta kayo? So do not go directly to your lecture. So, di ba?
Yeah.
Kamusta kayo? And then, sometimes they would open up, bigla na lang, Dean, hirap na po kami. Bigla na lang makakarinig kayo ng gano'n. So, yon. After all, we're still humans despite of these modalities. Thank you.
Thank you, Sir B. Sir
Mhm.
Tom?
Yeah, I agree with B, no? So, sa akin, wala rin ako masyado maisip ng mga key moment. Although, I just realized that as a teacher, we usually give jokes when we give our lecture face-to-face. And I just realized we can also do it online. So I remember in one of my classes, I was asking my students a question, and I think you have all experienced it when we're teaching online. Nobody will really raise their hand
Yeah.
or reply to your question.
Many
And
times.
so what happened is that the rooster beside our house, you know, say
Crowed.
something. And I would say, oh, buti pa yung manok sumasagot, kayo hindi. So something like that. No, it's something funny in a sense, but I think tama si Bino. We need to really find a way to continuously engage and interact with our students. Kasi in online learning, we don't see them unless, as mentioned by Ma'am SP, we ask them to open their video, which they really do. So we need to find ways to interact with them to make the conversation as if we are in a real classroom. Yeah.
Oh, thank you, Sir Tom. May comment dito si Ma'am
Yeah.
Alayla.
Magandang
Totoo
comment
to.
yan.
Uso din po ang ghosting sa synchronous
Yeah.
classes, di ba? Parang
That is
two
true.
new words, di ba?
Mm.
If you do not know what
Mm.
ghosting and synchronous is, di ba? But, di ba, that's a reality. No? Tama. Thank you, ma'am, for that comment. That is true.
Nandun yung picture niya, no? Tapos wala siya.
Yes, that's true. That's
Tapos
true.
nag-end ka na ng meeting, nandun pa rin siya.
Yeah.
Natulog na siya nun.
Hindi, ma'am, naka-next class ka na.
Naka-next class ka na doon.
Yung nga, ma'am.
That
So,
is true.
so following up from what Sir Tom said, so sir, what do you think are the current demands of teaching, or what is the is the current way of teaching more demanding, or is it the same, or how is it? Paano ba yun, sir?
So you're asking me about the challenges of microbiology education, siguro, no?
Yes, sir. In a way, yes.
Yeah. Mhm. So as I've earlier stated, no, microbiology is a skill-based program, and I think for me the challenging aspect of teaching microbiology online is how do we develop this technical skill. We usually teach microbiology as a lecture, and there's always this laboratory component. And in online education, when we teach the laboratory component, we usually do it by providing them with instructions that we hope they will follow, or we just provide them with a video demonstrations of how things are going to be done, or we tell them to do it using virtual simulation, or what we call virtual laboratory. And the thing is, we don't know if the students are actually doing things correctly. So this is for me the challenging aspect. So how do we ensure that the students are doing things correctly? Okay. For example, streaking. This is a very basic microbiology technique, no? Streaking. Um, in face-to-face, students will do the streaking, in front of us, and we can always provide them with feedback. And the feedback is real time. We tell them, oh, that's wrong, or you do it this way, this is easier. And the students learn immediately. In online learning, the feedback will come after. So they provided you with the result or the output of their streaking. We don't know if this is um, done correctly in the process. So I think para sa akin yun yung isa sa mga challenging aspect. How do we ensure that the skill of the students will progress even if we use these virtual simulations or if we teach the laboratory courses in under an online setting.
Thank you for that, Sir Tom. We have a comment here from Sir Rance.
Kay Ma'am Minette.
And also Ma'am Minette, yeah. So let me read first the other comment was, I think one of the challenges of microbiology education is we need to emphasize other areas in microbiology, since based on my experience, teachers and students think microbiology solely on human pathogens. Yes,
that's true. That's true.
That's true. Tama, ma'am. And then from Ma'am Minette, during the pandemic, my subject, microbiology, of course, was not offered. Oh, that's very bad. I missed teaching it. What I did was check my skills all alone in the laboratory. I was just working or experimenting on my own. Okay.
Yes.
So
here's another comment from Renz.
Oh, yes, there's a follow-up comment. So we need to address other areas of microbiology like food micro and microbial ecology. Yes, definitely.
That is true.
Yes. So, Ma'am Cabrera, with this in mind, so how do you think one as a teacher would be able to equip ourselves with the, if suddenly may pandemic, then everything we know about teaching suddenly change, and then we don't know, maybe tomorrow it will be a different way of teaching. So how does one equip oneself for this eventualities?
With what are happening now, we have to equip ourselves in two different fields: how to offer the course online, how to handle the LMS, how to monitor their exams, etc. Another one would be retooling ourselves, updating our course contents. Okay, and especially in microbiology, we have so much resources in microbiology that it is a sin if we do not update what we are teaching. Kasalanan yun. Ang dami-dami hindi mo nag-meet. And so, alam mo, sir, well, I prepared a little something.
Yes,
may
ma'am.
pinipare ako,
Yes.
I will share.
Please share.
Yes. And because I'm so, you see, in whatever course, not only in microbiology, but very especially in microbiology, because it's so dynamic, it's so complex, it's so abstract, it's so hard to teach. So we have to really be updating ourselves. And of course, we have the traditional resources, like we have the hard copies of references, etc. But we are so blessed because there are just so much resources in microbiology, especially so in the internet. I would like to emphasize that. It's a sin if we do not use these properly, but we have to be very judicious in choosing the sites from which we're going to take. So if, let us say, for example, we talk about the resources in the internet, and so I will not go through that because everybody knows how to source. We just have to have the correct keywords, Google, and then we have it, and then we choose the correct one. So here, for example, we have this online course. Let me just take away something I cannot see here. Ayos siyang malis. Should I admit somebody in the waiting room? I will admit na para matanggal na.
Sige po.
Yes. Okay. So here, for example, I just keyed in online resources for microbiology. And then you'll come up with so many. Example lang yun. Kasi everybody can do this. It's so simple. Microbiology Online, that's the site. And then you now have for teachers, micro passports for students, podcasts, videos. We are familiar with those. And another example, here you also have virtual microbiology classroom. And here you have the virtual micro classroom instructor's corner. There's also another one for students, etc. Another example from ASM, you also have the virtual resources to teach microbiology techniques and experiments. So this would be more for the laboratory course. But the thing is, sometimes the title will be so promising, but when you go to the site, ito pala yung laman nun. It can also be very disappointing. But that is not my point. What I would like to share with the group is that sometimes we have the course syllabus. We always have the course syllabus. But when we now update the contents, many times our main focus would be what we have in our reference books. We follow the contents there and we update on those particular contents. But the thing is, if we are now going to use the different textbooks, we can see, for example, this is Brock's Biology of Microorganisms. And this is the latest edition, 16th edition, published in 2021. And this is our Bible in the PALM certification exam. So this is quite new, 2021. And the thing is, when I went through this, when I was trying to update my lecture, it doesn't say much about COVID-19. I see only it appearing 4 times, and the SARS-CoV-2 virus was not discussed at all. So talagang kung ito ang ating basehan, wala tayong makukuha. Aside from that, I would also like to stress that we have to include in the updates of our teaching materials information that are new, interesting, fun, and relevant. So hindi tayo mag-stick sa ating mga textbook dito. And I have problems going to the next page.
Mhm.
So here, for example, you have, let me share why I like this. Key in updates in microbiology, and then you have these several resources that we can use. You have Microbiology News Science Daily, and microbiology latest research and news, Nature, etc. So if we go to one of these, Microbiology News Science Daily, pasensya na kayo, time to buy another laptop. Here, from Science Daily, look at what you can see. So this is July 18, 2022. And so if, let us say, you are working on particular topics in general microbiology, and you can just look at this one here. The first one here would be insects harbor over 1,000 genes from microbes, which help them survive. So you can use this material in your discussion on introduction, on evolution, or on microbial genetics. Here, you talk about as glowworms, infected pines starve and disease-causing fungi thrive. So you talk about host microorganism interaction. So you can Clostridium difficile. If you're talking about the human microbiome, then you can talk about the role of your Clostridium difficile in so many other diseases. And here, zombie fly fungus lures healthy male flies to mate with female corpses. So interesting topics. The thing is, you can just click on them and you can have information, and you can have abridged information, and you can decide if you'd like to proceed with that particular item or not. Dito kunyari, Nature featured Omicron, a shift in the biology of SARS-CoV-2, July 12, 2022, etc. And if you now click this Omicron, you now go to that write-up about Omicron, and you can download the PDF. And if you now download, it is a short review, a brief account of that paper. So research briefing, everything is in a nutshell. And then you say, okay, I can use this for my class. I can now go to the article. So here, it's like shopping for whatever you can include in your updates. Yung ganon. And that can be so interesting. I'd like to jump because I don't like to monopolize the show. We always talk about prokaryotic cell, eukaryotic cell. The main difference is that the eukaryotic cells will have organelles bound by the fluid mosaic membrane, whereas the prokaryotic cells do not have that type of organelles. And they now found the bacterium, which is, aside from being the biggest so far, and this is in comparison to a coin, you have this organism. You have your Thiomargarita magnifica. It is said to be about 1 cm in length, so macroscopically observable. And they now found that the DNA molecules, which are present in so many copies, are surrounded by a membrane. So it's like a feature of the eukaryotic cell. So now they're saying, okay, so how can you now define what a eukaryotic cell and a prokaryotic cell is? So something like that. It could make interesting introduction, part of an update. So yun lang. So my message here is when we update our teaching materials, include new information from the internet that may not be found in your textbook. Wala sa Brock yung SARS-CoV-2, for example, so much. Yun lang. I will end my share. Thank you.
So thank you, Ma'am, for the sharing. No? But very interesting. No? Tama kayo eh. The interesting thing about our current mode of teaching and the current resources that we can have, you have an unlimited source of materials.
Yeah.
And then, very interesting, even our Bibles, they don't have the latest. So there are other sources. So ngayon, the question is, how as teachers do we guard ourselves against fake news?
Oh yeah.
Because
yeah.
I'm sure, diba, I'm sure you have encountered some information on microbiology. Initially, pagbasa mo, oy, this is very interesting. I would like to share to the students. But I'm sure our audience here will also be wondering about that. How I'm going to protect myself? Baka mamaya, I keep expounding on this new discovery, but then just to find out a month later, biglang i-withdraw pala ng journal yung article na yon, or something.
Yun ang mahirap.
Yes. Or later you find out it's a fake news na create lang pala ng isang tao na on the internet. How do we guard ourselves as teachers from these things, ma'am?
Kasi the first answer is look at the source. Is it a credible journal that you're taking it from? If it's from Nature, siguro naman okay because they have been vetted. Yung mga tipong ganun, are the articles published in that journal peer-reviewed and ISI or Clarivate Thomson Reuters, however it is called now, no? Clarivate, Web of Science, Index Journal, Scopus Index Journal, yung mga ganun tipo. Kasi maraming predatory journals din. So we just have to be very careful. Even, and if I may add, even if, for example, you have so many YouTube videos on simulation, right? How to streak. We also have to be very careful. Hindi lang yung assign natin. Papanuin na because I've come across so many videos that are faulty. Okay? Minsan, pinapanood ko yun sa estudyante and I ask them, pick out what are wrong in the videos. So that one is also a learning experience. Pwede rin yun. Pero yun ang tanong mo na mahirap sagutin. Paano ka ma-assure parati?
That's true.
Yeah.
Look at the source.
Yeah, if I may add to what Ma'am SK is saying, we need to be critical as teachers with the resources that we use in class. Hindi dahil maganda or na-share sa feedback sa newsfeed natin sa Facebook, we immediately share it to our students. We need to, check it first. Okay ba to? Hindi ba to okay? But I like the idea of Ma'am SP, na if you see some faulty materials, you can still use it as a learning material, no? Not to teach the wrong aspect, but to find or to make the students see the mistakes in those materials. So which is also a good strategy na pwede rin natin gawin. So basically, it's teaching our students to be critical. And I think yun na importante right now, no? We have so many fake news, misinformation, disinformation. So we need to teach our students to be very critical with the information that they are getting. Now it's easy to get information with just a click. You get all the information that you need, and critical thinking, yun yung medyo mas challenging na dapat na matutunan natin at ng ating mga estudyante.
Tama, sir, no? It's really, no? Parang maganda yung sinabi ni Doc Cabrera talaga, no? Instead of pining over this fake news, use it as a teaching moment, di ba? Maganda yan, looking at the positive side of it, no? So this fake news is a way for us to teach our students also to be critical, to be able to weigh the facts, put it together. So it's parang ano yan, parang as you learn, they are also learning at the same time. Very interesting, no? Yes, very nice, very nice. So I guess there's a lot of chat. I'm so sorry. I have to skip
Yes.
through a little bit because medyo inaano-ano ko ng ano. Sabi daw, next topic na daw ako kasi sumobra na kami sa but very interesting our sharing. So we were talking a while ago about the how the need to be updated, and then the need for retooling. Then we ended up about fake news and creating new strategies like developing critical thinking. That's very interesting. Now let us now move on to one of the, I think this is one of the big problems in microbiology education right now. How do you deliver your lesson and then ensure that you're able to cover the content, di ba? That is very, very difficult. So maybe let me ask Sir Tom,
Mhm.
so uh, what are the realities relative to this, sir, in terms of how do you deliver the content that you need to cover, di ba? Like before, kunyari, kung kailangan ma-cover mo in 20 chapters, broke, let's say, for example, but how do you do that right now? Is that possible? Do you still do that? Ano ba yung dapat consideration, Sir Tom?
I think ito yung siguro dapat magkaroon na tayo ng paradigm shift no in terms of our mindset. Sa Pilipinas kasi we are very used to this kind of idea na okay, we have this core syllabus, we have 20 chapters in this core syllabus, we have to cover all these 20 chapters. And if you don't cover these 20 chapters, you as a teacher, you feel, okay, I did not teach well. And I think we need to have this change in mindset. No, I would like to follow up on what Ninang SP actually said a while ago. No, she said that we need to update our content. Okay, this is an important aspect that we need to do when we are trying to now deliver or design our lesson plans. So updating our content, but at the same time, we need now to choose which of those content are actually relevant and more important to teach. So we need to prioritize.
Yes.
Okay. So we have 20 chapters in our core syllabus. Do we teach all these 20 chapters? Well, the thing is, if you teach all these 20 chapters, how many of the 20 chapters will the students remember after your course? I'm sure they will not remember all those 20 chapters. So you need to decide now which of these is more relevant to them and which of these chapters they can actually learn on their own. So if you still need to cover these 20 chapters, that's fine. But tell them, okay, I will not cover chapters 1 to 5 because this is something that you can learn it on your own. So do it, your own time and your convenient time. Or maybe these chapters 1 to 5, you learn it already in your general biology class. So why do I need to repeat it in the new lesson? So you just focus on what is more relevant, more important. And I think one thing that we should also be doing is we teach our students how to learn themselves, no? Importante yon. Kasi tayo parang we have this mindset that we provide them this information. All information are given to you. Sabi nga, spoon feeding. And our students never learn to be independent learners. And I think yun yung dapat na baguhin natin. Na we should make our students now an independent learner so that we don't need to cover all these 20 chapters. We only cover 10 of it, and then we still assume that the other remaining 20 can be learned by our students on their own, at their own time, at their own place.
Tama, Sir Tom, no? Very interesting the point, no? Kasi it means that you don't have to cram everything. You don't have to worry about, I need to finish all of these things. Hindi maging capable ang estudyante ko. That's not the point, di ba? So like sinasabi ni Dr. Cabrera kanina, you make use of the situation. Turn it around, di ba? Make it a teaching moment. So let them learn that, you can learn on your own so that you are not worried about your future job. So you study, right? Find out all of these topics about, and then you ask your teachers if you don't understand them. So you're converting it so that the response teaching is really a shared responsibility between the students and the teacher, no? So instead of hinahabol mo yung topic, trying to come up with ways to cope up with these things, pinapagod mo sarili mo, pinapagod mo yung estudyante. But whereas you can just address the problem direct head on, right? By teaching them 21st century skills, right? Tama, no? That's a very good point. No? So siguro this is, I think, I didn't realize it until we were discussing, you know, what Ma'am Carves and Sir Tom said. Talagang, ito talaga yung kailangan natin right now, no? We have to
Yes.
realize that the moment is changing, we have to change. We cannot
Yes.
stick to the 20 chapters. You have to wake up
Yes.
to that
Exactly.
reality. But instead, teach your student that you a need for this, so learn it, di ba? And then
Yeah.
come to me if you do not understand, and I will teach it to you, whether it's
Yeah.
face to face, online, or any other modality. No, so it really changes the paradigm of things, how education works right now. So
Yeah.
maybe I'd like to
And
ask
Joel,
Dr. Bassett. No, yes, yes, sir.
yeah, I also would like to add to that. Now, let me
Yes,
share with you this experience that I have.
yes,
In
yes, sir.
2018, I went to Germany, and I went to this laboratory, or to this group, as a postdoctoral researcher, and I decided to attend actually the class of one of the classes of my professor, my host professor, and I was really amazed by which or by how he actually taught his course. So his course is fungal ecology. Okay, and it's a one semester course, and I realized, when I was listening to all the topics that he is discussing, I said to myself, well, those topics I would usually teach as one chapter in in my course in the Philippines. So I would be able to teach all the lessons in one chapter. And then when I try to reflect on how he taught that particular lesson, I just realized that instead of expanding it, it's he actually go deeper to the concepts.
Yes.
So the topic is just very few topics, which, as I've said, you can teach in one sitting
Yes.
in your syllabus, no? In your syllabus. But when he was teaching it, he was going deeper, and I think this is something that we should also be doing right now.
Yes.
So it's not
Yes.
about the amount of lessons that we are teaching our students at this point, but it's more of how deep we should be teaching those particular topic.
Yeah. If I
So
may,
yun yung aking
Yes, ma'am.
experience.
yeah. If in the way we handle our course, and Dr. Lynn Moron is here in the audience, and we
Yeah.
teach micro together in our school. It's more of covering the depth rather than the breadth, because they can very easily just read and learn the surface materials. But it is the understanding where we go in. So let us say, for example, in bacterial structures using Brock, I take up with them the structures of the peptidoglycan and the cell membrane, plasma membrane, because those we take up at the molecular level. So they have to be guided. They will be reminded of their phospholipids, etc., in their biochem courses. But then when it comes down to the other bacterial structures, which are easy, lightweight reading material, like the inclusion bodies, the appendages, etc., they are asked to read on their own, and we provide guide questions so that they would know what to emphasize. And then we don't, if, for example, the term just will not allow it, even before the COVID pandemic, sometimes you have suspensions, etc., class suspensions, and now we don't force ourselves to finish everything. We don't machine gun through a topic just so that we can cover the entire syllabus. It's more of the quality than the quantity. Parating ganon. And then we always tell our class, we are teaching you how to learn. So you should learn how to learn by yourself. Parating ganon yun eh. Hindi na yung spoon feeding. Yung mga ganon. Pero mahirap mag-balance kasi may mga questions sa evaluation mo. Yung mga ITEO evaluation. Minsan doon ka nadadali eh, no? Isipin nila hindi ka nagtuturo. Basta yung ganun tipo,
That's
yung
true.
pag-balance nun.
Yeah.
Tama, tama. So, tama yan, ma'am, no? So, the gauge of a teacher right now is not the same. Evaluation
Yeah.
tools also have to adapt, no?
Oo,
So,
yes.
di ba, ma'am? So,
Oo.
maybe I'd like to get the opinion of Doc Bassett, no? So, for a teacher, di ba? So, how should you be measured, no? Are you measured by the quality of your lesson? Are you measured by the quantity of your lesson? Or how do you enrich your lesson, Doc Bassett?
Well, first of all, I'd like to address the first question that you'd asked me. What are the metrics being used to measure the faculty members, right?
Yes,
Well,
yes.
fortunately, I'm part of the QMS of our institution, Quality Management team, and I was part of the committee who revised the evaluation
Okay.
tool,
The tool.
evaluation
Yes.
tool, and
Yes.
we give actually premium on on the presence of the faculty member virtually, and how he or she would be able to translate the virtual presence into the realization of intended learning outcomes. And having said that, I think the one the thought that was given a while ago by Dr. De La Cruz and Dr. Cabrera would play a big or significant merit on it. Meaning to say, you teach a certain lesson, not not just not just the tip of the iceberg, but
Yes.
the actual depth of that particular topic. However, I have to balance. I have to balance the paradigm of that matter, because let's say, for example, our program has a PRC licensure exam,
Yes.
and let's say, for example, I'm teaching clinical bacteriology. So, if you will be looking at the TOS of the PRC, table of specifications, it indicates there that you have to teach all of these. Imagine, I think Doc SP
Yes.
can can
Yes.
relate with me being a medical
Yes.
technologist
Parusa
herself.
yan.
Enterobacteriaceae should be taught in one meeting. Will that even be
Yes.
possible, right? So,
Yes.
ang dami no, di ba? Not in 1 week, not to mention na yung gram-negative bacilli, clinically medically important gram-negative bacilli
Ang dami.
in 5 weeks, for example. So if we're going to really if we will actually go deeper in each of the topic, you might be sacrificing some of the topics that might be asked
In the board exam.
in the board exams, and that would be a reflection of your institution and you as a faculty member. So
Yeah.
it's quite difficult. It's quite difficult. It's quite difficult to balance it. So that's the reason why. That's the reason why. Personally speaking, when it comes to content, I have a pre-recorded lecture. So I think for me, what works best even before the pandemic was the flipped classroom. So meaning to say, flipped classroom, I have given already the content, because even before the pandemic, I have already screencast of my lecture, so that they can study ahead of time at their own very convenient time. But when it comes to actual classroom setup, I do not repeat what I've already lectured virtually. So in that case, in order to go deeper, maybe I can discuss a clinical case related to the lesson, or I could discuss a journal related to the lesson. However, they will not be able to blend in with the discussion if they haven't prepared ahead of time.
Yes.
So you're right, Dr. Cabrera, the maturity of the students would play a significant role.
Yes.
After all, teaching is not a one-way street. You as a faculty member, we have to do our part, but they have to do their part as well. So it's really a very intricate and complex discussion. But
That is true.
the root
That
of the
is
matter
true.
is everyone has to be mature and adapt and blend well to the
Yes.
situation.
Yes.
Yes, I agree with B on that one, no? Kasi I remember when they implemented the problem-based approach in
Ay,
medicine.
oo.
So it was a nice strategy actually for medical students to do this problem-based approach. However, pagdating sa PRC, yung kanilang board exam is the pen and paper style, which is more on memorization. So there were many schools na hindi nakakuha ng magandang results pagdating sa board exam. So what they did is to go back to the traditional approach.
Yes.
So I think it's very important na hindi lang sa atin ito. No, dapat it goes beyond. From the teacher, students, administrators, even our government officials, policymakers, dapat align talaga tayo lahat.
Yes.
No? Including PRC. Yeah.
I agree.
Lalim na nung discussion natin.
Oo nga, lalim na. Grabe, grabe.
Mapaiyak na ba tayo? Mapaiyak
Yes.
na ako.
Classroom.
Oo nga.
Kala ko ba light lang to?
Oo nga, let
Kala
us
ko-
lighten the mood. So I have a question, no. This is a interesting question that came to mind while you were talking, no. So ang tanong dito is that is common sense still common in a microbiology classroom right now? Hindi, no, 'di ba?
Define common sense.
Yeah, kasi 'di ba minsan gano'n 'yan, sabihin ng teacher, "Ano ba common sense lang 'yan, don't you get it?" Parang gano'n, 'di ba? But I mean, 'di ba? What's your take on this? 'Di ba? I have heard this. I very interesting. This is a very common comment, but very few teacher, acknowledges that this is a problem right now, no? Many teachers think dati iniisip nila yan eh. Ba may common sense pa ba ang estudyante ko? 'Di ba? Many people are saying that or thinking that, but they're failing to acknowledge that fact eh, 'di ba? So I guess I'd like to throw this to the body. Parang lalong lumalim pala. No? I thought it was a lighter moment. Few minutes of light moments lang pala. So, but yes, is common sense still common right now, or are we using the wrong terminology? Parang ganon, parang ganon ba? What do you guys think? Naku, napatigilan ng pati
Oo
yung
nga,
panel.
Sir Howell,
Yes, ma'am.
when you say common sense, it's like you can tell and answer from other information. I mean, you can
Yes.
extract
Yes,
answers
correct,
from
correct.
other info. So my answer is common sense is not so common anymore. Hindi ko alam kung dahil sa kulang sa critical thinking, dahil sa ang questions natin madalas multiple choice, lalo na kung online.
Okay.
Mas hindi na tayo nag-essay questions.
Okay.
Ganun. O kaya pa na enumeration para madali lang matapos. Yung tipong ganun. So we really have to be careful on how we give assessment exams to our students. Kaya ang nadidevelop sa kanila route thinking. So walang connect-connect na yun. Wala nang synthesis. Wala nang critical thinking. Walang common sense. Kaya minsan, di ba, we feel so bad na parang hindi tayo effective as teachers because we cannot make them think well. Hanggang route memory na lang. Minsan wala pa yun. Yun ang problema.
How about Doc Bassett and Sir Tom?
Sa akin naman, if we want our students to have common sense, it should start at the very beginning, at the early stage. So, yun yung problema kasi pagdating sa atin, since we're dealing already with college students, we expect that they were trained well. But the thing is, learning starts at the early stage, particularly the basic education, level, and it's very important na doon pa lang na-develop na yung kanilang skills. Now, just to give you an example, my daughter, I live here in Berlin for about a year now, and my daughter is actually studying in the German public school, elementary school, and we were surprised because she's in grade 2, all right, and they had research in one of their class activities. And in that class activity, they were taught how to construct research question. They were taught how to do mind mapping. And I was telling my wife, Jean, I taught mind mapping to my first for undergraduate students. And here, they're already teaching it to the grade 2 levels. Yes, they're doing already mind mapping. And the teacher was telling them, it's not important for you to be able to answer your research question. For them, it's not about answering the research question. It's about the process of how to construct the research questions, how to find the answer to those research questions. And they had this research presentation. One day I went to this school, and they have to explain the research question. They have to explain what they found regarding the problem that they have, and there's answer. So I think this is something that we should be teaching also at the early level. Now, I don't know how we teach basic education in the Philippines, but I think importante, if we want them to have a common sense, turuan natin sila magkaroon ng common sense sa umpisa pa lang. Hindi yung pagdating ng college, saka lang natin sila tuturuan magkaroon ng common sense.
Yes.
Wow.
Okay.
That was very interesting, Sir Tom. Pero personally kasi when you say common sense is how a student would make a practical judgment And again, the metrics for measuring it would be quite challenging for us because nowadays, even let's admit it, some of our faculty members are not even trained on how to do assessment, and majority of the assessments would just on the lower level of Bloom's taxonomy,
Yes.
on the lower order thinking skills. Because if you really want to measure common sense, then you have to go beyond, let's say, on evaluative or synthesis. So let's say you can do an essay question, but then again, there's a problem. I even have a student of mine submitting an essay, halatang halata na copy paste because pati yung ano, yung hyperlink ng Wikipedia yata, na copy paste
Walang common sense yun. Talagang walang common
sense, di
sense.
ba? So, and then when I confronted her, kasi her talaga yun, when I confronted her, I gave her 0 for that, and I said why, and said, and I was even, the audacity of her responding to me saying, can you consider it because after all, everyone is doing it. Just so happen na hindi lang niya naalis yung hyperlink. So, nowadays, wala. I'm out of word for it, di ba? So, ayun. So, depende na rin yun sa faculty member. If you really want to raise up the bar, then of course you have to upgrade yourself as well. You have first of all, you have to understand and realize and admit that there is a problem so that you can do something about it. If you're not able to accept that there is a problem, if you'll go blind about it, then you'll never able to realize it. Then no intervention could be made for that matter.
Oh, thank you very much for that, Dr. Basit. Very interesting, no, the point that you raised, no, that yes, Dr. Cabrera say that common sense is not common anymore, and what Dr. Basit is raising is that yes, because the common sense is now different, no. So, and that is a good point of Sir Tom that yes, because we now have to start earlier,
Yeah, that's true.
so
At
we need.
home.
Yes, at
At
home,
home.
and during the early years of education. So we really need to look at this. So I don't like to say systemic, but unfortunately,
It is.
that's the word. Yes, it's a systemic thing. So it's really a reformation, top bottom reformation of education. No, even though we are talking about microbiology alone, they are interconnected to other other skills,
Yes.
other knowledge, common sense, and etc. So this is what teachers are really facing right now. And then the sooner one realizes it, then the sooner that you can be able to deal with it, di ba?
Yes.
Like sabi nga ni Ma'am Cabrera, di ba, as soon as she realized, she started dealing with the problem, di ba? Yan. Very interesting take, no? I hope everybody was able to get something out of it. Now, another very common thing that many people say right now is that they say that student well-being should come
Oh.
before microbiology content. Di ba? I don't know if you've heard this argument before, no? Sabi niya sa akin na yung student well-being should come before the need to finish your microbiology content, or the need to finish this lesson, or to target this, to accomplish these learning objectives. Do you think this is true? Who wants to go first? Doc Basit? Since you already
Well,
mentioned the blooms, so maximize na natin.
of course, the personal well-being of the students, okay, these are very important because after all, we are actually talking about the sanctuary of our soul, the body being the sanctuary of our soul, and also if you do not have the body, um, if you do not have the anatomic structure and even the physiologic structure or a physiologic state to go on with this with studying, no matter much how much you put in on that particular brain, then it would be useless. It would be pointless. And in fact, during the pandemic, I've realized that mental health a problem is real. I even experienced anxiety myself. Imagine 3 days hindi ako makatulog. So that's actually real. However,
Really?
the problem is Yes, po, it's real. So, however, the problem is many of the students are actually using the mental health card
Yes,
in
as an
every
excuse.
excuse. Yes. So, sometimes it's like the boy who cried wolves. You may not even it's hard for us to determine whether he's saying what he or she's saying is true or not. So I think if you're able, I think my advice here is that if someone would say that particular matter to you, then you have to treat it seriously, but you have to refer that particular student to an expert or professional who's trained for that particular matter, because they are the best ones who can make evaluation. Because it's so hard for us to disregard it. Because after all, you are the steward when it comes to the classroom. Even if it's an online setting, you're still the steward of it. So you are still accountable to the welfare of your student. So we are microbiologists, we're not psychologists. So do not attempt to evaluate whether what the student is saying is true or not. So
It's
refer
like
them. Thank you po.
Yes, Ma'am Cabrera.
I think, as a teacher handling microbiology, our mandate is really to give them the proper microbio education. And that contributes to their well-being, because that's why they are enrolled in your class, because they have to learn. So having received the correct education from you, and you have given it properly, it's like you delivered the course efficiently. They also have to do their part to learn. That is already contributing to their well-being. And then you're handling a class of, let's say, 40 students. And so you cannot just cater it to just one student at all times. It's that you want to be fair to everybody. So you have to give what are expected in that particular course. You're not going to water it down because you have 2 or 3 students who are not doing well. So it's like setting them aside, because I look at their grades, and those who are not performing well, I call their attention. So parang naging one-on-one na lang yon. Kasi yung buong syllabus mawa-water down dahil lang sa mga nahihirapan sa course na yon. Because you also have a mandate. Yun lang. So it's like pinpointing them. You can tell ang dami kong kasong ganun. And it's just like, number one, at the first day of class, I already tell them I'm really very open to consultation in whatever form, kahit na sa pag-aaral nila o sa family nila. So I can better understand them and guide them. Ginaganon ko na sa umpisa pa. And then pinpoint, you can tell from their performance, and then talk to them, etc. Tapos may kaso rin na talagang with their permission, nire-refer na namin sa guidance counselor. Kasi hindi nga natin pwedeng gawin, baka ano pang gawin sa advice mo, no? Yung ganun tipo. Nahirap talaga.
Thank you for that, Doc Cabrera. Sir Tom, parang ang lalim ng iniisip ni Sir Tom.
Oo nga eh.
Should we ask how are you?
Hindi naman. I agree. No, we should always consider the mental health of the students and the teachers as well. No,
And the teachers as well.
In our teaching of microbiology. But I also agree with Bido. Mahirap kasi malaman who is just using it as an excuse. And sa akin kasi, I really do not have any answer. How do we know who is lying and who is not lying? So the only thing that I always tell my student is, I just trust you enough to do the right thing. So if you are lying to me, well, I cannot do anything about it. It's up to you. As long as for me, I'm just giving you 100% my trust that if you tell me that you can do it because you have some mental issues or you have difficulty, then I accept that. I will consider that. So, mahirap talaga kasi malaman kung sino.
Yeah, we cannot. Hindi tayo magaling dyan.
So the basic idea is to just trust your students, di ba? And develop that trust. Tama sa Ma'am Cabrera, di ba? If you start, I think, sorry, I have to take it away from what Sir Basit was saying a while ago, di ba? You establish the boundaries of the relationship, and then what Ma'am Cabrera is saying that, and you establish early on, if you have a problem, you talk to me, di ba? And then Sir Tom is saying that then put back the trust, di ba? Kasi I think that is what is missing right now, di ba? The students, they have this notion that our teachers don't care. That's why they complain.
Yeah.
They don't know that we actually worry about them a lot, di
Yes.
ba? And so that is what we need to imprint in their mind, that we're here for you, and but we're not only here for you as a person, but also to teach you microbiology or whatever concepts you need to learn, 'di ba? Para makita nila dapat yung roles natin, 'di ba?
Yeah.
And then once that acknowledgement is there, and then you can start teaching, and then you can start facilitating, and then the learning will start.
Yeah.
Let me just share, no. I'm so sorry, I'm not supposed to be the one sharing, no. But when you were
Oh, but please
talking, kasi
share.
I was triggered because I remember a faculty of mine was telling me that there was a student, ayaw niyang umalis ng klase niya. This is an online class. So
It's not.
this student wants to be always in the online class.
Oh, okay.
Yes. Hindi siya umaalis. And then she will find ways and means to extend the class and to keep the class going. Di ba? So eventually, the teacher, siguro nagtataka siya. Di ba? Even if, di ba, when you end the sessions, our others, ang bilis, di ba? It takes a long
Masaya
time for you
sila.
to start your class, biglang mawawala. Online, you just, it's a press of a button away, di ba? So this student, hindi. She comes in early, she leaves late, and she lingers. And then, so the faculty, tinanong niya, and then he found out that the student was going through a lot of things at home.
Oh, yeah.
Oo. And then, you know what the student said? I want to be in your class because once you talk about your concepts, I'm distracted about my problems at home.
Wow.
I don't know if that's a good thing or a bad thing, di ba? But that is true, no? But
Yeah.
that highlights the current reality, di ba?
Yeah.
Oo. May mga ganun eh. Yan. Kaya very interesting, no? But let us flip the idea, no? As much as I think this was mentioned a while ago, I forgot who mentioned it. But we also have to safeguard our well-being. So, kayo ba, how do you safeguard your well-being? Kasi 'di ba, like Ma'am Cabrera, Doc Basit, sis Tom, you're always smiling, you're always, on point on the topic. So, I'm sure you guys umabot kayo sa point na you're so frustrated, you really do not,
Yeah.
you do not feel it. So how do you cope? I guess everyone here would like to know, how do
Yeah.
you do that? How do you cope? How do you keep yourself sane?
Netflix. We just have to we have to remember that we are human beings for ourselves as well, di ba? Yung ganon.
Yes,
Well,
ma'am.
no, I'm not kidding. I really do that, and I'm just
Me
lucky
too.
that I have a wonderful family, and the home is really so nice to stay in. Kasi kung pati sa sarili mong pamilya may problema ka, talagang hindi mo na alam saan ka pupunta.
Yeah.
I think it's keeping ourselves grounded and appreciating what we have. Talaga, minsan we don't We don't
Netflix and
appreciate, we
baby.
don't realize na ang bait ng asawa natin, bait
Narinig
ng
niyo,
mga
ma'am,
anak
yun?
natin.
Sabi ni Sir
Sorry.
B, ano daw, Netflix and baby daw.
Yeah, kasi during the pandemic, that was the time when my child was born. So, ayun. Kaya yung height ng kakitan, di ba? So,
kakaingit ka naman, I'm way past that na eh. Hanggang Netflix na lang.
Ano, ma'am, pag nanganak, ma'am, yung mga microbes yung sa laboratory, you have new babies.
Namamatay na nga eh. No. Buti na sa minus 80 sila.
But that's true, no? The learning to appreciate the simple things in life.
Yeah.
Diba? Learning to appreciate life, diba? That's what keeps us sane, no? Diba?
Yes.
And keeps us going as teachers, diba? Kasi diba, you cannot go to class na ikaw mismo may problem.
Diba? Ang sungit mo nun.
Oo, ang sungit mo sa klase, diba? Yung estudyante pala, ang dami palang pinoproblema. Tapos isipin mo, magsasubmit pa ako kay ma'am, may essay pa siya, may exam.
Well, that's true. It's true.
How about you, Sir Tom? What keeps you sane?
The same din, Netflix. And Disney Plus kasi I have
Teka,
three
parang
girls.
kailangan ko na yata mag-disclaimer. Okay, so let me just clear, no? The things that they're mentioning, these are not promotions, no? Or anything. Kasi kanina we've been mentioned a lot of, di ba? These are just, we are just saying this share, di ba? Our
Yes.
experiences, okay? You do not need to subscribe to Netflix or it can be something else, other
Yes.
platforms. But the
Baka,
point is, find an
Sir
outlet,
Howell,
di ba?
baka dumami
Yeah.
mga anak ng taga PSM dahil sa
Oo,
dalawang yan.
ay, oo nga. Sir Basit, no? Sabi ni Sir Basit, kasi kailangan magkaroon ka ng anak para gano'n.
Pati si Sir Tom, yun din ang sinabi. Tatlo ang girls niya.
Pero actually, you need to find time, new hobbies siguro.
Yeah.
Those are the things na
That's
pwede
true.
mong gawin. Kumbaga, do not just focus on your teaching, your courses.
Yeah.
So, siguro isang bagay na kailangan nating matutunan din is when to make a compartmentalization or when to say no to all those memos and emails na napapadala sa atin, no? And
Yung mga
maybe,
email na yan.
yeah, like for example, I personally try not to work during weekends. So
Okay,
as much as
yeah.
possible. So pag weekends, I don't try to reply to their emails, although syempre hindi mo rin naman maiwasan, no? Pero yun yung gusto ko sana mangyari. And I've been telling also my students that do not send me email or messages on weekends because I try to make it also time for my family. And I also want them to use that time for their family and not to discuss other matters so that are related to classes. Siguro yung isang bagay na dapat nating matutunan din as teachers and probably as students when to send message and when not to send message. Parang gano'n, no? That can help, yeah.
That is true.
Thank you,
Pero
sir,
hindi
for
ko
that.
magawa. Hindi ko magawa.
Oo, ma'am.
Yun, ma'am.
Kasi
Hindi natin
kung hindi ko,
magawa.
kung hindi
Lagi
ko
may
sagutin
tawag
ng
ng
Saturday, Sunday, sa Monday, isang katutak na yung sasagutin ko, tapos may klase na
That's
ako.
true.
Pero that's so nice. Sana Sir Tom pwedeng gawin yan yung bang Saturday, Sunday for family talaga.
So parang ma'am, no? You just have to know your limits. Parang gano'n.
Yes. You have to know. And always do not take your family for granted. No? Lalo na kung ang babait ng spouses, sometimes sila na ang nag-a-adjust sa iyo, which is not fair. Always remember that you also have responsibility to them. Not only to your students.
Ma'am, there's an observation that was shared to me. I can't remember if this was a student or faculty. I don't know if you guys experience the same thing. Sabi nila, dumadami daw yung nagkakaroon ng baby ngayon.
Oh.
Especially among students daw. Natawa si Tawa. I don't know.
Akala ko virtual na ngayon. Ganun ba ka-effective ang virtual? I thought they don't meet their classmates anymore.
They find ways, ma'am.
Talagang napaka-stressful.
Parang mali yata. Parang naging R or X-rated na yata yung mga topic na let us shy away from. Baka ma-censor tayo ng ating mga
ma-misunderstood tayo.
But we're just sharing these things. No? These are,
that's true.
I don't know if you guys, please do share your thoughts on the chat box or through messages to us, personal messages.
Yeah,
you can share.
That would be nice. We can hear from them.
Yes. So, okay. So, ma'am, let us move on to our next topic. Okay. So, our next topic is about laboratory skills. So, I guess, yes, I know. Nakita ko yung reaction. Talagang malalim
yung... si Sir Tom doon, ha? May sinabi na si Sir Tom doon.
Yung nga di ba? So, bottom line. So, laboratory skills should be face-to-facely taught. Is that it?
Definitely. I think for the three of us, I think.
Okay.
But I searched online and I found several universities, University of Florida, University of Nottingham. They offer full online MS in microbiology courses. Full online. So sa lab nila, podcast, laboratory simulation, ganun, no? And I was really surprised kasi I have not changed my mind kahit na ganun sila. I still feel that you have to have hands-on experience. Yung paghawak lang ng petri dish may style yun
Virtual na daw kasi yung petri dish, ma'am. How about Sir Tom, Sir Basit?
Yes, it has to be online. And in fact, we're doing it now in our institution. But, you know...
Ano, sir? Online or on-site?
Sorry, it has to be in person. It has to be in person.
Nagulat ako.
Sabi ko, tatlo tayo pareho.
Oo nga. Si Ma'am Cabrera patingin sa screen.
Sorry. In fact, we have a permit already from CHED to do our internship in person in the hospital, and even the professional courses, we're doing it in person. Yeah. But you know, it's still a limited face-to-face, so some of the courses are still being taught online. But you know, I just shared with you what I was doing during the pandemic. So aside from Labster, so I think everyone is familiar with Labster. Right?
Yes. Si Ma'am Lynn nga was mentioning that in the chat. Well, I'm so sorry I wasn't able to read it. Yes. Yes, sir. Please,
so everyone
proceed.
is, but personally, I'm doing the love vlog. That's the term that I have invented. I call it the love vlog. Wherein there was even this scenario that I even have my blood extracted and do the live the HIV testing. I think I have it here. Share ko lang.
Okay.
Sige nga.
Sige.
Ayan.
Andito dito sir, walang ano to parang
wala.
baka R-rated to or something
Okay. And now, medyo kinakabatin ako. So, eto na. I-prick na po niya ako so that there would be blood. Ayan. Hindi naman po masakit. So, we need to remove the first few drop of blood and make a big drop so that dito tayo para makita nila. We'll make a big drop.
What is that test for?
Yan. And then, can you please show? Yeah. And then, we add 4 drops of the buffer. 4 drops of the buffer.
Sir B, anong test yan?
HIV.
HIV?
Yes, HIV. Screening test of HIV. Okay, and then we wait for 10 to 20 minutes. Okay, so we'll be right back after 10 to 20 minutes. And so, buti na lang non-reactive.
Hindi pinakita yung result, ma'am. Di natin alam.
So, ayun, sa example ng love vlog. So, because there are some, parang nabore ako masyado sa Labster. So, since nakakapag-report, we're able to report on-site. And then I asked my technician, what if we do the actual experiment so that they can actually do it, and they can actually view it, and then explain the procedure. Unlike yung pag mag-watch lang sila ng YouTube videos. So, like for example, di ba, Ma'am Cabrera, what you were saying before, may mga maling procedure, di ba? So at least this time, even if may mali, I'm still accountable naman, di ba? So, syempre, pag mali, we can always edit it out, di ba? You have it critiqued by other people, and ay, mali yung ginawa mo doon. You can have it edited. Unlike kapag sa YouTube, you are at the mercy of the content provider. But this time, I'm doing the love vlog. So, I have several videos of love vlog, and it's like you're hi guys, welcome to my channel. Hindi naman ganon. But you know, doing the procedure that you can actually do in your lab. So at least nabubuksan pa rin yung mga equipment, and then yung mga reagents na ma-expire nagagamit pa rin. So yun po yung ginawa ko during the pandemic. So to augment the laboratory needs. And so ako na mismo yung nakukunan ng dugo, that's fine. So yun po yung love vlog. So I'm also saying to my other faculty members that they can also do it.
Kaya pala 3 araw ka hindi makatulog
So parang Doc Basit, parang you're saying that, you know, there are other opportunities, diba? We're not saying that this can replace the face-to-face, but there are other opportunities to show students to sort of experience what you are experiencing in the laboratory, but virtually, and they can even review it, go back to it, critique it. Like Ma'am Cabrera said, you know, not everything is perfect, so you can utilize this as a teaching moment for critical thinking, diba? Seeing how the student has been able to how do you call this, imbibe your lessons, diba? And see if they are able to spot, you know, mistakes and other things, you know. These
Yes.
are very good points. Yes, yes, Sir Tom. Parang ano, itch na
Yeah,
itch.
hindi, hindi naman. Uh, I actually agree, no, na we should now try to have face-to-face or on-site, at least for the
Yes.
laboratory. But at the back of my mind, there's also is this reality na not all of us are prepared to do this, both for the students and for the teachers. I heard of colleagues who have this feeling na ang tawag natin sa Filipino, agam-agam to to
Yeah,
to really
that's
come to
true.
the university and
Yes.
teach face to face, no? Um, but I think we are all going to that point now. We should, try to open the doors and go back to the lab. But what we could do, of course, is also try to innovate our lessons to make sure that students, if we teach, for example, laboratory courses online, they'll be able to do or lab activities even at home. So let me just share. So since nag-share yung mga slide yung aking mga kasama,
Ating
mag-share na nga rin ako.
mga
Kasi ako na,
prepared talaga, no? Sobrang
Oo, no?
prepared ang aking mga guests.
Nakakainggit, ha?
So I hope you could see this. So this is actually a home-based experiment that we did.
Okay.
This is done by my students. We work with slime molds, and we use slime molds as a model organism to teach basic ecological concepts. So we come up with a paper that's published this year in the journal Slime Molds. It's a free access, online access journal. And in this activity or in this experiment, we provided some information on how teachers can use mixomycetes for home-based experiments. So we use these materials which they can easily buy from Shopee or from Lazada. Yung camera phone, we have this digital pH meter. Hindi siya accurate, but I think this will work. And then we try to use also materials that are available at home or equipment that are available at home. For example, we use pressure cooker to sterilize the materials. And we read this paper that pressure cooker can actually replace or at least works at par with laboratory autoclaves in sterilizing materials. So we use that one. And in this paper, we also provided some lesson plan which the students, which the teachers can use in implementing this home-based experiment. So the idea here is to come up with experiment that the students can do at home using materials that are available at home, using equipment that are easily, or that are common in our home place. I also try to develop this another do-it-yourself learning activity in mushroom identification, since I teach mycology. So we implemented it in our course in mycology last semester. So the idea in that do-it-yourself learning activity is that since the course will be deployed online, the students will identify mushrooms. Okay, I'm going to turn this off now. So they will identify mushrooms using virtual specimens. So we provided them with mushroom pictures. We make cross sections, put some rulers so that they can measure the size of the mushrooms and things like that. But we don't end by just working with these visual specimens. They have to really experience doing it. So aside from these virtual specimens, the students need also to practice or apply what they learn at home. So they have to collect mushrooms in their backyard, and they have to identify the mushroom using the skills that they learn from the virtual activity. So I think the challenges for us teachers really how to create innovative learning strategies that our students can do at home to develop further their technical skills while we are still, not implementing a full face-to-face or on-site laboratory activity or classes. Yeah,
Thank you, Sir
yun po.
Tom. Yes, very interesting, no? The points that we're seeing put forward, no? Uh, so the importance of innovating, di ba? I think Ma'am Cabrera was the one who mentioned this before, that there are a lot of resources online. Okay, some of these are being put forth by our panelists right now, but there are a lot more of this. You just have to explore it. Get over the fact, get over that, that whatever it is that is keeping you from from looking into these things. Check the veracity of these things, right? And then you can innovate, right? Innovate, and then demonstrate it to the students, like what Sir Basit is saying. And then, you know uh it's like showing the students how you as a teacher are able to innovate, and they themselves, as your students, may learn that, parang teaching by learning, di ba? Parang, I don't know if that makes sense. Teaching by doing pala, sorry, not by learning.
Teaching by
Baliktad.
doing.
But
But
teaching by doing.
you
Yes,
also
you
teach by
learn. Yeah.
learning.
Yes, I'm so sorry because I was thinking of us as teachers, but to the students, diba? It's you're teaching them by doing it. You're doing it, and then you're showing how you're innovating, diverting from the normal, diba? And then teaching them, oh, you can innovate, and you can find new ways of doing the same thing, and it will live up to par, diba? And then, and also demonstrating to our students that, if I can do it, you can do it. It means that,
Yeah.
you can adapt, you can be resilient to the present times, you can get over your mental, uh, how do you call this, challenges, whatever they may be. So I think that's the takeaway that I'm getting. But though we are still, I think everybody's saying that we still need the hands-on experiences. But these hands-on
Yeah.
experiences can come in different forms and sizes. Then the technicalities are still there. That doesn't change. But it doesn't mean that, you should be limited to this, di ba? The possibilities are there. You just have to innovate. So I think that is what, Ma'am Cabrera, Sir Tom, and Sir Basit is saying. No? We have to innovate. So, nandiyan na nga tayo. Online cannot teach the skills of laboratory. Yes, we are
Oo.
there. But you can utilize it to augment the things that you teach in the laboratory. And you can use this as a teachable moment for innovation, for learning to be adaptable, by learning to be resilient to the circumstances. I don't know if I got tama ba ang message? Baka mali ang message na pipick up ko. Yes, thank you very
You
much for that.
summarized it very well. You synthesized it very well.
Yes, thank you very much, ma'am. Now, may uno na ako.
Oo, 4 sa Lasal.
4 pala, sorry, baliktad pa.
Sa Lasal,
Yes po, 4 pala yun, hindi pala uno.
Sa UST. Sa
Oo,
dalawang
sa
yan.
UST uno.
Si Sir Tom at saka si Sir Basit.
So, ma'am, that brings me to the next, our next topic. So, quality assessment, ma'am. So, what is quality assessment now? Kasi, ma'am, before, when you talk about, I remember, I don't know if you remember this, a few years back, we discussed this I think you were pondering something about your classes, and then you questioned me about it. Parang, but I saw that you were just bouncing your ideas. You were thinking about this. And then, the quality of assessment that time was multiple choice exams. That is parang the quality of assessment. But right now, what do you define as quality assessment ba?
With the LMS that we have, when you talk about assessment, it's not limited to exams alone.
Okay.
Like, you can have discussion boards, and then you post questions there for them to answer on their own. And then you check for plagiarism. Di ba? Okay. Okay, you can have them in groups. Di ba? During your class, you can have breakout rooms. You can raise a question for them to discuss and interact with each other. And after that, they report. So you can give each group a different question para hindi pa ulit-ulit yung reporting nila. So that is also one way that we assess. And for graduate class, mas madali kasi mas maliit ang klase. Kaya madali magbigay ng essay questions. Ang mahirap kasi pag malaki ang klase, lalo na kung ilang klase yan, tapos essay question, talagang Dr. B, baka isang linggo ako hindi makatulog. Hindi lang 3 araw. Yung ganon. Pero talagang maraming limitation ang multiple choice. Kahit na sabihin mong, oh, you can have your multiple choice questions in such a way that you have synthesis and critical thinking. May ganon din eh.
Okay.
May ganon din eh.
Yeah.
But at the same time, sasabihin mo, then I have to lengthen the allotted time. Kaya, nandun na naman yung fear mo. Anong ginagawa nila sa long duration? Naghahanap na ba sila ng sagot sa internet? Kaya ang hirap mag-balance pag online. Pag in person, pwede ka magbigay ng mahabang oras eh. Sa ganyan. Kasi nandun ka eh. So, yun lang. In short, assessment is not limited to exams. You can have it in different forms.
So ma'am, you're saying na when you say essay question, you're not, is this the same as saying like a problem-based method or different?
It could be a problem-based.
Problem-based is just one kind.
Oo.
Yes,
I don't know how you define problem-based. For example, in medical microbiology, you can talk about a patient with this, yung ginagawa natin, Dr. B, a patient with the following clinical pictures and the specimens submitted, what should you do? Now, after the gram stain, you found them to be gram-negative bacilli. What would be the next step? May mga ganon. So they have to put everything. And then you vary it para hindi nila minememorize lang. Kaso, ang daling mag-search sa internet. Ganon.
You know, ma'am, I have something to share with you. You actually know the person, but I will not say it on the screen. So, you know, when I was a grad student, you know, there was this teacher lumapit sa akin. Galit na galit sa akin. It's galit na, it's not galit na, you know, galit na blambing na galit. Diba?
Malambing na galit.
Sabi sa akin na, you know, I do not know if you know it or do not know it. Kasi he or she gives, I'm not going to say who, para cannot be guessed. He or she gives essay questions. And then inis daw siya sa answer ko. Kasi ang ikli-ikli daw, kung pwede ko daw sabihin yes, no, yung answer, lalagay ko daw yun. Tapos baliktad daw, siya yung parang tinetest kung ano bang grade bibigay ko dito. Kasi parang alam niya na I know this answer, but hindi niya alam kung I really know it or I just guessed it. Sa sobrang ikli daw ng answer ko. Yes.
Hindi mo in-expound.
Yes. So yung nga, ma'am. So when you say essay questions, how do you measure the knowledge that the students give? Is it the length? Is it the like what you said?
Ang sa akin nasagot ba niya yung tanong ko? Yun lang naman eh.
So it's not the length. It's kung nasagot mo yung tanong.
Yeah. In fact, pwedeng mahaba ang sagot, wala akong bibigay na points, sinayang mo lang oras ko. -5 ka dyan.
Walang sickness factor.
wala yung effort.
Walang effort. It's really if the question was answered.
Kung nasagot o hindi.
Okay. Very good. Very good. Thank you, ma'am, for that. So how about sino ba, Doc B or Sir Tom? Yes, please go.
I actually agree with Ma'am SP, no? So I think as teachers now we should realize that assessment is not limited to just exams. Before the pandemic, no, this is the initiative of Ma'am Sean Raimundo. She spearheaded these training workshops on OBE. In microbiology. So I think Ma'am SP, you're part of that, right? So we've been giving seminars all over the country, and I participated in a couple of those training workshops. And the idea here is because we wanted microbiology to be obedized. No, this is because this is what CHED is mandating the universities now in teaching all the courses. And when we talk about OBE, we should realize that in OBE, you need not only test or you need not only assess using the traditional paper or pen and paper assessment. There are other ways by which we could do that. Like if they will submit a project, you could use it as a form of assessment. And personally, I feel na maganda yung mga tinatawag nating real life assessment. No, for example, I attended again another course here in Germany. And in that course, the students have to do a research work. All right. So they have a lecture activity where they will learn the principles of the topic. And then after that, they will do some laboratory experiments. And their assessment is not an exam. Their assessment is an oral presentation of their research topic. And to me, it makes sense because the idea there was to teach them how to do research. And so when you give them an assessment of like define what is research or what are the different parts of a research paper, you are not really assessing their ability to be a researcher. But if you ask them, okay, make a presentation of your research output, this is what researchers or scientists are doing. So a good way of assessing their experiences as well as their knowledge of what research is. So yun yung sa akin.
Yeah.
Thank you, Sir Tom, for sharing that. No, very interesting. No, the need for performance tasks, di ba? I think some people they call it performance tasks. But sir, one thing, no, I think I've encountered this before. I cannot remember again. Hindi ko sasabihin ko sino. Baka mamaya sabihin chichismis ko mga other faculty and other baka nandito nga yung tao. So, but anyway, sir, iba in performance task. One big problem here, how do you translate it to a grade? Because I remember I heard this complaint no before na. Sabi ni isa, lahat ng students ko uno because they were able to perform. So sabi ko, how is that possible? Ibig sabihin, ang dali-dali ng subject mo, di ba? Parang ganun. So Sir Tom, how do you translate this performance task to grades?
Yeah, that's why it's very important to create a very good rubric, no grading rubric. I think yun yung comment ni Eds.
Yeah,
so
yeah.
I was reading yung comments. Yeah, so we should create a rubric that will really reflect yung ability ng mga students, no? Tulad nga sinabi ni Ma'am SP kanina, marami mga online resources and that's also true for creating rubric, grading rubric. So you can just go to, type rubric there. And you can find online materials that will help you how to create rubric. But the challenge is for us is to really make sure that the rubric that we create or we adapt really reflects the skills developed, for example, by our students. So kung hindi maganda yung rubric natin, then we will not be able to evaluate clearly the performance of our students. Ito yung sinasabi mo nga na eventually all of them will get grade of 1 because all of them submitted the output.
Yes.
So for the simple simple idea that they submitted an output, may grade na sila. But if we create a rubric that will really scrutinize their output and not only the output but the process itself. No, paano ba nila ginawa yon? So you can also grade that. Yun nga lang, it's very challenging on our end as a teacher.
Very difficult.
Very difficult, really.
So, sir, do you agree with Dr. Cabrera? Kunyari, nag-submit yung tao. Kunyari, nagbigay siya ng 30-minute video. Di ba? But then you feel na, ay, hindi naman nasagot ang tanong ko. So, 0 yun. Do you grade for effort? What do you think, sir?
It depends, I guess, sa rubric na lalagay mo. No? And think, what is important is you tell them ahead of time the criteria. So if, for example, you said then, okay, there will be no grades for effort, then their students need not complain if you give them a grade of 0 because the output that they submitted is not what you want them to submit. So okay lang yun kasi at the beginning na-inform na sila na ito yung ating policy, and that's very important, I think, in this kind of assessment.
Parang sinasabi niyo, sir, that unlike kasi before when you give multiple choice questions, true or false questions, di ba? Questions like this, like the essay form or the performance task, you have to give clear instructions and to explain the students what I am expecting. So if you don't achieve this expectation, do not expect a grade, di ba? From this. Parang tama ba, sir? Yun yung sinasabi niyo, di ba? Yes. Yes, thank you for that. So I think si Basit may gusto i-share ata. Regarding the grading, sir, quality assessment. What is it right now? Ay, sir, naka-mute ka. Ay, minus grade yan. Walang audio. joke lang.
And so I think whatever the platform is, whatever the modality is still the traditional alignment of the intended learning outcome to the teaching, learning activity, and assessment.
Hmm.
So the teaching learning activity and assessment may vary, especially now that we are in the online platform, but the intended learning outcome remains the same.
Hmm.
Meaning to say, for as long as you're able to achieve the expected exit competencies of a graduate of a certain program, and for as long as you're able to ensure that during that assessment during the assessment you're able to achieve the authentic assessment, then I think you have done something as an educator. However, here's the challenge. Nowadays, TLAs, or the so-called teaching learning activities, vary. However, some of the faculty members, especially those who are not trained for the digital for the digitalized platform, would still be sticking to the traditional part. It is as if they're still in the in-person classes. But doing the traditional lecture, synchronous lecture for the long time, for the longest time, for the most of the 3 hours, may not be effective. Katulad nga na sinabi ni Doc Cabrera and ni Tom kanina, we have to dig deeper. And the situation calls for a different scenario. So perhaps we can be more resourceful and more artistic for that matter on how are we going to achieve that. And then another problem is the assessment. So it's so easy to cheat. We have bought Respondus lockdown browser, but it has become a white elephant. Ang sobrang mahal niya, but we never use it. You know why? Because there are many clamors, there are many petitions, and there are many bad uh, uh, bad reputations being created by fake news peddlers that Respondus would even destroy their unit and all. So in the end, what we did was, the very crude take your exams at the back of the mirror so that we can see your hand and your screen, yung the so-called online live proctoring. So there are many challenges, but if we're going to think of it, no matter what the modalities is, in order for us to achieve the outcomes-based education, we have to align the intended learning outcome with the TLA and assessment. However, the TLA and assessment are two important factors that you really need to synchronize so that at the end of the day, you can actually see that you have achieved the intended learning outcome. So yun yung take off for that matter. So that's the reason why during the pandemic, I was able to publish a paper on the alternative forms of assessments. So there are several alternative forms of assessments aside from the usual traditional pencil and paper. Katulad nung sinabi ni Sir Tom kanina, we can do the mind mapping, case-based pedagogy. So there are several ways to check the achievement, the formative and summative achievements of our students. However, you have to go extra smile, tama, and extra mile. Extra smile kasi nga you have to have a positive outlook or attitude regarding this matter. But yun nga, unlike kapag multiple choice, the LMS will check the paper for you, di ba?
Yeah.
But di ba, parang wala na masyadong effort, then you can just export it to your Excel grading sheet. But kapag may mind map ka pang nalalaman, may essay, and then may mga rubrics ka pa, it's gonna be an extra effort on the part of the faculty members. And imagine, you're handling several sections. Each section has 40 students. Wow.
Yeah.
So, masalimuot ang usapan natin to.
Parang
Yes,
ayaw pa
sir.
na magturo
Naku, ma'am, wag niyo gawin yan, ma'am. Stay away from the light, ma'am.
Stay
Joke lang yun. Yung tama, Sir Bassett, no? Very interesting, no? But many people, I think many teachers, this is what they struggle with. Tama yung sinasabi mo mga principles, guiding, evaluation, and assessment, right? But their difficulty now is, how do I explain this in simple terms to students so that they will understand? Diba? Kasi a lot of the things you said, these are understood by the people here because they're teachers, they're used to the terminology. But students, hindi ganun eh. Diba? Ang students nga, once you start talking about mga objectives, biglang nag-zone out na sila eh. So how do we as teachers, like sabi nga ni Sir Tom and Ma'am Cabrera, set the expectations at the start of the day or before your exam in terms that they will understand. Na at the same time not threatening, 'di ba? Kasi minsan 'di ba ang ginagawa nila, sabi natin, pag hindi niyo sagutin 'yan, zero kayo, babagsak kayo. I mean, how do we parang cross the divide, 'di ba? Parang gano'n, Sir Basit.
Well, tama yung sinabi nila that you have to set your expectations. You have to
Yes.
set the expectations. Kaya nga mahalaga yung orientation during the first day of class. And
Yes.
let them realize why they are enrolled in this course. Is it just for a mere course requirement, or is it something that will contribute to their lifelong learning in their future profession? So if they're able to realize it, even without using these intricate terminologies, then I think they will find their class very interesting. However, if their purpose is just to pass it and get a degree, then they will find all ways, just so to pass your class, even without assimilating the important and relevant things about it.
Tama nga, no? Tama nga, Doc Basit, no? You connect it to what they want, di ba? What do they want to achieve? So, if you know what they want to achieve, then you explain it in terms that they will understand. So, connecting the outcome, di ba? I think that is the whole basis of the outcomes-based education, di ba? You look at the end product. When the person graduates, when the person is already on the workplace, what do you expect the outcome to be from all the learnings they get from you? So if you bring that to the student, di ba? Na if you're already working, or what is your dream, what do you want to do? So how will you achieve that? And that is what you need to answer in my exam, or my questions, or my essay, or my performance, or mind map, di ba? And etc. etc. Or like sabi nga ni Ma'am Ma'am Eloreta, yung case studies, di ba? Or problem-based learning. So these are different methodologies, but the bottom line is we need to explain to the students, we need to let them understand what we're expecting, and then we need to be able to connect this to the outcome, but not because this is the outcome of my subject, but put it to them that this is an outcome you need, di ba? That you will need later on, whether it is just you want to pass the course or you want to achieve something in your life, di ba? Parang ganun. So that is really how the outcomes-based education is hinged on, di ba? Letting the person realize that this is your objective. This is an outcome that you will achieve, di ba? But you will only achieve that if you're able to answer my question, di ba? Parang ganun yun eh, no? So very interesting. Thank you, sir, for that sharing. Parang,
Yeah.
no?
Sir Howell,
Yeah.
I agree with Sir B.
Yes.
You make it clear to them. I always tell my class, I take my teaching very seriously. So you have to take your learning very seriously. You're not in my class to pass or to get a high grade, but to learn from it. And by learning, I mean that you still remember what you will learn from my class. You don't have to Google it every time. You might forget it, but then when you have to recall, it would be easy for you to understand already because you have understood it in my class. And I always tell them real time. Kaya meron tayong synchronous class, doon mismo naiintindihan mo na. Huwag mong hintayin na papakinggan mo pa recording para maintindihan mo. You just tell them that you're really serious. Pero syempre, kasama yung mga facial expression kasi feel mo yun eh. Feel mo yun.
Tama, ma'am. Di ba parang it's like, di ba? Many students, they ask that will we have attendance? Do you check attendance?
Ay, oo.
Yes, di ba? But they don't get it that it's not the attendance that's important. It's the fact that you experience these things with me, and you are one with me in terms of you want to achieve this knowledge, di ba? At the end of it, eh. So the attendance is merely a tool that we are using, but it's not the end, di ba? Kasi I remember that argument, eh. What if nawala yung estudyante kasi nawala ng internet connection, di ba? But the person is religiously following you asynchronously on all the class activities that yet achieve the same outcome in the end, di ba? So attendance now is a very I know this is another topic. I think this is not
Yeah,
a
it's
great
another
topic.
top. Yeah,
So sorry, but I had to mention it kasi nakita ko yung
yeah,
connectiveness to our the
that
what
is
that we're
true.
yes we're
And
saying
Ed's
right
here
now.
has
Oh yeah, yeah.
Ed's in the chat. He has
Yes.
a relative relevant comment.
Yes, yes. So we have to emulate the values we want them to imbibe.
Yes.
We have to set good examples. Yes, tama,
Yes.
Sir Geronimo. Uh, uh, we parang we demonstrate like what Sir Tom was saying right na kanina, 'di ba? We do it and or yung sinasabi ni B, napapakita ko yung video. So pinapakita mo what they should do or they should not do. Like Ma'am
That
Cabrera
is
saying,
true.
it's not necessarily what they should do eh. Pwede rin yung what they should not do. 'Di ba? So we hone their critical thinking, innovation, you know, and et cetera, et cetera. No? The skills, 'di ba? We're
Yeah.
developing, not bounded by simply the face-to-face or online. So it's not an argument actually of face-to-face online, whether you pass the exam or not, but it's more of, did you learn anything from me? Parang ganun eh, di ba? The more basic question was that eh. Did you learn anything from me? If you didn't learn anything from me, why should I give you a grade of 4, di ba? Parang ganun. So, thank you
At saka
very
if
much.
I may add ha,
Yes, ma'am.
kaya very
Yes, ma'am.
important pag nagde-deliver ka, di ba interactive yun eh, no? Pero madalas wala kang nakukuhang sagot. Ikaw lang
Yeah.
nagsasalita. No? But still, kahit na ganun, you still have to show them that you're enthusiastic kahit na feeling mo napakaganon na, no? You still have to show them because like I've heard my students complaining that one of the faculty just reads from his PowerPoint.
Oh,
Diba?
yeah.
Meron ganun eh. May PowerPoint tapos nandun na nakasulat lahat, babasahin na lang, eh nagturo ka pa. Kaya you have to show na ikaw bibo ka rin para sila rin bibo rin.
Yes, yes.
Hopefully. Yung ganun.
Totoo, ma'am, yan, di ba? That is true, no? That's a common comment na sabihin na, ay, naku, sir, ayaw ko makinig kasi binabasa lang naman niya yung PowerPoint niya. Rehearse na yung speech na yan eh. Parang gano'n, di ba? Yan, di ba?
Thank you, Ma'am Minette.
Yes, thank you for a very enthusiastic sharers in our chat box. Parang kasama sila sa ating mga talk, no?
Kung pwede nga, pasok na sila sa discussion natin.
Oo nga,
Si
yes,
Ma'am Minette,
yes.
si Ed, si...
Even Ma'am Raimundo earlier and then
yes,
Ma'am
Dr.
Colette,
ACR.
Yes.
Marami-rami din sila.
So anyway, I'm so sorry. We are running out of time.
Okay.
So we are at the end of this discussion, but not the end of this, concept, because as they say, learning is lifelong. So we are the four of us is just implanting the seeds of ideas to you. You can continue to interact with us through to chat, through email, and etc. We just don't guarantee that we will answer you during the weekends. Joke lang yun.
I will answer but not Sir Tom.
Yes. Yes, pag weekend, wag kay Sir Tom. But take note, iba ang weekend ni Sir Tom kasi iba ang time zone niya.
Oo naman. Anong oras sa'yo ngayon, Sir Tom?
12 p.m. po, lunchtime.
Magla-lunch na pala si Sir Tom.
6 hours, 7 hours, delay lang naman.
Okay.
Time.
So anyway, so we are at the end. So we would like to give everybody a time to collect their thoughts and give their main takeaways on what we discussed today. Maybe they also learned something they would like to share to the audience, or maybe give some inspirational message, or maybe add additional sharing, because it was very interesting, no? Let me just share to the audience that when we started talking about this, we did away with the formal presentation. I'm sure sawa na kayo sa mga lecture, but very interesting that our panelists, they were still able to show us some materials even they were not prepared for this. These are biglaan. Kasi akala nyo they were prepared eh. Or pinag-usapan namin, oy, Ma'am Cabrera, kunyari magpa-present ka na ganito. So these are not contrived.
Hindi mo alam.
These are biglaan na sharing. So I think that's what is needed by a teacher right now, no? But so let me now give the floor for main takeaways. Maybe we can start with Sir Tom. Sir Tom, would you like to start the summarizing or the synthesis of what you realized from our talk today, this afternoon?
Yeah. Thank you, Howell. Thank you also to PSM for this opportunity. This is really a wonderful experience. This is my first time to be involved in a discussion forum. So
Oo.
nakakakaba talaga, no? Kanina pa ako kinakabahan eh. But I enjoy the moment. Medyo relax lang, no? Nung meeting natin, medyo seryoso tayo eh. So at
Yeah,
least ngayon
yeah.
hindi tayo masyadong seryoso. Now, I just realized that online learning is really not that bad. There's always the beauty in teaching online, and I think
Okay.
siguro ito yung isang bagay na dapat natin din makita.
Mm.
Since I think online learning will definitely stay uh, with or without any
Yeah.
pandemic or catastrophe, no, but definitely it will be something that we'll be using in our classes. For me, the beauty of online learning is that it provides us, it gives us some power. So for students, it gave them the power control, so they can learn when, where they want to learn, how they want to learn. So that is the power that they can get with online learning. And for us teachers, I'm trying to think of a term, but the item that I have here is the power of management. Hindi ko alam kung tama yung word na management. This is because as a teacher in an online learning setup, we have the ability to manage our classes. No, of course we do that. We manage our classes even in a face-to-face setting. However, in a face-to-face setting, we usually are limited by what is available within our classroom. Okay, so if we don't have a projector in our classroom, then we cannot teach with our computer, and we use the blackboard and the chalk or the whiteboard and the pen. So we are limited by what is present in our classroom. But in online learning, we don't have that limitation, and I think that's what online education made or gave us. It removed all these limitations, and therefore the challenges for us as teachers is how to make use of this new power that we have in order to progress or to advance our teaching, particularly of microbiology. So maraming salamat sa PSM for this opportunity.
Thank you very much, Sir Tom. So let us now hear from Dr. Basit for his main takeaway from today or tonight's discussion or this morning's discussion kasi iba-ibang time zone
Oo
tayo.
nga.
Okay. First of all, thank you, Howell, and of course, PSM, particularly, especially to President Donna. Yeah, so congratulations in advance. So for me, my main takeaway is that nothing replaces the traditional way of teaching microbiology. So we miss the smell of the agar in the lab, or we even miss the noise of our students in the lab.
Yes.
Okay,
Yes.
so it has been 2 years since the pandemic. To say that you have not yet adopted these past 2 years on the context of evolution, I will surely say to you that you are a candidate for extinction as a teacher. So the key word here is adapt. What's in your
Yes.
hand? Make the best with what you have. And then, of course, empathy is very important. Try to be in the shoes of your stakeholders. So when you say stakeholders, not just your students, the administrators of the schools, the parents. In this particular situation, we had this research measuring the perceived 21st century skills of the students when they are in online internship, campus-based internship, and hospital-based internship. So based on the findings of our study, online internship ranks last, but
Yeah.
students still scored it fairly as an opportunity to get 21st century skills. So it's not really that bad at all. So all you have to do is to look at the bright side of it. So opportunities during online learning from my personal viewpoint. Well, I can share a few. First, my enrollment increases. So that's one. And then uh, board rating is still stable. And in fact, I even produced a top notcher last March 2022. So I was able to invite speakers and lecturers from different parts of the world. Okay, so I have adjunct professors from Saudi Arabia, Canada, and USA right now in my pool, in my roster. Students were able to join and win international research conferences without paying for airfare. So there are several bright side of it. So really, the pandemic makes the world borderless, right? So let us also take it as an opportunity to make an education borderless as well by designing courses that will allow seamless assimilation of knowledge and skills being offered by the world or the universe rather. Thank you very much, PSM.
Thank you, Doc Bassett. So let us now hear kay Doc Cabrera.
Oh yeah, it's so intimidating, huh? After Sir B and Sir Tom. Well, of course, I'd like to say thank you to PSM. I'd like to say thank you, Sir Howell. You have taken care of us during the preparation for this particular talk. And my take is online classes will be around. It will be around with or without COVID. It will already be part of the educational system. So we have no choice with its limitations. We just have to embrace it, accept it. As B was saying, we'll be extinct if we don't do so. And so it can be effective if handled correctly, except for the lab portion. There are so many limitations there. So whether it's online or in-person class, us as educators, whether it's in microbiology or in other courses, we have the responsibility to our students. So we still have to continue updating ourselves, no matter how old you are. Update yourself and be dedicated, be dynamic, and that would make you an effective teacher. And that is what you should really be doing as an educator all the time, no matter how tiring it can be. Aside from that, aside from taking care of our students, we also have to take care of ourselves. Talagang nakakaweng-weng magturo ng ganito. It's really so demanding because we have so many roles. We're not just teachers. We have our family as well. So do not forget your family. It's very important. In my case, they come first, whatever. And it's just that we have to be thankful for what we have, which sometimes we forget. We forget how blessed we are. That's it. Thank you.
Thank you, Ma'am Cabrera. So thank you to all our speakers. So I would like to take this opportunity to award our panelists with certificates of appreciation. So can we flash the first certificate? Yes, so we would, the Philippine Society for Microbiology, in cooperation with Trinity University of Asia, awards a certificate of appreciation to Dr. Thomas Edison E. de la Cruz for having served as speaker during the pre-convention activity of the 51st Annual Convention and Scientific Meeting at the Philippine Society for Microbiology Incorporated with the title Mu Talk on Microbiology Education given this 20th day of July 2022. Signed by me and Ma'am Donna. So thank you very much, Sir Tom.
Thank you very much, Sir Howell.
So similarly, we would like to also present this certificate, not the same certificate, different certificate. Iba yung pangalan kasi baka akala nyo parehong certificate. Magkaiba pong certificate. But the same content in terms of the idea of it, okay, to Dr. Supacha A. Basit, and to acknowledge his contributions to our Mu Talk on microbiology. You know, after this Mu Talk, parang I'm looking forward to the next one, no? So, thank you very much, Dr. Basit. And again, also, we would like to award a similar, again, not the same, certificate to Dr. Cabrera. Note that this is virtual, but we will give you, of course, a certificate later.
Thank you.
Later on. So we would like to present this also certificate of appreciation to Dr. Esperanza C. Cabrera for sharing her knowledge, you know, and inspiring us to be better teachers, di ba? Parang in-encapsulate ni Ma'am Cabrera at the start, sabi niya na, if you cannot hack it as a teacher, then don't.
Ang sungit, ano?
But thank you very much, ma'am, for gracing us with your thoughts and ideas. Ano, para nakaka-enlighten, even though, you know, it's at the end of the day, you know, but it's like as if this is the first, you know, topic for the day, you know. Thank you very much.
Thank you.
Yeah, and so thank you very much. So we have Ma'am Donna, the president of PSM. Ma'am Donna, do you want to say a few words?
Gulat naman ako. Can you hear me po? Sorry.
Yes, 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 of course.
Yeah, sorry. Just would like to thank, of course, our speakers this afternoon, this evening actually. Of course, Ma'am SP has always been an inspiration to me ever since my master's. Na panel ko yan ng MS and PhD ko. So talagang I've really learned a lot from Ma'am. And
Masungit ba?
hindi ko makakalimutan lahat ng tips ko ni Ma'am. Kahit na, of course, syempre face to face, but of course, from the comment kanina na, I forgot yung comment kanina, pero may shout out kay Ma'am kanina. So I really like to thank Ma'am SP also. And of course,
Thank you.
Sir B, of course, a good friend of mine. I mean, we've been classmates, diba? Classmates po kami. And yes, I think I've seen the growth. I mean, sobrang grabing yung growth. And Sir B has always been also a good friend, and sobrang from the start of our careers hanggang sa right now. No? So, sobrang, and kanina yung sinabi nga niya na, I mean, he has invited lecturers and nagbo-board top notchers yung mga students nila. These are all very inspiring, no? Kasi the challenges of the pandemic, you always think that there's a challenge. Pero it's such a good point yung sinabi ni Sir B na yung kailangan, let's look at it also in a different way. So that's really very inspiring, no? So thank you, Sir B. And of course, Sir Tom, of course, one of my mentors, mentors for sa PSM, and like with Ma'am SP as the this microbiology education with teaching awards, and has always been an inspiration to me kasi I know it's very challenging for Sir Tom also. He has been teaching from Germany. For us kasi, here in the Philippines, we are already challenged with the online classes. But I have seen Sir Tom also teaching from Germany. And he has inspired a lot of our students. And hanggang ngayon, working yung laboratory niya, he has always been monitoring his students. So balancing between teaching and family, of course, important ang family always, but balancing everything. And I've seen that kay Sir Tom. So thank you so much to our speakers, and also, of course, to all our participants this afternoon. Ang saya-saya, nagbabasa po ako sa chat, and it's a good thing, katulad nga sabi ata ni Maminat kanina, maybe we can also do this again next time. And Sir Howell, congratulations. Thank you so much for making this a very interactive and very happy. Saya-saya po nung talk. Hindi siya yung tipong, parang, ayoko nang makinig. It's not like that. It's just I'm always waiting for the next topic and the inputs and everything. So thank you so much po sa inyong lahat. To everyone, maraming salamat po.
Thank you very much. Thank you very much, Ma'am Donna. Thank you very much to everybody. Thank you very much also to our participants. This discussion would not be possible without you guys, kasi it's really the inputs and your interest in this topic that really drives us with this discussion. Please do take note that tomorrow will be the start of the convention. Para hindi pa pala nag-umpisa, pre-con pa lang to. So, but please do take note that tomorrow in the convention, to attend it, you have to go into the website. There is no Zoom link or anything. Everything is done through the website. And please do remember to also fill in the evaluation form that will be posted. Oh, it's there already. Ang bilis. Ang dami pa. Evaluation form that is posted. And then please enjoy our exhibits. Our sponsors really took time and effort to put things together online. Parang sila gumagawa ng online class din. They have activities. These are hidden throughout their pages. Please do take time to visit them and leave some comments and ideas. And also to our poster presenters and paper presenters, there's a poster exhibit. Please do visit and leave your comments as well. So thank you very much to everybody. Thank you. And I hope you have a good evening lunch or dinner or kung ano man yung time zone ninyo. So that is it for today.
So I hope we were able to enlighten your and brighten your evening or your morning.
Thank you. Thank you so much.
Thank you po.
The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors. The 51st Annual Convention and Scientific Meeting is made possible by our major sponsors, Procter & Gamble, ESCO, 3M Philippines, and Defensil, and our minor sponsors.